behalf of the Division of Microbiology and Biotechnology, Yenepoya Research Center, we welcome you all to this inaugural session of National Conference MiCON 2025, Divergence and Surveillance of Microorganisms. I now request Dr. Rekha PD, Chairperson of MiCON 2025, and Dr. Bhagwan Rekadwad, Secretary MiCON 2025, to escort our dignitaries onto the dais. Before we begin with our proceedings, I invite Mr. Amjad Hussain to lead us in prayer, seeking divine blessings and inspiration.
Good morning, all. أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. أولم ير الذين كفروا أن السماوات والأرض كانتا رتقا ففتقناهما وجعلنا من الماء كل شيء حي أفلا يؤمنون وجعلنا في الأرض رواسي أن تميد بهم وجعلنا فيها فجاجا سبلا لعلهم يهتدون وجعلنا السماء سقفا محفوظا وهم عن آياتها معرضون وهو الذي خلق الليل والنهار والشمس والقمر كل في فلك يسبحون. I will go with the translation. Do those not believe, don't realize that the heavens and earth were once one mass? Then we split them apart, and we created from water every living thing. Will they not then believe? And we have placed firm mountains upon the earth so it does not shake with them, and made in it broad pathways so they may find their way. And we have made the sky a well-protected canopy, still they turn away from its sign. And He is the one who created the day and the night, the sun and the moon, each traveling in an orbit. Thank you.
Thank you, Mr. Amjad. Let us commence this conference by lighting the lamp of knowledge and wisdom. It's our honor to invite the dignitaries on stage to light the lamp. With this luminous start, we now officially declare the opening of National Conference MiCON 2025. Now I request Dr. Rekha PD, the organizing chairperson, MiCON 2025, to deliver a welcome speech.
Good morning, all. It's a great pleasure for me to see the participants, dignitaries who have registered for this MiCON 2025. As some of you may be aware, we have been hosting this from 2021 in the series to bring all microbiologists and biotechnologists and healthcare workers to the same platform to deal and to discuss about the developments in the field and exchange the ideas that each one of us have on this field among ourselves for the greater good of the universe. With that, respected dignitaries, colleagues and participants, as we gather here for the National Conference MiCON 2025, which focuses on the advancement of our understanding about the role of microorganism in fostering sustainable future, we aim to achieve substantial progress in utilizing microbial potential for societal benefit and tackling obstacles through the knowledge exchange, cooperation and innovation. I take this opportunity to welcome all of you gathered here to this conference. It's my privilege to welcome Dr. Sharmila Mande, Ayush Distinguished Scientist Chair, Ministry of Ayush, Government of India, who is a visiting professor at IIT Kanpur and IIT Gandhinagar, is a consultant to TCS Limited as chief guest and keynote speaker for this conference. Welcome you, madam. Yeah, please. May I request Professor Shripati Rao to hand over a bouquet from the university and the sapling for your presence. Yeah. 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 Thank you, sir. Thank you, madam. Yeah. I warmly welcome Dr. Tanaji Kudre, Principal Scientist, CSIR, the Central Food Technology Research Institute, the guest of honor and an invited speaker for the conference. I welcome you, sir. He has traveled all the way from Mysore here, and I request Professor Shambat, sir, to kindly hand over the sapling and a bouquet. Yeah, 1 minute, yeah. Yeah. Thank you, sir. Thank you. Yeah. Yeah. It's my pleasure to welcome Dr. B.S. Shripati Rao, Honorable Pro Vice Chancellor, Yenepoya deemed to be University, to MiCON 2025, who is the presiding officer to inaugural ceremony of this conference. And it's my privilege to welcome you, sir, to this, and you have been a constant support for all the MiCONs over the years. I do remember and acknowledge this. I request Dr. Bhagwan Rekadwar, organizing secretary, to welcome Dr. Shripati Rao to the conference. Yeah. Yeah. Yes, sir. Madam, we will get back to you. Not botanist right now. And Dr. Rambhat in the audience is a botanist. He should be able to tell the name. Right, yeah. Maybe during the course of time, yeah. So I extend my welcome to Dr. Lakshmi Kant Chatra on behalf of the organizing committee. I request Dr. Rajesh P. Shastri kindly hand over a sapling to Dr. Chatra, Principal, Yenepoya Dental College. I request Dr. Asif to give a sapling to Dr. Sham S. Bhatt, Dean, Yenepoya Dental College. A great support for us for the MiCON all the years, and your support is duly acknowledged, and we are having great pleasure in welcoming you, sir, to the event. I am pleased to welcome the delegates from the different institutions representing different states, my own colleagues from Yenepoya constituent colleges, research scholars, faculty, students, undergraduate, postgraduate, and my colleagues from Yenepoya Research Center, Dr. Bandari, the Deputy Director, Yenepoya Research Center, Dr. Rambhat, the HOD Biotechnology, Alwas College, Mudabidre, and other dignitaries to this conference. Dr. Sarpraz, the Vice Principal, Yenepoya Dental College, to this this MiCON conference. And I also extend warm welcome to our sponsors from Tarsans, High Media, Gene Technology and LabMate to the conference. I welcome one and all to this conference, and I am hopeful that this conference is going to give a lot of discussions, debate and new insights for fostering and furthering the research in microbiology to the greater extent, and seeing the same developments in MiCON 2026 in the same venue with greater promises. Looking forward for a wonderful deliberation in the day. Welcome all of you.
Thank you, Madam, for setting a delightful tone for the rest of the event. We would like to request Dr. B.H. Shripati Rao, Pro Vice Chancellor of Yenepoya, deemed to be University, to deliver a presidential address.
Good morning. I think all are sleeping. Good morning, I said. Yes. I always like, in any function I go where, you should be alert in the morning. And saying good morning brings positiveness in you. Very important it is, isn't it? So good morning to all of you. And chief guest of today's function, Dr. Sharmila Mandek. Madam, we are privileged to have you here as the chief guest for today's function. And the guest of honor, Dr. Tanaji. Is it Kudre or? Kudre, okay. You know what is Kudre in Kannada? You know, yeah, horse. Yeah. All right, and my two pillars of Dental College, Dr. Chatra Shambhat, organizing chairman Dr. Rekha and Bhagwan. On behalf of Yenepoya University, I would like to once again welcome each and every one of you to our sprawling campus here in Derlakatte. We are in the Dental College premises here. I always feel very proud to come here because I was also here as the Dean of the college in the past. One more person sitting at the back, the Vice Principal of our college. So we have got many pillars, not one, two. I see Dr. Sarfraz sitting at the back. Come front, Sarfraz. Because the dental college grew because of so many stalwarts there, not because one person. From the beginning, we had a fantastic team of faculty. They're all there for more than 20 years. That's why you see the growth in dental college. Anyway. So I'm extremely happy that the Department of Microbiology and Biotechnology, they are the division of Yenepoya Research Center, organized this 4th MiCON 2025. Fortunately, I was there for the last 4, I think, yeah, all the 4, but not the 4, because it was, yeah, it's happening on Saturdays always. And our Vice Chancellor normally, he doesn't stay on Saturday here in the campus, so they have to catch me only. So that's how I'm here. Anyway, I'm fortunate I say that. So it's an opportunity for me to share and to learn from all of you, because it's not my subject, but still as a surgeon who deals with the facial-macular surgery, we also deal with lots of infections. So I'm familiar with the problems what we face. And congratulations, madam, for organizing this for the 4th time here. And I hope this will continue. This trend should continue for years to come. And very happy to see so many delegates who have come from various parts of the country. Very, very important. Because we believe in sharing the knowledge. That's why you call it a national conference and invite everyone. So that whoever is interested to upgrade their knowledge and skills, then always welcome to our institution. And anything happening in our institution, we always welcome all the neighboring colleges also, irrespective of whether it's a national conference or anything. We always invite all the neighboring college faculty and students, because as I rightly said that we believe in sharing knowledge. So everyone should be benefited when you get some good speaker to our institution. So that's the trend going on in Yenepoya University, and that's being followed by all of them. Thank you for that. And before I say anything, my dear delegates who have come from various parts, take some time after the conference, if you find, you should visit some of our centers, and Rekha will definitely take you to our research center. We have a incubator, you should see that. We have a beautiful simulation center, because why you should see is, only when you see you will believe it, how it is. So I want you to personally go around and see, so that you can collaborate with us in future. We always say Yenepoya University extend their hand open-handed. Anybody wants to utilize the facility of our university for research purpose, you are always welcome, irrespective of which place you belong to, because we want more and more research and innovation happen in the country. So that is the motto of the present government also. We are seeing in the last 1 decade the way they are giving support for innovation and research. I don't think any other country is giving now. They are encouraging youth to do the innovation research in our own country, because facilities are there now. They have improved the infrastructure so much, they are encouraging every institution to develop the infrastructure. I am sure those days are gone where people used to look at the West to go and do research. I am sure it will reverse will happen soon. People will come to our country to do advanced research. See, COVID is the best example. When they had a COVID-19, nobody thought India can develop a vaccine of their own, and that also in a bulk. I don't think any other country could produce so much of vaccine in such a short time, not only helping our own population, they also help the other people also. So that is creditable. That shows how efficient are our scientists, how capable are our, the younger generation. So I want all of you, increase the knowledge. To get knowledge, you can go anywhere and you can gain, but my question is, there should not be a brain drain. You should utilize the opportunity given by our country, facility given by our people, utilize it in our country, try to develop. 100% will have plenty of opportunity to come up in life. So that is my first request to all the youngsters. That is a must that you should do that. Coming to today's topic, divergence and surveillance of microorganisms. I am not an expert in this field, I will not say something deep into it, but as a clinician, I know about the infections that are caused by the microorganisms, but let me tell you one thing. Anybody, when you say micro, the first thing strikes your mind is, oh, it's going to cause illness or diseases. We forget the fact that there are trillions of microorganisms are present within the body and on the surface of the body. If all of them have caused infection, we would have been no more, is it not? Whole mankind would have vanished. That means they are living and helping our body to function properly. You all know that all these microorganisms which are present in the body are very, very essential for the digestion of the food, to get the energy, the nutrition, the vitamins what is required for the body. All this is done by the microorganisms within the body. Very, very important. Not only that, they also protect your body from invasion of pathogens into the body. They are all called as the commensal organisms which are present in the body. Body forms a very protective layer all along the length of the gastrointestine in the form of a mucus lining, which has got, as you all know, that it has got antimicrobial peptides, you have the glycoproteins within that, and altogether they protect the layer, and also the integrity of the mucosa prevents the entry of any bacteria deep into the tissues and cause the disease. Once there is a breakdown, all the problems start. So integrity is very, very important. That's where your divergence comes. A lot of studies are going on in this. I was just going through one of the articles done in the USA where they feel the low diversity in bacterial colonization within the body is the main cause for chronic diseases. They noticed that, especially in the US population, they did a study, they found that low microbial diversity causes chronic infections. To put it very simple, like diabetes, type 1 or type 2, or irritable bowel syndrome, all these are caused because there is a very low diversity of microbes within the body. Now why it is happening in the US? Why? They have found out it could be mainly, one of the reason is indiscriminate use of antibiotics. Nowadays they don't go to doctors for for any control of infection. They themselves prescribe the medicine. Once they come to know the name, or they go to the Google, they find out what to take. They themselves, anyway, in USA they may not get the antibiotic that easily, because not like India, you can just go across and get it there. But then they keep prescribing it. There are two ways: either they take it very low dose for a short period, which can cause a resistance, or they take it for such a long time that will destroy the other organisms also within the body, causing multi-drug resistance. The main reason is indiscriminate use. And second thing is, in USA basically they all go for cesarean nowadays for a childbirth. They don't go through the normal process of delivery, which is very, very essential to get the normal organisms from the mother's womb. It's not happening. Third, they all use the processed food actually. The natural food they are not taking. Then from where do you get all this? It's not possible that you can get. All these reasons they felt that it is causing these problems, low diversity of microbes within the body. My dear delegates, is very, very important. I'm sure they'll go into detail about all these things. I don't want to dwell more. But what is challenging for all of us is, even in India, that we are getting multi-drug resistant organisms within the hospital setup. I blame both doctors as well as the patients. Nowadays, the present patient, when they come to the doctor, they're so restless, or they come with their Google knowledge and start asking the doctor, "Doctor, I have got fever for 2 days, give me something with which I'll get cured within 2 days." They want everything quick. Body takes its own time to develop the immunity, isn't it? The defense mechanism. It will take some time. Even if you take drugs, it will take time. They don't have. So what happens? When they go to the general practitioner, they load them with the drugs. Even for an ordinary fever, they load them with antibiotic, paracetamol, vitamins, everything. Something will work here and patient gets some satisfaction. With the result, I'll tell you, nowadays, especially after COVID-19, we have noticed that the infections are not getting controlled. Even a simple cough, cold remains for months together now. It doesn't go within a week or 10 days. Why is all happening? Because slowly all these bacteria are developing resistance. I'm only worried that one day it should not happen that the superbugs appear in our country and that is the end of it. Then our people will not be able to afford a treatment with the novel antibiotic which are going to come. Even there is no guarantee that that will cure. Because USA already they see a lot of deaths because of this superbugs. Plenty of people are losing their life. So you have got a great job to do, my dear students. I will not speak much about this. And one word about this artificial intelligence. Now everybody is after artificial intelligence. In every field you talk this. If I talk about my own profession or as a surgeon, already is going to replace the doctor, the nursing faculty, the instrumentation, everything will be done by AI supported robotic, then where is the role for us? Where is the role for us? We are losing actually. You should use it properly where it is actually required. Not for everything. I know now even in the microbiology they must be using it for early detection. Yes, that is required. Diagnosis, early diagnosis, if you can do it even before the clinical signs and symptoms come, that will be a boon for the doctors to treat so that you can prevent it early. Or for detecting which bacteria can go for antimicrobial resistance, if you come to know, excellent. Or if you come to know that such microorganisms are going to cause havoc in the community, if you are able to find out early, excellent. There you can use your artificial intelligence. For everything, if you depend upon that, tomorrow you will be like a robot only. You will be only knowing how to operate the equipment, but your brain will not grow. I keep telling all this because somebody yesterday called me and said, "Sir, I have developed a gadget, even from kindergarten till 5th standard, any question they have, it will answer." I said, I just give a chip, they have to go and put it and ask their question, it will answer. So it's very easy for the child. I said, with the result, what is going to happen is the brain is going to shrink, I said. He said, why, sir? I said, you are not giving any chance for the brain to grow. See, why we are all able to do calculation without even thinking? Because we have been forced to learn the table when you are small kids by heart. So even today, the present day children, just for a small calculation, they require a calculator, which we can do it just by, calculating through our mind. So that's why I'm telling you, every, advancement has to be used with caution. Then it will be very, very useful. Whether it's a machine learning or a AI, it has to be used. Otherwise, it is going to replace all of us. That you keep it in mind. I wish the function every success. I'm sure we have a galaxy of stalwarts in the field, Madam and Dr. Taneja. You will enrich your knowledge. Most important is interact with them. You have, if you have any doubts, don't go back with the doubts in your mind. You should always interact with the speakers. Then only they will also be satisfied that yes, it was worth coming all the way here. Students are happy. They have, clarified all their doubts. Not simply sitting mum and when we ask, do you have any doubts? Just nodding your head, that's all. That should not be the way, okay? With this few words, thank you one and all. Thank you Rekha for giving this opportunity. I wish the function every success. I was 2 days back, I said I'm not coming, I had a bad throat. But still I said when Rekha calls, this is my 3rd or 4th time, I didn't want to miss. But fortunately today morning when I got up, I was able to speak. Thank you.
Thank you, sir, for your insightful and inspiring address. I now request Dr. Rekha PD, Chairperson of MiCON 2025, to present a memento to Dr. B.H. Shripati Rao, Honorable Pro Vice Chancellor of Yenepoya deemed to be University. Dr. Shama S. Bhatt, Dean, Yenepoya Dental College. And Dr. Lakshmikant Chatra, Principal, Yenepoya Dental College. Now I request Dr. Bhagwan Rekha Dwad, Secretary MiCON 2025, to deliver the vote of thanks.
Good morning all. I would like to take this opportunity to express my gratitude on behalf of Yenepoya Research Center, Yenepoya deemed to be University for successful 4th MiCON 2025, which focused on the theme Divergence and Surveillance of the Microorganisms. Our first and foremost, our deepest thanks goes to our esteemed patrons, Dr. Yenepoya Abdullah Kuni, Chancellor, Yenepoya deemed to be University, Mr. Mohammad Farhad Yenepoya, Pro-Chancellor, Yenepoya deemed to be University, Dr. Vijay Kumar, Vice Chancellor, Yenepoya deemed to be University, Dr. B.H. Shripati Rao sir, Pro-Chancellor, Yenepoya deemed to be University, Dr. K.S. Gangadhara Somayaji, Registrar, Yenepoya deemed to be University, Dr. B.T. Nandish, COE of Yenepoya deemed to be University, Dr. Amir Baba Ahmed, Finance Officer, Yenepoya deemed to be University, for granting approvals to hold this conference and supporting MiCON in all the ways. We are thankful to our dignitaries, Dr. Lakshmikant Chatra sir, Principal Yenepoya Dental College, Dr. Sambhat sir, Dean Yenepoya Dental College, for lending their support and providing this venue, Yenepoya Dental College Auditorium. The conductive environment of this auditorium definitely facilitates productive discussions during the MiCON, I believe that, and I am sure. We are deeply indebted to our eminent speakers, Dr. Sharmila Mande, Ayush Distinguished Scientist Chair, Ministry of Ayush, Dr. Girish Mahajan, Senior Vice President, High Media Laboratories Thane, and Dr. Tanaji Kudre, Principal Scientist, CFTRI, Mysore. Their captivating talks definitely bringing us with the cutting edge research and definitely acts as intellectual bedrocks for MiCON. And I also believe they ignite passion for the potential of microbiome research for innovations in healthcare, sustainability, food, and personalized medicine. Our organizing team led by the Dr. Rekha PD deserves immense applause. Your tireless efforts and dedications and unwavering cheer kept us entire team motivating throughout the conference. I am thankful to my colleagues, Dr. Rajesh P. Shastri, Dr. Asif Hamid, for their extended support in all forms of for this conference, and it will be remembered for MiCON 2025. I would like to thank Students Working Committee, Mr. Yurajan, Ms. Amun Sonam, Suchitra, Akshata, Sanjana, Dhanyashri, Mayuri, Megha, Nishmat, Retik, Disha. If I am forgetting someone, please excuse me. For their untiring efforts during the entire days of MiCON and for playing various roles at all the levels. Our sponsors, High Media Labmates, Tarsans and Gene Technology deserve the special mention. Your generous support and contributions were instrumental in bringing the success of the MiCON. I also thanks more than 80 participants, those registered for this conference and presenters of this conference in physical mode. Without their participation, this conference would never have been completed. This definitely proves the success of this MiCON 2025 in this year of millennia. We express our sincere thanks to gratitude to the chairs of session that will be in coming scientific sessions, moderators, judges, Professor T.S. Keshav Prasad sir, Dr. Rogia Sultana, Professor Yashodhar Bhandari, Dr. Rajesh P. Shastri, Dr. Asif, for their meticulous evaluation that will be done and for their insightful comments that will be done in next scientific sessions. Our deepest appreciation goes to entire YRC support team, Sweta, Diksha, Vijay Kumar, Savita, Sanyukta, Rupa, Sajida, and Mr. Pawan. Their tireless support behind this scene of this MiCON 2025 ensured a smooth operation for this conference. We extend our sincere thanks to staff of the IT department for their expertise in setting up and maintaining a YouTube live stream and allowing conference to reach wider audience along with the organizing the and fetching one of the speaker, Dr. Girish Mahajan, online. Finally, we are thankful to staff from the Yenepoya's departments such as Yenepoya Printers, Dental College, Account Section for their continued supports, also from the Yenepoya Purchase Department for their extended service throughout the event. MiCON 2025 have been a phenomenal success in this millennia and a testament to the collaborative spirit and dedication of everyone is involved. We are proud to have brought all the such a diverse group of microbiologists, scientists, and researchers, students under the banner of divergence and surveillance of the microorganisms. This conference has undoubtedly sparkled important conversations and paved ways for further advancement in this critical field. Thank you once again to everyone who contributed for MiCON 2025. Such a rebounding success. We look forward to build this momentum and hosting even more impactful events in future. Lastly but not least, louder is important, but being a sound is very important. Thank you very much. We welcome to the MiCON 2025. I hand over session to the MC for further scientific sessions.
Thank you, sir. We are honored to have with us an incredible lineup of distinguished speakers, each one of them bringing a wealth of knowledge and experience, making this conference not only informative but also an enriching one. As we move forward, let's acknowledge that this conference is not just about listening to the great ideas but also engaging in meaningful discussions. Okay, we now raise for the national anthem. जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मागे गाहे तव जय गाथा जनगण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
The Islamic Academy of Education was established in 1991 by Mr. Yenepoya Abdullah Kunhi, an entrepreneur philanthropist, with a vision to provide quality higher education. As a non-profit trust, the Academy established Yenepoya Dental College in 1992 and set up medical, nursing and physiotherapy colleges subsequently. Over the years, institutions developed by this trust have created a niche in healthcare education not only in hinterland of Mangalore but also across the globe. In recognition for this contribution to the education system, in the year 2008, the Ministry of Human Resource Development, on the recommendation of the university, Grants Commission conferred the deemed to be university status. Yenepoya University thus became the first unaided deemed university to be established in the Dakshina Kannada district of Karnataka. The university, following the accreditation in 2015 by the National Accreditation and Assessment Council with an A grade, has diversified its academic prowess and scope by establishing Yenepoya Pharmacy College and Research Center and Yenepoya Institute of Arts, Science, Commerce and Management in the year 2017, the Yenepoya Ayurveda Medical College and Hospital and the Yenepoya Homeopathy Medical College and Hospital in the year 2018, the Yenepoya Institute of Allied Health Sciences and the Yenepoya College of Naturopathy and Yogic Sciences in the year 2021. Yenepoya University has secured accolades by various national and international certification and accreditation agencies. The significant ones being consistent ranking within 100 by the NIRF for 3 consecutive years. ATL ranking of institutions on innovation achievements by the Ministry of Education on 2 occasions under self-finance or private institutions category as an endorsement for the innovation and entrepreneurship development efforts among students and faculty of the university. The university has also been ranked with 4-star grading in KSURF Young University category in 2019. In recognition of contribution to the new knowledge generation, the university has been ranked among the top 50 institutions in India in the SCI MA Geo Institutions ranking consecutively during the last 2 years. There are multiple faculties of study in the university which are represented by the standalone constituent units. These institutions are spread over a total build area of 144,453.11 square meters to accommodate academic and administrative activities. Curricular diversification has been the focus of the university. It offers more than 100 programs at the undergraduate, postgraduate, super specialty, doctoral and postdoctoral levels. The university has enrolled more than 6,000 undergraduates, 600 postgraduates and doctoral students, complemented by over 500 well-qualified teachers trained in ICT-enabled teaching-learning processes. The classrooms, seminar halls and demonstration rooms are ICT enabled with Wi-Fi facilities extended to all strategic locations in the campus. The integration of information communication technology in teaching learning meets innovative educational standards. Self-directed learning is supported through e-resources on the customized learning management system. Choice-based credit system incorporates a wide spectrum of electives in majority of the programs offered. Internships, industry visits, community postings, skill development training and early exposure to research equip the students professionally competent for employment. The mentorship program supports the mentees throughout the duration of the program to enhance learning experience and personal growth. Community-based training is imparted through the urban rural health centers, PHCs, Rural Healthcare and Development Center, and mobile clinics to inculcate social responsiveness. The university library caters to the learning needs of the students and faculty. It has a vast collection of books, manuscripts, ancient books, digital learning resources. The library transactions are fully automated, facilitating accession and lending of books. The digital library, which is available at all the constituent units, helps the students and faculty to access the e-resources. All the academic departmental laboratories are well equipped for providing training. To enhance experiential learning and skill acquisition, the university has created Centers Advanced Surgical and Skill Enhancement Division (ASSEND) and a state-of-the-art Advanced Comprehensive Clinical Training and Simulation Center. These facilities are one of their kinds in the country and provide training support to the students and faculty from other institutions. The examination section is fully automated with an indigenously developed examination management system procedures involved in the pre-examination, examination and post-examination processes. This has enabled to bring in several technological reforms in the examination system, enabling speedy announcement of results. The university has been making constant and concerted efforts to actively engage in research. Research support facilities available in the university include state-of-the-art central research facility with modern equipment for conducting advanced research in emerging areas like microbiomes, cell signaling, nanoscience and microfluidics, stem cell and tissue engineering, systems biology and molecular medicine, omics technologies, bioinformatics and biostatistics supported by highly qualified manpower, funding, informatics tools, Animal House and Ethics Center. The Ethics Center has the unique distinction of imparting NIH US.
Now we will begin with the sessions. I request Dr. Rekha Pedi, Chairperson MiCON 2025, to introduce the keynote speaker to the gathering.
Yeah. After that session of inauguration where we got some insight into about the proceedings of the day, we are starting the first session with a distinguished speaker, and it's my privilege to introduce Dr. Sharmila Mande to the audience. Dr. Sharmila received her PhD degree in physics from Indian Institute of Science, that is the IISc Bangalore, in 1991. She pursued her postdoctoral training in the University of Groningen, Netherlands, and University of Washington in Seattle, USA. She started TCS Bioinformatics and Computational Biology Research and Development Group in 2001. Post her superannuation, she continues as an advisor to life sciences research at TCS. She is presently holding Ayush Distinguished Scientist Chair position from Ministry of Ayush. She is also a visiting professor at IIT Kanpur, IIT Gandhinagar, IISER Trivandrum, and many more where she is an advisor. She is a pioneer to initiate cutting-edge research in the field of microbiome in India. So that is why she has been called for this conference, considering her pioneering work in the field. Dr. Sharmila has developed many algorithms and solutions having huge translational potential, particularly in microbiome and bioinformatics research. The innovations include novel methods for disease diagnostics, therapy, health monitoring, and their other industrial applications. She has to her credit 77 granted patents and more than 100 publications in highly reputed international journals. She is a fellow of Indian National Science Academy and the Indian National Academy of Engineering. So with this very brief introduction, I'm inviting Dr. Sharmila Mande to give the keynote address to this MiCON 2025. Over to you, madam.
Projected. Okay. A very good morning to everyone, and thank you, Dr. Rekha and Dr. Bhagwan, for inviting me in this conference. Really excited that, it's privileged to have the dignitaries attending that in spite of the busy schedule. Lovely to talk to all the youngsters. I feel really young when I talk to the, when I give lectures to the young the researchers. And So thank you for inviting me in this conference. So what I'm going to talk about is, since this conference is about microorganisms, the diversity in different regions, I'll focus on how microorganisms can be exploited for health and wellness. I'll talk about our health, and which is the microorganisms which are within our cells, as well as something which is surrounding us. So I'll just talk about some of the research work what my group has been working. So if you really look into the microbial world, there are about 1 trillion microbes which are there on this planet, right? So out of that, do we know enough of all these? We don't. So originally the way microbes were studied, where they would culture them, look into the microscope, under the microscope, and see what kind of microbial shapes are there and so on. Then they went into sequencing the genomes of the DNA which are present in these microbes, right? Each of the organism, they have a DNA, and then they try to sequence those, and then try to find out what genes are there, how they are functioning, and so on. So each organism they would. So but out of this 1 trillion, you have only 400,000 microbes which have been sequenced so far. So the question is, why is that you have only 400,000? The sequencing machines have been there for a long now, right? So we should have solved as many as possible with the advances, but we have been able to solve only know the sequence of the DNA of these microorganisms, that is the genomes of these microorganisms, only 400,000. So we know only tip of the iceberg, and we don't know majority of them. Now let's see where they have been studied from before we go into the problem, why we have not been able to solve the genomes, understand the genomes of all of them. I'll go to that. But if you see, out of this 400,000, majority have been studied from humans. As you all know, you heard from Professor Rao that we know that there are so many pathogens which are there, which infect and so on. So people have been looking into the microbes and health, and we always used to think that bacteria or any microorganisms in the body, that's bad, right? That's what, that's the bug, which is the pathogen, and so on. So people have been focusing on the role of this microorganism, the human. So out of these 400,000, you have, if you see that majority of them have been studied from the humans, about 150,000 have been studied from humans. But there are other places also people have studied. As you can see, there are plants, some animals, goat, cow, and so on, and different environments, soil, water, and so on. So about only 150,000 microbes have been 150,000 microbes have been studied from the humans out of the 400. The question is, why is that? The first question which I asked, that 1 trillion microbes which are there, why we have studied only the 400,000 microbes? The reason is that if you try, so you have to culture it, right? If you have to sequence the genome, you have to extract the DNA, culture the microbe, extract the DNA, and then you sequence it, right? You put it in the sequencing machine. But the problem is, majority of these microbes that cannot live by themselves. They like to live in a community. So if you try to take just one out, they die, right? That's a problem. But thanks to the advances in the sequencing platform, science, technology, and engineering, there are sequencing platforms which can sequence the entire microbial community's DNA. So what you do is you take the samples. I think this will not work on this, I think, because of now. So you collect the sample from any environment. Suppose you want to study what is there in the soil, right? The soil around this campus. So you take the soil, extract all the DNA, and then put it into the sequencing platform, and what comes out are the sequence fragments from all the microbes which are there in that environment. So it is possible, because of the advances in the science, technology, and engineering, these sequencing platforms can sequence all of them together. So now the question of, some microbes cannot survive without the others does not arise, because you are sequencing everything out, right? You are not doing culturing of each bacteria. So this is culture independent. Now the problem is, what comes out from here, sequencing platform, is really a complex data. And this microbial community, what I have been talking about, this is called as the microbiome. So microbiome is nothing but the collection of all this, the microbial community which is residing in any environment. So if it is from the body, you say that human microbiome. If it is from the soil, you will say soil microbiome. If it is from water, you will say water and water microbiome and so on. So what comes out is the microbiome data, and this data is really, really complex. And the computational biologists, then they have to address 3 questions. You have to know, because you have got only the fragments of the DNA of all those trillions of bacteria which are there in that environment. So you have to answer what microbes are there, what are their relative proportions, how are they functioning, and if they are functioning in some way, what are the functional genes they have, and so on. So these questions one has to address through computational methods. So, and if you look into is it possible to switch off this? I mean, hide that. So this just shows that if you see the number of studies which have been done and published in the international journals, you will see that it is thank you. So it is exponentially growing. So this is the data which shows that it is exponential growth. Everybody is interested in understanding this microbiome because they play very, very important role. So I'll give some of those in that. So I spoke about, they are present in different environments, and human is one which has been studied, in depth. But there are other places where they have looked at. People have studied from the extreme environments, acid mine drainage, hot spring, then you have the soda lake, hydrothermal vent, and so on. These are really harsh environment. One would wonder how these microbes survive. So if they are surviving there, is there any functional potential which can be utilized for any benefits for, in the industrial processes and so on, right? And that's why people have been studying this. So it's not just the human body or the soil around or the water around us, but the people are studying such harsh environments as well. So now, if you look into the microbiome data, I'll not go into the data per se, but I'll tell you from the data what all can be mined. So, but just to give a brief, that if you see the kind of microbial data which comes out, is very complex. The volume is really large. The length of the sequence, depending on which sequencing platform you use, the lengths are different. Complexity of the taxonomy, that is the kind of microbes which are there. In some environments, some microbes might be at very high number and some might be at low number. But the low numbers are also important. It's not enough to say which are the ones which are abundant, but the lower ones also play some important role. So one has to understand that. Quality of the sequence is really, really good. So those of you who are doing computational part, the very first thing in any experimental, if you have to analyze those experimental data, is identify the really high quality data. So the quality, removing the low quality data, because otherwise what happens, all the downstream analysis, although you will get some answers, may not be correct. So the how do I, identify the high quality? And then, so you need specialized algorithms for analyzing this complex data. And just to give an idea that in acid mine, there will be 1 or 2 or few microbes which might be there. In the sea water, about hundreds of them. And the human gut itself, there are thousands of different type of microbes which are there. And in the soil, there are 10,000 or more microbes. This is just to show that the complexity of the taxa which are there in the environment. Now just to start that, I started my journey in this around 2008, and that's when we started developing lots of algorithms for analyzing this complex data. But as you can see, there are along the path, I've been working with several hospitals and academic institutes to understand some of the diseases. I'll cover few of those. We studied malnutrition, gastric microbiome, diabetes, on the skin infection, vitiligo and leprosy. Then I'll talk about a little bit on the preterm birth, and something we are doing right now going on is on the upper gut health, which is the duodenum microbiome, something which is going on with PGI. So that's the last one which is going on. All the collaborations which I have put in are in the magenta color, and those are our collaborators. Fortunately, I haven't done anything with the dental ones, but although I have a collaboration going on with the dental clinicians from 5 different countries, which is going on looking into the AMR. But on the microbiome, we haven't really done anything, but would be exciting to see that. So having said that, now let's see how these microbial, so I'm not going into the details of how those, that will take series of lectures to how do you analyze the data. But let's see if you have to exploit it for improving the health, diagnostics and so on. So if you look into our body, so we have 10 raised to the power 13 cells. Each cell has a DNA, and we know our human genome, right? So we know the DNA, and people have been looking at this DNA, trying to understand what are the SNPs which are responsible for, which are correlated with the, say, cancer and so on. So people have been looking at it. So we have about 25,000 genes in our own genome. But if you look into our other aspect of the human body, we have equal number or more than that number of microbial cells. Dr. Rao, he mentioned during his inaugural talk that, we have, many microbial cells, and they are present in the organs which are exposed to the external environment, our skin, gastrointestinal tract, right from the oral cavity all the way to the large intestine. Then you have on the lungs and so on. So these are all the places where there are lots of microbes which are there, and these are now known, and since they outnumber our cells, so out of 25, so as compared to 25,000 genes which are there in the human body, we have more than 3 million microbial genes, and they play very, very important role in our health and well-being. So, so it's now referred to as the second genome, or our human microbiome is called as the second genome. So both play a very important role. Now if you look into our body, so you will see that, as I said, there are different microbial, the 100 trillion these symbiotic microbes are there at different places. For example, 600 plus different types of microbes which are present in your lungs and so on. Esophagus and the lungs and so on. Then you have thousands of them on the skin. Then you have 500 to 1,000 in the gastrointestinal tract. About in the stomach, you would have thought that in that harsh environment, maybe just Helicobacter might survive, but there are no more thing. But there are 25 different types of species which are there even in that harsh environment in the stomach. So there you will see that different parts of the body there are these microbes. So are they all harmful to us? What are they doing is the question. So if you see the human cells, as I said, is more than our own cells, and most of them they reside in the gut, the large intestine, and 150 times more microbial genes, as I said. So we are more microbial than that. So when we say we are not human, we are basically, we are all made up of the microbes. And so microbiome actually is the new descriptor of health. And I'll give you example why I'm saying the new descriptor of health in the next few. So what has been seen that, as I said, majority of them reside in the gastrointestinal tract, and they actually have lots of role to play. I've mentioned a few, nutrient harvest from the food. I'll go into little more details on that. The pathogen colonization, these were all mentioned during the introduction by Professor Rao. They regulate the immune system, the secondary metabolites which are produced, which are helpful, and so on. There are many functions of the microbes. The 95% of the microbiome or the microbes which are reside in the gut. And Hippocrates, way back in the 450 BC or so, he said all diseases begin in the gut. So that's so true now when we look into from the microbiome perspective, it's very, very true that it is indeed. So we are what we eat. So what has been seen that if you see the gastrointestinal tract, the gut microbiome, and that has been linked to diseases and disorders which are affecting different body parts. If you look into this particular figure on the right, you will see that on the lower thing, when you say that diabetes, IBD, ulcerative colitis, these I think, these are, you will say, okay, they are affecting the gastrointestinal tract, and you are linking that with the the gut microbiome, that's okay. But if you look into liver diseases, cardiac functioning or the brain behavior, you can see that anxiety, depression, autism, Parkinson and all these things, multiple sclerosis, these all also have been associated with changes in the microbial community. These are from different studies. These have been reported from different scientific studies in that. So it's so true that all diseases begin in the gut is very, very true. So we are what we eat is really a factor. Now the question is, since the gut microbes is so important, what really constitutes a healthy gut? That's the question we asked. Because we have to monitor, if you have to monitor our health, indirectly we can monitor the gut health is what we have. So what you, as I said, that what you do is that you collect the sample. When I said that you have to study the gut microbes, you basically take the stool sample. You take the stool samples, you extract the DNA, give it into the sequencing machine. What comes out is all those sequence data, you do all the analysis, and what comes out is finally in each of the samples, you'll say bacteria 1, how many of them, what is the abundance, bacteria 2, what is the abundance. So this is called as the bacteria, this is called as the abundance table. So you have ecosystem 1, 2, 3 could be, you take 3 sets of individuals, one is healthy, one is say disease 1, one is disease 2. It could be pre-diabetes, diabetes or whatever. The 3 different sets of or the cohorts of individuals, and in each of these you find out what are the proportions of the, what are the abundances of different bacteria which are there in the stool sample, that is in the gut. And then you basically find out the diversity. And it was mentioned that the higher diversity is associated with the healthier gut is what has been seen. But I will come to that diversity aspect little later on. So you basically see that what kind of different bacterial groups are there and what are their relative proportions and so on. So this richness and evenness is what you say. So this, when you get this data, the abundance table, from there you have to find out how do you actually monitor the health of the gut. Now the first question we asked was, this was one of our very first, actually my first collaborative work, where we tried to understand malnutrition. You said that some children are malnourished, some children are healthy and some in the borderline. So we wanted to see what is there in their gut microbes. So this was from a rural setting in West Bengal, and we took a spectrum of children from the same rural setting where they had the similar kind of food and water and so on. Unlike in the cities, people will have, today will be pizza, tomorrow will be burger, and the day after tomorrow will be something else and so on. But these children were all below the age of 5. And we took a spectrum of children where at one end it was absolutely healthy child and seemed to be a healthy child. At the other end was severe malnourished child. So what we did was we tried to understand, we actually did the sequencing of these, and then we tried to see what microbes were there. And what we found were they all had, including the healthy child, had even pathogens. But the community structure, the way the pathogens associated with the commensal bacteria and so on were very different. And as you go from the malnourished to the healthy child, this community structure was disrupted in such a way that the pathogens got disrupted from their own clubs, or they were forming a community, a very strong community. They were dislocated, and the healthy, there were good bacteria which were dominating, and then with the pathogens which were also there, which indicated that indeed the community structure in the gut microbiome is very, very important. Then what we did was this WHO recommended ready to use therapeutic food, what is called as the RUTF. So RUTF was given to, this is another study what we did on to the children who were all SAM, which is a severe acute malnourished children, and tried to see what happens when they are given RUTF. And what we found was before the RUTF given and after the RUTF given, not everybody became healthier. Some still remained in the borderline case and so on. Again, the same situation, that the dysbiosis or the the structures of those microbes in the gut in some of those malnourished children were so bad that even the IUTF could not help them to regain that. So that means that it's one size fits all is not going to work. So one has to look into the personalized therapy. That's something which came out with. Now having said that, what is actually this gut microbes doing? So as I said, there is a link between the community structure and the malnourishment. That's one which I said. What actually doing? They have some functions which they do. So what happens is when the food is, we ingest food, it gets digested in the stomach, but the undigested food goes into the large intestine, and there a set of the undigested carbohydrates, when it comes to the large intestine, a set of gut microbes, they actually act upon these complex carbohydrate, undigested carbohydrate, to convert them into monosaccharides and disaccharides. And then another set of bacteria which are there in the gut, they convert these monosaccharides and disaccharides into short chain fatty acids, butyrate, propionate. You all must have heard of these, and these are all useful for the gut homeostasis and the integrity of the gut and so on, and for the immunity and so on. On the other hand, if you look into the undigested proteins, when they get into the large intestine, there is another third set of bacteria which act upon this. They are happy to digest these undigested proteins and convert them into peptides and amino acids. And these peptides and amino acids are again taken care by another set to convert those into ammonia, phenol, indole, cresol and so on. And these are all harmful for the gut. So if you realize that depending on the gut microbes which are there, you will be able to see how many of these microbes are degrading the undigested carbohydrate, how many of them are degrading the undigested proteins and so on. Okay? So this is something which we have been looking into, and we asked the question, can we find a set of bacteria which can serve as a reliable indicator of the gut health? Because finally what these gut microbes are doing is something they are converting into good, the butyrate, propionate and so on, and other sets are converting into the ammonia, phenol, which is not good, right? So how do we quantify this and come up with a reliable set of microbes which are, this is what we asked the question. So what we did was, so we analyzed the butyrate and this putrefaction pathway, which is the protein degradation pathway, and then we could come up with a set of microbes as a gut health indicator. And so this set of microbes, and basically we looked into the functional potential of these microbes. We looked into the functional potential of these microbes, and we came out with a gut health score. Again, I'm not going into the details, these are there in the paper, some of these papers. Gut health is not here. If you look into the gut feel, you will get that. And then, so we came up with given a microbiome sequencing data, we actually can compute this functional potential of these microbes which are there, and say that whether given 2 time points, say if I take the sample today and after certain therapy, I take the sample after the therapy, I'll be able to say whether the gut health has improved or not. So the question is, how do we know that this really works? You might say this is only from the experimental data we have got it. So what we did was, these are all the different data which were there. As you can see, the left side is all the diseases which affect the gut. On the right side, the lungs and the brain. So these data sets were taken. These were the studies which were already done by people, and they had healthy cohort and the disease cohort, and we checked the gut health of the healthy versus the disease cohort, and we could see that the healthy cohort had better health, better gut health, much better health, gut health than the disease ones. So this is something which we did. Now the next one which comes to mind is that, so what happens is that in any individual we have pathogens as well. There is a healthy balance amongst all the different types of microbes which are there in our body. But in any part of the body, if there is an imbalance, and that has been shown to be linked to disease and disorder. Okay? So the question is that can microbiome variations be used as diagnostic marker is one of the questions which we asked. And we actually did with for preterm delivery, and if you see about 2.6 million babies are actually about 15 million babies are born preterm, and about 1 to 1.5 million babies they die every year globally, and India actually unfortunately stands one on the top ones. And this, there are out of the other cases, the complications, I mean the 2.6 million babies who die in the first 28 days, the first thing, the prematurity actually is the major cause. We asked the questions, can we predict it? So we were looking at some of the data sets which were, so there were 4 public available data sets which were there, the 3 studies which were done from the US and the Chinese thing. So though we were going through those papers and trying to understand what they have said, and none of the papers said what are the key microbes which are responsible for these preterm delivery. So what we did was we pooled all the samples, all of these samples we pulled together and tried to understand. And so what we have is that these are all the samples, and in each of the samples what are the different type of taxa which are there, the microbes which are there. And so we asked the question, are there signatures, and these are not the gut microbiome, these are the vaginal microbiome. So remember that the higher diversity is better when we say that for our gut, not for all regions. For example, in the vagina, it's supposed to be as low diversity as possible. Okay? So we asked the question, are there signatures in the vaginal microbial community structure that can indicate predisposition to preterm birth? So we have about 1700 samples which we had, and when we combine all of them, if you look into, this is the gestational week on the x-axis, and y-axis is the diversity. You will see for a non-birth delivery the diversity is very high. This is the blue is the diversity is high, and the red one is for the preterm delivery. The diversity is low. So which we thought that maybe we are making some mistake, and then we said that why is that, and then on in the second and third trimester the diversities are very similar in both term and the preterm. So we asked the question, why is the diversity so high in the first trimester for the term cases, which should not be, because people think there is some vaginosis or some infection, but there are some other reasons, but I'll not go into that. But then question is, can you use, since we see a difference in the diversity in the first trimester, can you use that as a marker? Can you use that as a diversity as a marker to predict the risk? But we cannot. But as you can see, the term, and if you use this diversity measure, which is standard, what is called as the Shannon diversity, which is used, if you use it, there is an overlapping region. Because of the overlapping region, this region, you cannot say for those women who come into that, you will not be able to say whether it's going to be term or preterm. So we asked the question, can you amplify the signal to separate out these two gaps? Gaps between increase the gap between term and preterm in the diversity. And we developed a metric called as the taxonomic composition skew, in short TCS metric. And this is all whatever I'm talking about, these are all published. So you can go and see the publications, they are all patented also. So we could create that, and we could show that we could predict the preterm delivery in the first trimester with very high accuracy. We are validating this with Lady Harding Hospital in Delhi right now. This particular one is going on. We have other ones, as a microbiome, as a diagnostic marker. We have been working on breast cancer, colorectal cancer, and so on. And some of these are actually we are trying to validate with some of our partners. But I'll not go into that. Now come back to the next one. So I talked about the diagnostic, that is, can you diagnose this tilt? Now you think of other way, there is a tilt, can you rectify it? Can you bring it back to the balanced state? That is the therapy, right? So let's talk about the microbiome, how microbiome can be used as a therapy. So there are different ways one can do it, right? So we can give probiotics, people use probiotics. If you use probiotics, there are only few handful of probiotics which are there. But we don't know how it's going to affect. It's not going to change the complete microbial community. Some of them may accept, the community can accept, some of them may not, and so on. But specific strains of probiotics will have to be designed. The prebiotics, which are the food for the good bacteria, fructooligosaccharides and so on, they can be provided so as to increase the community of the good bacteria. That's another way. Specific bioactive producers, for example, butyrate producers. Can you put those butyrate producers and then see whether it improves or not? Antibiotics is a big thing because the antibiotics, if it is antibiotic actually is non-specific. As was mentioned earlier, that when you give antibiotic, it's going to kill everything. So you don't want that. But can you identify specific antibiotics which will kill only specific pathogens which are there, but which is a very challenging, which has not been developed, is still not in that. One of the things which is done is the fecal transplant. I'll talk about this fecal transplant, which has been quite successful. So what was done that these mice, they are grown in the germ-free environment. So this, what on the left side, what I have is the germ-free environment where they actually create these mice which are devoid of any microbes. And what was done was, there was a study, it was a very beautiful study which was done more than 10 years ago, was they took a twin, the human twin, and one of them was obese, other was lean. And what they did was, they transplant this so they took a germ-free mice, the two of them. In one, they transplanted the gut microbes from the fecal transplant from the obese twin. The other one, it was from the lean twin. And they gave the same high-fat, low fiber diet and so on. And lo and behold, you saw that, they noticed that one became fat and one remained lean. So which indicated the gut microbes actually dictate the phenotype of the individual. So this was something which was a big success. After this, they have been doing this in US for Clostridium difficile infection, and they have been quite successful in that, not 100%, but they have been quite successful. Clostridium difficile infection in the Western world is a very a chronic case, unlike in India. And even in India now, people have started doing for Crohn's disease and so on, the fecal transplant. And recently, about a year ago, there is already a FDA approved fecal transplant pill, which is also has become available. Now the question is that the right way of going? I personally feel that it's the fecal transplant, we don't know, because they give it from the healthy volunteer to the diseased person. But there are other microbes which are also there. How is it going to be long term? We don't know. So the ideal, it will be the microbial consortia, the personalized microbial consortia, which has to be identified and can be given to that. So that's going to be with specific functional role is something which I feel is going to be the real future. So if you really look into our human, the complete life cycle before the baby is born, I talked about before the baby is born, microbiome can tell you whether the baby is going to be born term or preterm. And if the baby is born, in the normal, the baby gets the inoculation, the ones from the mother. If the baby is born in the cesarean, they normally get the microbiome from the nurse who held the baby first, and that has been shown in the literature. Then the newborn, you can, something which you are working on in the neonate sepsis is something which we are right now trying to understand. But the risk assessment in the early stages, in the asymptomatic stages in young age, middle age, and in the old age is something which can be done with the microbiome. I gave one example of, some of the examples which I have put in. But at the old age, the wellness monitoring, to looking into the gut health of the person, and then you give the diet recommendation. So we basically we're moving towards a wellness-driven healthcare, and microbiome plays a very, very important role in understanding what is there and how do you proceed to make the health better. So preventive and personalized therapy. So if it's very simple, the subject or the patient or the individual comes in, the microbiome sample, it could be a stool sample, it can be saliva sample and so on. Just take a little bit of that sequence, and then we can analyze the risk of that, and the recommended therapy or diet can be made based on that. So being a physicist by training, we have any equation, that's a constant and a variable. The constant is our genome, which you cannot change, but the variable is our microbiome. We can change our environment, change the food and so on, so that you can change it, So I just talked about the microbiome and health from the human microbiome perspective, and in the human microbiome test is going to be really something which is going to become a very common thing, but it has applications in other areas: agriculture, energy, environment, animal husbandry. You think of any area, microbiome plays a very, very important role, and it's actually really something which is important. Do I have time to go into this? If not, I will stop here. This is for waste to wealth. Hmm? 10 minutes, yeah, 10 minutes I'll finish, yeah. So I'll now switch the gear. Now I move into how microbes play a very important role in converting waste into wealth. That's something which I'll talk about. So if you look into, our office was located very close to a dump yard. I used to tell people that, there's a gold mine, and people used to laugh at me. I said, yeah, there are microbes which are there. They're surviving there in the pollutant environment. They're eating those pollutants. So can we use those for our benefit to clean up the environment? So, so there are different, types of pollutants which are there. I'll not go into that. So how are they utilizing these pollutants? So you know that in any compound, if it has to be degraded to non-toxic, you have a certain pathway. So I've given a pathway right here on the right side, that if you have to convert the PET plastic into, say, pyruvate, it has to have the enzymes which will convert into different intermediates, and finally it gives the non-toxic thing, right? So the idea is that, can you identify microbes which have all the genes which are responsible for converting one pollutant into some intermediate or to the final unpolluted ones, is the question. So there are different genes. So for example, here I have shown A, B, C, D, E. These are the 5 genes which are required for converting one pollutant into a, say, intermediate or to a non-toxic thing. In the genome, you have lots of these genomes which are already there, and can you identify these? In one genome, I might find all the genes which are there, but they are present in different locations. And what you need is they have to be present in proximity, because the product of one has to go and be acted upon by the second enzyme and so on, right? The genes have to be present. So so we analyzed all these microbial genomes, the bacterial genomes, and we came up with, we tried to identify which bacterial pathogens which bacteria have those pathogens and so on. And we utilize those to say that when given any pollutant, if you give any pollutant, I'll say which are the set of bacteria A which will convert into an intermediate something, and which are the set of bacteria B which will convert that intermediate into a final one, or A, B, C, and so consortia of different groups. So basically, for the complete degradation of the pollutant, we'll have a set of microbes, and you choose which are the microbes we want to see. Of course, these are all the computationally we've predicted. Now it's being validated which is the best combination which will work best with the yield and so on. So that's something which we are trying to. Now, if you look into this, the importance of this is when I say waste to wealth, any of these industries which I have mentioned on the left side, they all produce lots of pollutants. Different types of pollutants are produced by the chemical industry, automobile industry will have different pollutant, pharmaceutical or cosmetic industry will have different pollutants and so on. So all these industrial waste what comes out from here can be, something can be utilized from here. The set of microbes can produce certain compounds which can be utilized back in the same industry. I'll give you an example of this. So let's take PET, right? The PET has, I've given you bacteria A and D. The PET, whatever terephthalic acid is used in the packaging industry and so on. So I've written out in these, in the oval boxes. If you combination of A and D, they are used in the bioplastic production. Combination of A and B can be used in the cosmetics industry and also whatever the product in between intermediates which is produced by a combination of these can be utilized in these kind of industries. So I just give you example of PET. So you can think of all those chemicals which are produced in each of these industries, how that can be utilized to produce wealth from there. So what we have been doing is that I have an active collaboration going on with IIT Kharagpur, where one of my associates who is doing PhD with Professor Pinaki sir and myself to understand this Piranha landfill. She has taken the samples from the Piranha landfill in Gujarat, and what we have is that she tried to look into the plastic degrading consortia, because we have been looking into the plastic as one of the things, but we can work on any of these, and that's her, she's doing the experiments. And this is just to show that, when you look into this LDP plastic degradation, this is to start with, you dip the plastic, and the plastic is in this form, which is on the right side, and after 120 days you can see how plastic has become, the last one. So definitely that consortia is acting upon these microbes to do it, although it is very slow, but it has a potential to do that. So finally, I want to finish that I just talked about the human health, how the microbiome plays a very important role in personalized therapy, diagnostics, monitoring health. Agriculture I didn't touch upon, but you can think of nitrogen fixation, increasing the yield of the agriculture products, or the drought resistance, and all those can be thought of for the microbes' role as that. Degradation pollution, I just talked about. The food industry, another area which is, but think of an industry, in all of these industries, there are applications of the microbes can be exploited for our own benefit. With this, I want to thank my entire team. I have not presented everybody's, but I just want to thank my team who have been doing research in my group. I presented only a few of them, the gut health and the degradation, the bioremediation component, and the diagnostic one, but there are many more interesting stuff which we have. These are the present ones which are in the left side, and the right side is those who have been in my group. With this, I want to thank you all for the kind attention, and thank you again for inviting me to give you this. I hope, and I'll be very happy to interact with the students, answer the questions or whatever they have doubts. As was mentioned, that it's always important to interact and.
Thank you, madam, for that enlightening talk. We will culminate with an interactive question and answer session. The session is open for the questions.
So if there are no questions, I take this as either they were not attentive at all, or I was so, I'm a very boring teacher that, they just, everybody fell asleep.
That's a question.
So, I don't know, or maybe it's...
Hello, this is Prithvi from Damgir University. Ma'am, how actually we can know particular prebiotics enhance the particular growth of a probiotic bacteria?
See, prebiotics are known to, so for example, certain prebiotics are known for increasing the Faecalibacterium population, right? So these are known. So which bacteria utilize what kind of prebiotics? For example, if you take fructooligosaccharides, not all bacteria will utilize the fructooligosaccharides. So you can find out which bacteria they use the fructooligosaccharides, and that they will increase the growth will increase, right, of that.
Experiments.
Oh, what experiments to do to see whether the which are the bacteria.
Yeah.
See, basically what you will have to do is that you will have to, if you take the entire community, right, and you give the fructose, so the way we looked at it was that before the fructooligosaccharides was given, we took the sample, the stool sample, we saw what all microbes are there, all the taxa, right, and the abundances. We gave the fructooligosaccharides at different dosages and different time points, and we saw that certain, for example, Faecalibacterium increased in number. Okay, so all other bacteria were more or less similar, but some bacteria where they increased. Okay, so this is all from the microbiome. So these experiments are called the metagenomics experiments, right? So from the entire microbial community, we saw that with the fructooligosaccharides, certain bacteria really increased, which is good. And then we said, okay, now if we stop, what happens? When we stop, it again went back to the original one. So it's actually, it shows that Faecalibacterium increases. When you stop, it slowly actually comes back. So you have to keep giving that. So you can see which all bacteria, Faecalibacterium is one, but you can also see the other bacteria which are growing.
Thank you.
Good. Should announce that there will be. Good.
Ma'am, in the previous slides, I didn't understand what kind of high throughput technology you were mentioning. I didn't understand that part.
The high throughput technology.
Like you were saying, I think mentioned AI. I think this kind of technology.
No, not AI, not AI. I can talk about AI as well, but no. See, sequencing platform used to, when I say high throughput technology which are enabled, here I am talking about sequencing platforms, right? Earlier sequencing platforms could sequence only one bacteria. I mean, it will take the whole day or maybe a few days. But then later on what happened was this sequencing technology is now so high throughput that you give as much amount, it will be able to do it within a couple of hours, sequence the entire thing, which used to take years, the days to sequence a particular one genome, right? But we are talking about the sequencing the entire community in one shot, right? Those technology I was talking about. Talking of the AI, again the AI, I think a lot of cases this is really a hype, because what happens is AI actually depends on the data. You have to train the system, right? So if the data is large, if you ask me that all these whatever I'm talking about, whether I could have used the AI, but the data which are not that sufficient enough to build a model for the AI to predict it, right? So AI for those areas where we have enormous amount of data, that can be, people are trying, it's not that people are not trying, but there is a power of AI, but you can't rule out, that, our, way of analyzing that is definitely, because the data is sparse at one. So technology, I was talking about the sequencing technology.
Thank you, ma'am.
There's one.
I would like to know, is there any role of prebiotics or probiotics in the department of oncology? I work in oncology, so for an immunocompromised patient, we avoid prebiotics, probiotics, since it will alter the, a patient gets mucositis. We never prefer that. We only, so I would like to know, are there any options for that?
So, so again, so you're talking about you avoid giving up prebiotics or probiotics. Again, it depends on. See, first thing is for any tumor. If you have to see, people have been trying to understand the tumor microenvironment, right? And then the microbial community which are there. You want the the pathogens to go down. That's one. Because it has been seen, for example, in colorectal cancer, they have looked into the colorectal cancer affected region and the non-affected, they do see a difference in that. What I would say is that so if you have to give the oral prebiotics which will go in the gut, one has to actually understand how it's going to, how is the link between whichever part it is affected. Say for example, if it is lung, so if the gut and lung, how is it connected, whether the prebiotic is going to make a difference or not. But there are no studies which have been done, so I can't say that how you can say that this prebiotic is going to help. Even the other prebiotics, when we say that this is a prebiotic which is good, I don't know whether it my gut microbes may not need, because that bacteria might be enough in mine, right? So one has to find out the link of this prebiotic when you give it in the gut, and because the gut has a connection with different parts of the body. Say if it is a lung, say lung cancer, how is it connected? And if you give a prebiotics or probiotics, what happens, right? But none of those studies have been done. Till those are done, I think it's too early to say that give a prebiotic and that's going to solve. And I agree that why you avoid giving a prebiotic.
Any other questions?
Not prebiotic, probiotic you said.
It's curious only, it's not a question. So nowadays there is an emerging issue for the microplastic in the food system. So can we make such an intervention for the, as you said, microplastic degradation, so bacteria consortium is there. So, so in future, because it will be emerging, it will be very toxic to health. So can we make such a consortia that can be come into our gut, so can once it is a microplastic is coming in food, it can degrade.
So that's a good idea. But the only thing is, first you have to find out this microplastic. How first you will have to see how it can be degraded. What is the consortia which will be needed, and depending on because you don't want to give the consortia when it is not needed. I know certain food you take in, the microplastics are there, right? But maybe I'm not eating those foods, so I may not need that consortia. Once I find that from the gut that these are actually there, then I can take the consortia. Yeah, that should be possible. That should be possible.
Madam, I have not a question, but what I understood from your talk. Two things I want to say. One, she asked in cancer why we don't give prebiotic, and especially if it is a gut cancer anywhere, maybe that by giving prebiotic the organisms will grow and it may affect more.
But we don't know.
We don't want to simulate, no. But normally we give prebiotic for the survival of the organisms. So by giving additional nutrition, it may multiply more. Maybe I am just thinking. I'll tell you, we use earthworms here to degrade the vegetable waste here. That means the microbiome of the earthworm is capable of degrading all and producing a good fertilizers. We are in tons we are preparing actually here. We have a vermicompost we have here. So that's one thing. Then there is a practice by some of the general practitioners who, when they give antibiotic, we know that is going to kill not only the pathogenic, so they give lactobacilli along with their coats, thinking that it will at least control the diarrhea of the patient. See, most of the people when they take antibiotic, they develop diarrhea. To prevent that, lactobacilli has been given. And some physician also advise for as you grow older, along with your other vitamins or B complex when you take, they also advise a capsule of lactobacilli every day. They say it is good for your digestion and for your health. It's not going to harm you at all.
No,
so they say that will be more useful. This is being practiced by some.
Correct. So actually, you are right that when we give antibiotics, it kills all of them. So they always prescribe some probiotics along with that, lactobacilli or the bifidobacter. So because you don't want to disturb those good bacteria. So that is definitely that is useful. For
elderly people actually.
For the elderly, that is also because the elderly you want the good bacteria.
Microbiome will change actually.
Correct. So for them also I understand, but when it comes to cancer, we don't know when we give the probiotics whether it is going to affect. So if suppose we take the cancer which is infecting the say GI tract, whether that probiotic is going to be useful or not, because again it depends on how it's going to affect the microenvironment around the cancer tissues, for example. We don't know yet.
If you identified the microorganism which is causing the cancer, and if you think that this particular probiotic is going to help it, then it may be.
Then it is. So yes. We don't have that studies. Those
we don't
studies
have that study yet.
If once we do the studies, we should
then
be able to. Then it will be useful. Yeah. For the cancer, that's why I said that for cancer. But for the geriatric patients, say geriatric subjects, and for the antibiotic given patients, this probiotics given. I mean, those are very, very common, and I think that should be given.
Any other questions?
There's one. Oh, good. They've woken up. I didn't cover the AMR part. Okay, so now the one which we are right now doing a study with Lady Harding Hospital, we are looking into the neonates who are born, preterm, and they normally have, I mean, they are all in the NICU, and they are given different antibiotics because that's the only thing which, in order to avoid the sepsis, right? And we are trying to study the, their microbiome and trying to see which are, because some of them develop sepsis. We are trying to understand which are the neonates which are likely to get into sepsis and which are not likely to. And this actually is the observational study right now. And based on that, we will be able to say that if you get the microbiome of the neonates in these, then certain antibiotics should be avoided and so on. So that's the really aim what we have. But right now it's in the preliminary stage what we are doing.
Thank you, Madam, for answering the queries.
Thank you.
It's now time to express our gratitude. I request Dr. Rekha Pedi, Chairperson, MiCON 2025, to present a memento to Dr. Sharmila Mande as a token of appreciation.
Oh, this is your
Yakshagana.
Okay.
Yeah. That's of Mangalore region.
Mm-hmm.
This is the MiCON cup.
Okay.
Yeah. Thank you.
Thank you.
Really nice.
Oh, thank you.
Thank you, ma'am. We will now move on to the next session. I request Dr. Rajesh P. Shastri, member of Organizing Committee MiCON 2025, to introduce the guest of honor to the gathering.
Thank you. It's my privilege to introduce Dr. Girish Badrinath Mahajan. Dr. Mahajan, a globally renowned microbiologist, biotechnologist, and visionary leader in the drug discovery, serves as a senior vice president at High Media Laboratory Private Limited. With over 27 years of transformative contribution to the pharmaceutical, biotechnology, and biosupplies industries, Dr. Mahajan has established himself as a pioneer in advancing biomanufacturing and developing innovative fermentation technology, biosimilars, and specialized media formulations. An alumnus of prestigious Institute of Chemical Technology, Mumbai, Dr. Mahajan holds a PhD in microbiology. He has played a pivotal role in several global collaborations with the leading institutions including National Cancer Institute, Kansas University, and many more. His expertise in drug discovery has led to identification of novel antimicrobial, anticancerous, and anti-inflammatory compounds which have significantly advanced in therapeutic research. Dr. Mahajan has also contributed substantially to the international scientific collaboration as a key leader in the Indo-German Science and Technology Center grant initiatives. With an impressive portfolio of 20 patents and over 175 publications in reputed journals, Dr. Mahajan is a prolific innovator and thought leader. He has mentored 28 postgraduate interns and successfully led multiple BIRAC-funded projects that have propelled industry advancements. His excellence in pharmaceutical research has earned several prestigious awards, including OPPI Scientist Award, the AMI Louis Pasteur Award, and UAA ICT Distinguished Luminous Award in Research. Dr. Mahajan is deeply involved in integrating academia and industry. His leadership and exceptional contribution to the drug discovery and dedication to the global scientific collaboration underscore his legacy as luminary in microbiology and biotechnology. With this brief note, I request Dr. Mahajan to deliver invited talk. Over to you, sir.
Hello. Am I audible now?
Yes, sir, you're audible, sir.
Okay, great. Thank you so much. So, first of all, good morning to all of you. A big thanks to Dr. Rekha ma'am and Dr. Bhagwan sir, and who has invited High Media for this forum and helped to share, would like to share something about the microbiome and probiotic. And already Dr. Sharmila ma'am has set a nice stage about microbiome, probiotic, and kind of research and the work she has already displayed. So it really set a good stage for my speech. So I'm just sharing my screen. Please allow me to share my screen.
It's allowed, sir. Please share.
Can you see my first
Yes,
slide?
sir. Yes, sir. It's seen.
Great.
Can you just, go to the next slide, please, sir?
Yeah.
Yes, yes, yes. Yes, sir. Thank you.
The problem is that my screen is hidden to myself. I have to remove this part. There is one bed, I think that is good. So let me tell you about from where I am coming from. So I am from High Media, as sir has already introduced, and we have the manufacturing facility across Maharashtra in many places, and we have also the main warehouses and office at Germany, USA, and Brazil, and we have our presence of different products in more than 150 countries. As a High Media brand, we have 7 verticals: microbiology, cell biology, plant tissue culture, molecular biology, hydroponics, chemicals and biochemicals, and consumables and equipment. So we are among the just 1 second, there is some issue. Hello, हाँ sir. कोणाल सर? नाही रमाकांतला मी फोन करत होतो, फोन लागला नाही माझा. मी हो या. So we are among the top 3 global powdered media manufacturers. We have more than 30,000 SKUs and more than 1,000 tons media produced per year. So media has been there in as an Indian company from last 51 years. We have a legacy of 51 years. Started with microbiology and diversified in many fields. So as a product, if you ask, 7,000 plus products are there, and we have DSI recognized R&D labs since last 25 years, and we have various quality certifications, ISO 13485, WHO GMP, 6 plants are there, ISO 9001, ISO 27001, and ISO 17025 accredited by NABL, or various services are accredited by NABL, and ISO 27001, and also registered with US FDA to supply to US, and marked with IVD CE mark to supply to Europe. And also we have been the distributor, authorized distributor for various ATCC cell lines and plants, ATCC cell lines as well as bacteria, DNA, RNA, and various media from ATCC. One of the world's best microbiology media manufacturer, and we are the leaders in India. So we have world-class manufacturing facility, means as on now, today we have a capacity of creating a batch of 60 tons per day in our Nasik factory. Also we have a small research center on microbiome at Nagpur. Now, coming back to our main topic, microbiome research focuses on studying the community microorganisms such as bacteria, fungi, viruses. Basically it is bacteria and fungi that inhabit a specific environment. These microorganisms play crucial roles in maintaining the health of their host and surrounding system. Microbiome research delves into understanding the diversity and function of microbial communities, their interactions with the host, the impact of external factors such as diet, climate, and pollutants on microbiome. Now basically the importance of these microbiome studies lies with respect to health and sustainability. With respect to gut microbiome, the balanced gut microbiome is vital for digestion, immunity, and already Madam has told many other things. Therapeutic wise, microbiome based therapies are precision medicines, and we can also call it as a personalized. Infectious disease management, studying the pathogenic and commensal microbial interactions aids in combating the antibiotic resistance. With respect to sustainability, agriculture microbial solutions which promote the sustainable crop growth, reduce the chemical dependency, and improve the soil health, especially the kind of biofertilizers, pesticides, biostimulators which have been used. Not only microbiome but the prebiotics also. With respect to environment, microbial communities are used in bioremediation to break down pollutants, restore ecosystem, and recycle the waste. Climate change mitigation, microbes contribute to carbon cycling, helping regulate greenhouse and gas emissions. Many times we read about microbes and microbiome research, but the history is again a little bit important, how it came down to today's stage. So if you see the evolution of microbiome research tool, the techniques, early microscopy, initial studies relied on basic microscopes limiting the understanding of microbial diversity. Staining techniques are developed because these are the basic techniques where the one started studying various kinds of microbiomes. Culture-based methods followed by molecular biology revolution, especially techniques like PSR, ribosomal RNA, and especially the bacterial 16S RNA, ITC region, and 18S RNA, which are actually the gold standard for studying the microbial phylogeny and diversity. Later, the omics era came in the 21st century. Metagenomics allowed the study of all genetic material in the microbial community, overcoming the limitations of culture-based method. That's very important. Means you don't have to cultivate all those, which are sometimes the challenging part. Proteomics and metabolomics enabled the analysis of microbial proteins and metabolites, revealing functional roles in the host systems and ecosystems. High throughput sequencing, next generation like NGS, revolutionized the microbiome research by providing faster and more cost-effective methods to analyze microbial communities. Now today, advanced computational tools and AI modern era, bioinformatics pipelines and AI-driven data analysis actually allow the deeper and insights into complex microbial ecosystems. These tools help model host microbiome interactions, predict functional roles, and identify novel microbes. Strategic for culturing approaches, because ultimately only genomic data and measurements is not important. To make them applications, make them useful, it is necessary that we have to apply the various cultivation techniques. Enrichment culturing methods for rare taxa and techniques like selective media designed to favor the growth of specific microbial groups by modifying nutrients or environmental conditions, like adding various carbon sources, nitrogen sources, antibiotic. Gradient enrichment mimics the natural environments by using a gradient pH, oxygen, or nutrient. High throughput dilution to extinction methods enables isolation of single rare taxa by extensive dilution in microtiter plates. Now, application of all this to discovery of the novel microbial species. Not only data, metagenomics data, but novel microbial species which we can cultivate later and use for the specific application. And also uncovering the unique metabolic pathways. Of course, health-wise applications are numerous. If you Google it, you will find a number of applications, especially in gut microbiome in human, obesity, diabetes, mental health, probiotics and prebiotics, which are the strategic formulation and for delivery. And also we look for data from, there is one program, Earth Microbiome Project. So it has compiled a lot of data. So they have published first large-scale analysis of data from Earth Microbiome Project, a truly multidisciplinary research program involving more than 500 scientists and 27,751 samples acquired from 43 countries. So these samples represent a myriad specimen types and span a wide range of biotic and abiotic factors. So perhaps some of you who are interested in look into that. Obviously, when I say microbiome research, it culminates into a product which is called probiotic. So as per NIH, the definition is probiotics are live microbes that are intended to have health benefits when consumed or applied to the body. They can be found in the yogurt and other fermented foods, dietary supplements, and beauty products as well. Now, one more term which is go hand in hand with that is prebiotics. These are the non-digestible food components that selectively stimulate growth of or activity of desirable microbes. And there is one more term called as symbiotics, are products that combine probiotic and prebiotic together when a particular formulation is given to the human. Now, ultimately, the whole microbiome research till now has culminated into many probiotics. So this is a list of various bacteria which are currently in the market in the different formulations, which includes basically many lactobacillus species, including Acidophilus, Casey, Romances, Parsi, Sacchet, and also there is a Leuconostoc species, Streptococcus species, Bifidobacterium species, and others like Bacillus cereus, Escherichia coli, Lysel, Saccharomyces cerevisiae, and Saccharomyces boulardii. Not only that, we are working for human microbiome, plant probiotics is equally important, animal probiotic is equally essential. Now today, the research in that area is also blossoming, and several probiotic products are available for both these categories along with the human. Now, the reason the way the probiotic is moving, it is creating a huge market. So in 2018, it was about $49.4 billion, growing at a rate of 7% per annum. And it is expected that by next year, it will be around 84.85 billion. From this, you can imagine market growth is actually attributed to the rise in demand of probiotics in food, beverages, and dietary supplements, particularly in the emerging economics of Asia Pacific and RO. Now, our main focus with respect to product, I am saying, research-wise, it is multidisciplinary, but currently main focus is on the human gut microbiome. So, collective genomics of microbes, including bacteria, bacteriophages, fungi. And it is said that often the human gut contains 2 kg bacteria, 3.3 million genes, which is a metagenome data. Intestinal tract contains about 100 trillion microorganisms, majority in the colon. Healthy human has different kinds of family of bacteria, especially the firmicutes, bacteroids, proteobacteria, actinobacteria. And if I check the ATCC data, they have given 10 to 100 trillion microorganisms in the gut itself. The weight is around 2.5 pounds, and more than 1,000 species are there, and unique genes, and the ratio of the number of bacterial cells to human cells is 1.3:1. It means if there are 13 cells, bacterial cells, number of human cells is 10. Now, these various bacteria, why they become suddenly popular, means it is important in, you can say, disorders like diabetes, obesity, and sometimes a kidney stone also. So, because they are producing certain metabolites which are actually converting some bioactive things. Example, here is one example, like there is a drug called sulfasalazine. It is activated by certain enzymes in the bacteria. There is one more, methotrexate, which is actually converted into inactive metabolite. There is one more drug of the glucuronide category. So there are certain enzymes produced by microbes which convert into enterotoxic metabolite. So all such specialized molecules which are generated by the microbes, they are giving some advantage to the gut, and that's why it has becoming more popularized. The objectives of the various stakeholders in the probiotic field. One, scientist want to generate high quality science that has positive impact on the society. Industries want high quality profitable products which with validated and ununderstandable claims. Regulators want to protect consumers from misinformation, and consumers want reliable information to make informed decisions. So all this culminates into very brand probiotics. Possible and established distribution of mechanisms among the probiotics. See, if you look at the base of these mechanisms of probiotics, the most widespread is among the sterile probiotics, that is colonization resistance, acid SCFA production, regulation of intestinal transit, normalization of the perturbed microbiota, increased turnover of the enterocytes, competitive exclusion of pathogens. Frequently, other data comes with respect to the specific species, vitamin synthesis, bile salt metabolism, direct antagonism, enzymatic activity, gut barrier reinforcement, neutralization of carcinogens. And very rarely there are some strain-specific data which appears in the literature, neurological effects, immunological effects, endocrinological effects, production of specific bioactive. So this forms a different layers of the applications of the different kinds of the probiotics. That's why probiotic literature is seen starting from drug resistance overcome to the autism overcoming. So that type of data is available. Again, if you ask, in the probiotic industry, it is a live cultures as the definition goes. Again, probiotic involves probiotic drugs, probiotic medical foods, non-oral probiotics, probiotic animal feeds, defined microbial consorta, probiotic dietary supplement, probiotic infant formula. And of course, non-probiotic, already we have been using fermented food with undefined microbial content, undefined consortia, including fecal microbial transplant. When you look from the perspective of probiotic manufacturer, so the ideal probiotics should be non-pathogenic, lactic acid producer, production of antimicrobial substance, adherence to gut lining, genetically stable, safety in food and clinical use, susceptible to antibiotics, persistence in human intestinal tract, antagonism against enteropathogens, acid and bile tolerant. Now, some of the other examples of prebiotic example, like as a definition already I mentioned you, it involves various fructans like inulin and fructopolysaccharides or oligofructose can stimulate lactic acid bacteria selectively. Galactoolisaccharides, the product of lactose extension, GOSs can greatly stimulate bifidobacteria. So different kinds of prebiotics can support a different kind of the genera of the microbes. Starched glucose-derived oligosaccharides, they are again attract Ruminococcus brommi, Bifidobacterium adolescensis. Non-carbohydrate oligosaccharides like cocoa-derived flavonoids in vivo and in vitro experiment demonstrate flavonoids can stimulate lactic acid bacteria. So again, as Dr. Sharmila ma'am also mentioned, that not all oligosaccharides or prebiotics will attack all bacteria. Again, it depends upon what kind of gathering of the bacteria you want. Based on that, prebiotic formulations are decided. Now, within the probiotic, one of the two best examples I can quote here, Akkermansia myciniphila, a gram-negative anaerobic mucus layer degrading bacterium that colonizes intestinal mucosa of human rodent metagenomic strata, have shown inverse correlation between the abundance of A. muciniphila and disease such as inflammatory bowel disease, obesity, and diabetes. So, Akkermansia has become the buzzword for all those who are interested in probiotic. Oxalate degradation by anaerobic bacterium, Oxalobacter formigens, is important for human health, helping to prevent hyperoxaluria and disorders such as development of the kidney stones. So there are good number of formulation using such bacteria for diabetes, obesity, and even for the kidney stones using this Oxalobacter species. Now again, what role high media plays in supporting the probiotic industry? So within the microbiology, we have 20,000 SKUs comprising of various media. We have a range of high-wage media because again, probiotic need is that it should have a high-wage ingredients. And we have more than 1.5 lakh ready-prepared media means capacity per day. We have more than 75 high-chrome media. Now, then we did a survey of various probiotic manufacturers industry. So some of the challenges what they faced from the probiotic industry, stringent need for optimum growth culture. Means even if the raw material specifications are slightly here and there, the yield suddenly decrease because all the game of probiotic industry depends upon the yield. If they want 10⁻10 CFU per ml in a particular batch, it means they want that much. If it goes by even single log, it get affected. Second problem what probiotic industry face and where we play a role is batch to batch consistency of the raw material. Means if the ratio of carbon to nitrogen or if there are some other impurities in the raw material, that becomes detrimental to the yield of the whole cells of the bacteria and fungi. Compatibility of the various raw material with each other. Again, this is a very important part. Means one example I can quote you here, like a brand of yeast extract and a brand of peptone. They becomes incompatible when they are used for a probiotic yield. But if I use a specific peptone and specific yeast extract together, then only there is a good yield of the probiotic. And one very important, which majority of the industry face, is a fast bee cultivation. Means if there is a phage attack, the probiotic yield become watery. So the log cell, it reduces like anything. So high media primarily helps in providing a range of the different raw material for this purpose to overcome the 4 major challenges. We also supply the customized fermentation media, formulation and feed under the confidential agreement with the parties. Means if the party wants based on research, okay, this particular 3 things to be added to the media for my probiotic. So we have the customized media and we can give a single batch of 60 tons so that for complete year that is sufficient for the party to use that. Then we ensure growth promoting properties by extensive and intricate testing. A bigger batch size capacity ensures consistency and a scaled up supply. Moreover, we also test compatibility of raw material used in the finished product. Moreover, as I mentioned, phage is a big problem in probiotic industry. So we are providing the raw material at least which is free of phages. So we ensure that the raw material which is supplied is free of it. So we are providing gamma irradiated or other techniques we have by which we ensure that phage is not there. These are the range of various raw material. I just put as an example. and majority of them are high wage based. We have some very specific media supporting the growth of the particular probiotic in a quantity. Example, Hychrome bifidobacterium agar M1960. So this is an example of having the big and very clean colonies of bifidobacterium infantis. This is Lactobacillus mrs agar, which is supporting the growth of Lactobacillus fermentum and many other Lactobacillus species. So this is Hychrome bifidobacterium agar, and also it supports Lactobacillus plantarum. So in fact, Hymedia as a company, like those who are working with microbiome and probiotic industry, so we are like a one-stop solution for all probiotic manufacturing supplies. Like we supply various sugars, lactose, dextrose, dextrine, maltose, sucrose, fructose, raffinose, glycerol, acidic food grade antimicrobials like acetic acid, lactic acid, propionic acid, sorbic acid, benzoic acid bacteria, various kinds of nitrogen sources like peptones, amino acids, encapsulating agents which are required in making various formulation, salts, and various other nutrient sources like glycerol, useastat. Apart from this, they need a various solvents. So we supply that also, which are specifically for the probiotic industry. So apart from this, as I mentioned, again, majority of the probiotic industry prefer use of the wage bacteria. So we are providing the high wage bacteria, high wage peptones, and high wage peptones containing media. Because as all of us know, because animal origin raw material, they lead to transmission spongiform encephalitis, simply called as TSC, and bovine spongiform encephalitis, that is PSC. Plant peptones from high media reduce the carbon footprint as well, generated by animal peptones, and eco-friendly, high-yielding, and comprehensive range of the plant peptones. Because we have a data which clearly shows that there is a high yield when I use a high-wage peptone compared to the animal peptone. Apart from this, we are also supporting. One of the biggest challenge in study of the gut microbiome is a very authentic anaerobic environment creating system. Now we are providing automized anaerobic system which is called enoximent. So it not only supports obligate anaerobes, microaerophilic, capnophils, as well as if you want a particular ratio of anaerobic system, that ratio can also be maintained. So this has boosted the microbiome research across India. So we have already supplied to many of the microbiome workers. Moreover, high media also supports the trials of both aerobic and anaerobic fermentation. If you have a particular microbiome in your research center or in your facility and you want to scale it up and take a batch and then give trial for clinical trials, we support that. So we have a very pilot system up to 10 liter and also we have up to 50 liter. And we are also constructing a facility for 1000 liter capacity for all the probiotic work going on in India. Again, as a company, we have been the authorized partner of ATCC, American Type of Culture Collection. So they have a repository of microbial strains, over 18,000 strains of bacteria, and various microbiome standards which provide authenticated strains of metagenomics studies, develops mock communities. So I'll just give a click here. Again, site-specific microbiome standards are available. So these are the actual cultivated microbes which are coming out as a standard, like oral microbiome, skin microbiome, gut microbiome, and vaginal microbiome. So these products are available with us. And plus, ATCC microbial also has mock microbial communities and site-specific standards. So there is a growth quantification mechanism also, and these various growth factor mix are also available. So if somebody is working on microbiome, so we have all supporting data as a reference to show that, okay, what you're working is going on very smoothly and as per the requirement. So I would request you, those who are working on microbiome should go through our site and ATCC site on this part. So ATCC has a high quality probiotic strain, supplies well characterized strains like Lactobacillus bifidobacterium, verified for identity, safety, and probiotic functionality with a full sequence data. Probiotic standardization sets benchmarks for strain authentication and viability, offers quality standards for industrial probiotic formulation, and finally supports research in FDA EFSA, and other international regulations. ATC's impact is very high in microbiome research. The cornerstone of microbial and probiotic research globally ensures reliability, reproducibility, and regular adherence. And continued development of microbiome standards and novel strains repositories going on at DTCC. And today, it's not surprised that there are so many probiotic brands are available, which is a culmination of, ground level microbiome research. So these are the various brands. And, next microbiome application is in air. If you see the Scientist magazine of Jan 22, it means a couple of days ago. So there is a gut bacteria boost levels of 6 hormones with help of flatulence. It means this microbiome study is going everywhere almost. No, somebody asked the definition, what is air, which is the thing which is everywhere. So in the same way, I can say that in a lighter note, what is microbiome? It is like everywhere it is present in all aspects of our biology. Future trends, I would just summarize in a short way. Advancement in synthetic biology for microbiome engineering and use of nanotechnology in microbiome studies. So we have some recent couple of papers in this area because we have to constantly update ourselves to support all the researchers in India for the new things. So thank you so much for your kind attention for my presentation. Any questions? The forum is open.
Thank you, sir. That was an informative talk and for updating us on your new products. So the session is now open for discussion and any questions from the audience.
Any questions?
Okay, if there are no more questions, thank you very much, sir, for this update. And thank you once again. Have a great day.
Okay, thank you so much. Bye, everyone.
Bye, sir. So let us now break for some refreshments and we can assemble back in another 10 minutes. Yes, ma'am. There will be oral presentation. The next session, please assemble within 10 minutes. And those who are presenting orally, please give their presentations here. Please submit it.
Bina HB is there. Bina HB. Prevalence of?
It is open. Just a minute, yeah. It is opening, ma'am. This is yours, right?
Yeah.
You want to check the presentation?
Because it's looking a little, I mean, font is a looking, slight changes there in the font, I think, anyway. No problem, let it be. Anyway, it can be seen, no?
Okay, ma'am. Do you want to convert it to PDF and then show?
No,
this is
fine. Normally, how have they done?
We have all presented PPT version
PPT only,
yeah,
no? Better, yeah. Come down to the last, next. Yes, fine. Okay, it's opening. As long as it's opening, there's no problem.
There are no animations or anything.
No, I didn't put some. Yeah, fine. Presentation number 3, right?
The house is 3.
We will now name
We'll
like
name
that.
it like that
This MOP3.
Then Bina?
Bina, open it. Prevalence of expanded spectrum, that only, no? School of Life. Yes, that is MOP1.
She's here? No.
I don't know, but that is
MOP1, no?
MOP1.
Okay.
Bhagyashri is there.
Yeah, Bhagya Yenepoya conference.
That is Bhagyashri only.
Yes.
A comprehensive study on paradoxical.
You want to check?
That one? Okay, yeah.
This one? Yeah.
Because then it will be easy for them, no? Which you have to remember your presentation number. Here no presentation. Done, no? Saved, right? This is MOP3, right?
MOP2.
MOP2.
Pen drive
Correct? MOP4 is there? Anybody? J Lakshmi. Which name? That only, right? Correct? Right, MOP4. Yuvraj. MOP5 is there? Your
name? Yeah. Minu.
This is MOP? MOP5. Who is MOP6? Irfa Masum. Irfa. This one. MOP6 is the microbiology for the element potential benefit. Okay. So, MOP6. So, for morning presentation, all we have 1 to 6.
This and all, afternoon ka?
Afternoon ka 3 people I didn't get.
Is it? Yuvraj also it is not there.
Yuvraj will give later. You'll give now?
Yeah, that only 2.
No,
I put now only.
Except 7, 8, 10 I didn't get.
Okay.
Yours also will put. Yours also will put. Otherwise, you will open your mail only, no? Download it.
Okay. Take off. Then where you will manage? Now it will start.
You get, they don't give me second also. I wanted that to keep me my new room showcase.
You get award, you take that and keep it.
Copying here only.
Morning session, afternoon.
3 students I didn't get. I think they didn't register, came only. Otherwise, I would have made.
Just once.
There also, I didn't get their. Once more, I'll check. In the afternoon, sir, they will manage. Or you can announce. Whoever came, I was asking everybody. Yes. You can announce those. Yeah. So please submit. Sir, morning session. Registration committee. Wait. Which one you didn't get? 7, 8, 10. You take this. Take a chair.
Presenting, sir. Giving.
Afternoon last.
None month.
Why this photo?
Why?
This much only our team.
I found what improves p value and not needed.
You put that photo.
Hmm?
You put that.
Oh, you are not good, huh? No one will say.
Without me, one you took now. Please put that.
Hmm?
One more is there without me. That you put, please.
What, yeah? Without you, but you are in group.
Advocacy has led to government's initiative in launching a direct benefit transfer scheme for all TB patients to enable access to a better diet. This center has developed maternal nutrition advocacy, implementation, policy and planning in collaboration with Ministry of Health and Family Welfare, UNICEF, National Center for Excellence and Advanced Research on Diets, Yenepoya Technology Incubator, aimed towards identifying, fostering and mentoring innovators, entrepreneurs in the healthcare innovation process. Recognized by the BIRAC, Yenepoya Technology Foundation supports startups and more than 60 patents have been applied, published or awarded. The university has signed more than 150 MOUs with several national and international institutions of excellence for research, faculty and student exchanges. The 4 hospitals cater to the teaching and training activities of medical, dental, Ayurveda and homeopathic disciplines. In addition, there are yoga, naturopathy and wellness centers provide treatment and training facilities. The Medical College Hospital is NABH accredited with 1,100 beds with the latest facilities for robotic surgery, organ transplantation, 3T MRI, modular operation theatres, ICUs, and emergency services. The diagnostic laboratory is NABL accredited, covering services of biochemistry, microbiology, pathology, and transfusion. The Allamilu Charitable Foundation, supported by Tata Trust in association with the Yenepoya University, is establishing a comprehensive cancer care center. Medical College Hospital provides free food, free and subsidized treatment to all the patients. The hospital has received several awards from government and non-government agencies for its outstanding contribution to public service and workplace safety measures. The university provides equal opportunities to all students, upholding the principles of equity. The observance of cultural fest, religious festivals, NSS activities, and disabled friendly and barrier-free environment provide the opportunity for students from the diverse background to effortlessly blend with the campus life. The international student cell caters to the needs of overseas students to seamlessly adapt to the campus environment. The Student Council provides a platform to undertake various co- and extracurricular activities. The university provides scholarships, fee waivers, and fee concessions for financially weaker students and promotes gender equity. Career Guidance Cell prepares the students for entrance competitive recruitment examinations. The Placement Cell, in association with Alumni Association, organizes campus placement drives. Students participate in extension and outreach activities through camps, awareness programs, and have adopted several villages for comprehensive healthcare support. The university has created excellent sports and auditoria facilities to support the physical and recreational requirements of the students and faculty. Endurance Zone. The indoor auditorium has a multi-purpose hall, basketball court, shuttle badminton, volleyball, gymnasium, squash court, table tennis, and board games. The outdoor sports facilities include international standard grass soccer ground, cricket practice nets and stadium, kabaddi court, throwball court, and athletic stadium. There are 13 hostels for students, 7 for women and 6 for men, and are provided with solar water heating, uninterrupted power supply, and Wi-Fi facilities. The centralized kitchen, food craft, caters to the food requirements of students, staff, and patients, along with 2 canteens and 15 cafeteria spread across the campuses. The laundry services, laundry line in the university campus has capacity to handle 5 tons of clothes, is a boon for students, staff, and patients. The campus has several environment-friendly operations which include rainwater harvesting, water conservation, waste management, energy conservation, and promotion of alternate energy utilization. As an endorsement for these efforts, the university has been recognized and awarded by prestigious international, national, and state bodies and is amongst the top 400 institutions ranked by Times, the Global Impact Ranking, which mainly focuses on achieving sustainable development goals. The university has adopted multiple UGC programs to sensitize and inculcate social responsiveness among the students and cross-cutting issues like gender, environment sustainability, human values, health determinants, right to health issues, emerging demographic changes, and professional ethics. Commitment to our nation and society is embodied in telemedicine initiatives, relief activities during environmental disasters, and the COVID-19.
Welcome back. We will now begin with our next session, that is the oral presentations. Kindly be seated. Participants, kindly note, Each presenter will have a total of 7 minutes. That is 5 minutes for presentation followed by 2 minutes of discussion. Also, due to the paucity of time, we would request all the presenters to please limit their respective presentations within the allotted time. So the distinguished panelists for this session are Dr. Sharmila Mande, Ayush Distinguished Scientist Chair, Ministry of Ayush, and Dr. Keshav Prasad TS, the Deputy Director of CSBMM, and Dr. Rajesh P. Shastri, Associate Professor YRC, will be moderating this session. Over to you, sir.
Thank you. So we have 6 presentation. So we have 6 presentations and 2 chairs. We have Dr. Sharmila Mande and Dr. Keshav Prasad. I request first presenter, Dr. Beena.
So the presentation is for 5 minutes, followed by?
Yes, for 5 minutes presentation, 2 minutes question and answer.
5 and 2.
Yeah.
So I would request if
Yes,
you
madam.
press the bell after 4 minutes, so that they know that that you have 1 more minute, and second long bell.
Over to you, madam. Yes.
Good morning, good afternoon, good morning, ma'am. This is, my presentation is on prevalence of extended spectrum beta-lactamases and carbapenemases harboring Klebsiella pneumoniae in intensive care unit of the tertiary care hospital from South India, a molecular approach. So all of us know, like in Western countries, usually when the intensive care, this exclusive study was done in the intensive care unit, so I'm sticking on to the intensive care unit data. So in the intensive care unit, usually in the Western countries, all the superbugs like MRSA, vancomycin resistant enterococci and pneumococcus, penicillin resistant pneumococci usually, so they will cause the major illness. But in India and Asia specific regions, commonly the gram-negative superbugs such as harboring ESBL, AMC and the carbapenemases will dominate. And all of us know this study is on the Klebsiella pneumoniae as an opportunity pathogen, produces lot of infections in the intensive care unit and also the nosocomial infections. Due to the multi-drug resistance caused by these organisms, usually these gram-negative organisms are treated by the extended spectrum beta-lactam antibiotics, but due to the emergence of extended spectrum beta-lactamases, this inhibits the action of these and extended spectrum beta-lactam antibiotics on these organisms. So in the emergence of these ESBLs, the carbapenems and the cholesterins were used as the drug of choice, but later due to the emergence of carbapenemases, and these drugs also nullified in the use of gram-negative infections. So this drug resistance causes a great threat to the clinician as well as the economic burden to the patients residing in the intensive care unit. So the aim of this objective is antimicrobial resistance profile of clinical isolates of Klebsiella with special reference to ESBLs and carbapenemases, and the objective was, the study was to analyze the prevalence of ESBL and the carbapenem resistant Klebsiella pneumoniae in the intensive care unit, and also to identify and characterize Klebsiella pneumonia harboring resistant genes. The inclusion, all the clinically significant Klebsiella was isolated from the clinicals which was received from the intensive care to the laboratory, and these were assessed by the gram staining with looked for the polymorphonuclear cells along with the gram-negative bacilli. The surface contaminants and the colonizers were excluded, and also the non-repetitive isolates. This study was conducted at the Microbiology Department of Manipal Hospital, Bangalore. It was a 2 years duration, to 2016 to 2018. It was a prospective cross-sectional study. So sample size was 347, and the study isolates were all the samples which were received from the ICU to the lab. And the statistics analysis was analyzed by the SPSS version of 25. Software was used, and all the variables were analyzed using this Pearson chi-square test and the Fisher's exact, and it was found p-value less than 0.05, considered clinically significant. The clinicals, these are the clinical samples which were used, all the respiratory samples, ET aspirate, ball, sputum, blood, pus, and the sterile body fluids, CSF, intravascular catheter tips, and the urine. And the phenotypics, the sample was subjected to the culture, and before culturing, the gram stain was performed, looked for the polymorphonuclear cells and the gram-negative bacilli, and the culture was done on the as per the normal protocol of the microbiology department as per the CLSI guidelines, and this was McConkey, agar, and the blood agar, and the respective to the samples we have used all the culture media, and they looked for the mucodelactose fermenting colonies. Biochemical reactions were performed, and methods, manual as well as the Vitek 2 compact, and the antibiotic susceptibility test was done as per the CLSI guidelines, and also for the detection of ESBL we used the combination discs like ceftazidime and cefatoxim with the combination of clavulanic acid, and the AMC also was detected phenotypically with the use of cefoxitin and the cefoxitin boronic acid, and the modified Hodge test, and as well as the indirect carbapenemase test using imipenem was done for the detection of carbapenemase production, and as well as the agar dilution method was done for the meropenem as well as for the colistin drug. And this is the genotypic methods, and the ESBL detection, and we have done for the TEM, SHV, and the CTXM gene, and the carbapenem resistant isolates were genotypically identified for the NDM and the KPC gene. These are the visuals of the gel electrophoresis of the PCR. It was ESBL was used by the multiplex PCR, and the NDM KPC was done by the uniplex PCR. In the results, we found that when most of the isolates which were received from the ICU, and the ESB and these isolates were isolated from majority from the medical intensive care unit and others, and most of the samples used were, majority of the samples were these isolates were identified from the blood as well as from the respiratory specimens. And in the
Time's up.
Yeah, in the ESBL, and we found that 82.3% were the ESBL organisms, and 17.7% were the non-ESBL producer, and we found that SHV is the majority of the gene produced, and then carbapenem resistant isolates, 61.9% of the carbapenem isolates were positive for NDM and the KPC, and we found that antimicrobial resistance is due to the multiple group of antibiotics. Overall, colistin resistant by agar dilution method was 27.21%. We found that this, because of the SHV gene is a plasmid mediated which causes intrinsically resistance, causes the drug resistance, and all MIC of the meropenem and the colistin were found to be in the higher side, that is 4 microgram to 128. So on the ICU, we thought that nosocomial infection, these organisms caused drug resistance may be due to the prior hospitalization, indiscriminate use of antibiotics, as well as due to the mechanical ventilation. These are the variables which found significant in this study, because we could not, and because most of the carbapenem resistant, even the modified Hodge test and the isolates were positive, but we could not identify the other genes, as per only the KPC and NDM because of the funding, and this is further study maybe to the other genes, OXA48 and VIM and S. Thank you.
Thank you. We have 2 minutes for question and answer. Yeah. So, any question from the audience or anyone?
small question like,
Yes,
but
ma'am.
ICU generally have some preventive hospital de-infection strategies. So why still you are finding?
Maybe that's what in our study we found that, ma'am, the patients, we have taken after 48 hours of admission all these isolates when they from the ICU setup, and we thought from the analysis could be the patients were usually the hospitalized priorly, as well as they were loaded with the antibiotics when they came to this hospital. So could be that is the reason when they use the antibiotics here caused the drug resistance and they caused this.
So you showed some statistical parameters.
Yes, sir.
Where did you use them? Because you didn't show anywhere the statistical significance, some data.
So, yes, sir. It is there. So it was actually the
Where did you use it?
Yes, sir.
Where did you exactly use it?
It is for the variables like for the detection of the genes and in the samples, as well as in the identification, as well as the mechanical risk factors identification. We use the statistical significance.
So for genes, when you are telling that particular genotyping exercise, did you do it in triplicate?
Yes, sir. We have done it.
At the end, you just said that these antimicrobial resistance, which
Yes,
you said a few of the things it could be because of the antibiotic usage and also. Did you look into their history
Yes,
of
ma'am.
all the
We have-
patients?
yes.
And based on that, did you?
Based on that
say-
only this study was done.
But you
yes.
didn't show that,
Yeah,
what kind of,
that's-
how many
that-
percent of them
Yes, that I
had
did
a
put.
previous hospitalization
Hmm.
because of that. So, because the conclusion has to be supported
Yes,
by
ma'am. Yes.
some, observations which you
Yes.
would have done. But you have done it, but you
Yes,
didn't
ma'am.
show
It
that.
is
You should
not.
have
I
shown
could
that.
not add on to this slide, ma'am. Yes. Yes.
And all the future speakers, when you hear the first bell, try to conclude. Because next you have only 1 minute left. So
Yes.
you should not wait for the second bell. Second bell, you have to immediately stop.
Thank you.
Thank you. Next presenter is Bhagyashree from Department of Studies Microbiology, Davangere University.
Just to save time, I think you should just quickly the title has been read. You don't have to tell who you are from where, so that has been announced. So just to save time, because you have only 4 minutes.
So good afternoon, everyone. I'm here to present on comprehensive study on paradoxical zone phenomenon in marine actinomycetes. So I'll directly go to the materials and methods. Here I have collected the samples from Dakshin Kannada district, from various estuaries samples as well as mangrove samples and sand sediment samples. And later I had isolated them on selective starch casing selective media, where I had got about 24 samples, isolates, actinomyces isolates from these samples. And later on, for the characterization, we did it through the morphological, microscopic and biochemical tests, whereas for morphological, these isolates were characterized based on their growth on different selective medias like starch case, actinomyces isolation agar, oatmeal agar, gauze media and few other medias. And screening, that is primary screening through cross-streak method for checking the antibacterial activity, and for antifungal activity it was through dual culture method. So these two are RB60 and RB68 isolates are the ones which produce pigments isolated from Sasitlu beach. And this was on starch casing agar medium. and few of the isolates produced grey color colonies, white colors and light grey colonies, and pigments were dark green, dark brown color as well as black color pigmentations. So characterization through biochemical tests like citrate utilization, urease test, phenylalanine test, and triple sugar ion test, and various sugars were used to see whether they could they were able to ferment these sugars like raffinose, raminose, xylose, and sucrose, fructose sugars. So the primary screening was done on 3 different medias, that is nutrient agar, starch casein agar, and ISP5, that is actinomyces isolation agar. So you can here it is show 5 organisms were used, pathogens, that is Bacillus subtilis, Staphylococcus aureus, E. coli, Klebsiella pneumonia, and Proteus vulgaris. And compared to, here is the graphical representation showing that RB 17, 32 and 68 showed the highest antibacterial activity on starch casein agar plate compared to nutrient agar and ISP5 agar plate, and RB32 had shown the highest activity for Bacteria subtilis on starch casein agar plates, and here RB60 and RB68 showed the paradoxical zone phenomena or the eagle effect, that is these organisms may produce compounds that compounds where it can also decrease the compounds that are sorry. So they may produce compounds that can kill the pathogens at lower concentration, but at the same time when it is increase the concentration, they can also able to the pathogen organisms can survive at the higher concentration of the particular compound. So that showed the eagle effect on Proteus vulgaris organism. So this was on antifungal activity against Aspergillus flavus and Aspergillus niger. So the first plate was control, where the growth rate was 2.3 centimeters and Aspergillus niger 3.3, and compared to them RB6 showed the inhibition level, and RB32 also showed the inhibition level for fungal isolates against fungi. So conclusion here, in this work we managed to isolate novel strains maybe of actinomycetes, and among them 24 isolates, 5 of them displayed high antibacterial activity against 10 organisms. Each of them showed variations in antibacterial activity on different medias, and isolate 60 and 68 displayed paradoxical phenomena, and isolate RB 17, 32 and 68 inhibited exhibited the antimicrobial activity more on starch casein agar medium. So this was the primary step for the discovery of natural metabolites, and further secondary screening will be done for the identification and triggering them of the secondary metabolites. Thank you.
Questions?
Any questions from the audience?
What do you mean by paradoxical phenomena in its full spirit?
It's actually, it was first identified by H. Eagle in the year 1948, where he found it in a penicillin antibiotic against the streptococcal species. There, where he saw that during lower concentration of the antibiotic, it inhibited the growth of the staphylococcal species. But when they increased the concentration of the particular penicillin antibiotic, that resisted and the organism could grow again, that's what the paradoxical phenomena is. Like at the lower concentration, it can inhibit, but if the dosage is more than the optimal bactericidal concentration, it can grow again, the pathogen can grow again.
So you just showed that the different agar plates, the antimicrobial activity for some of those bacteria were high and some were low. Yes. Why is that? I mean, how do you explain that in different agar plates? What's the...
Maybe because the composition that is present in the particular media increases the production of that secondary metabolites that is present in the actinomycetes, and that inhibits the growth of a particular pathogen organism.
So have you looked into those secondary metabolites, or you plan to look at? Plan to. Okay, good.
So what is dual culture method?
Where dual culture, where test organism and the actinomyces that is placed, it's placed at the same time, but there will be 3 point inoculations where both the sides will be isolated organism and a middle will be the test organism. So that's how it whether it inhibits the growth of that particular test organism.
Do you have any example of active components isomycetase which is used in the clinic?
Thank you.
Thank you. Thank you. So I request audience keep silence during the oral presentations. So next presentation by Ethel Suman from Department of Microbiology, Kasturba Medical College, Mangalore.
Which is this? Next. One minute. Press this, sir. Start. Enter. No. Good afternoon. My topic is coagulase negative staphylococci, a commensal or pathogen. As we rightly heard in the presidential message itself, this coagulase negative staphylococcus is a commensal. Most of the times we don't give importance when we isolate it in the clinical specimen. But the importance here in our study is to know why do we have to report commensal staphylococci? Is it important? And if it is important, why? What is the necessity to report? So in this, the aims were mainly to study the various species of coagulase negative staphylococci, which were isolated from clinically relevant samples, and to study their antibiogram, to detect the slime production by qualitative method by using the Congo red agar method, and also to study the biofilm by the microtiter plate assay of O'Toole and Coulter. And finally, we wanted to mainly see the genes which were responsible for this slime and biofilm production, which is said to be an ICA operon, which is mainly found in Staphylococcus aureus, but nowadays we also see them in the coagulase negative staphylococci.
This ICA-A and ICA-D, they actually code for an operon, which is important, which is a polysaccharide intercellular adenosine, and it has been found to produce biofilm. So the materials and methods, whatever isolates we got from the clinically relevant samples, mainly from blood, from the ET, that is the endotracheal tubing, and from the deep tissues, wherever we thought it was clinically relevant, in the sense it was either only one which was isolated and with the heavy growth. So we took that and we identified by using MALDI-TOF and Vitek2 compact system, wherein we could isolate or we could identify the different species. Antibiotic sensitivity was done by the Vitek2 system, wherein we could also get the MICs, and this is very important for a blood isolate. So then we took those isolates, we ruled out all the commensals, like all the exclusion criteria where we excluded all the from the commensals could be the skin flora, where we thought all those were excluded. And then we detected the slime production by the Congo red agar method. So here, all those who produced black colored dry colonies were positive, red colored slimy colonies were negative, and in between were indeterminate. Then we did the microtiter plate method, and in the microtiter plate method, less than 0.120 OD were negative, 0.120 to 0.240 were moderate, more than 0.240 were positive, were high biofilm. ICA-A and ICA-D genes were detected by the PCR method. In the results and discussion, we found, as we can see here, out of the 100 isolates that we took in the study, which was done by a experimental prospective study, the hemolyticus, that is Staphylococcus hemolyticus, was 43%, followed by Epidermidis and Hominis. In the Congo red method, about most of them were slime producers, but we found about 7% who were, which were positive, and they were also strong biofilm producers, and most importantly, we found that they showed the ICA-A and ICA-D genes, and very importantly, what we saw was many of them were resistant to methicillin. And this methicillin resistance is important because it can transfer to the other Staphylococcus aureus, and also linezolid resistance and daptomycin resistance were seen in these species. So we conclude that these species have to be considered because they can transfer the drug resistance to the others. Thank you.
The session is open for a discussion. Any questions from the audience?
Yes, quality is negative, that is Staphylococcus saprophyticus, ATCC BAA 2, I think 24 or something, you know, I can't remember the, yeah.
Thank you.
Did you correlate the phenotypic results with the resistance, the identification results?
Yes, it was correlated, and wherever ICA-A and D were there was higher resistance and higher biofilm.
And this identification is done by MALDI-TOF.
MALDI and White.
Right.
White.
And is it 100% accurate to the species level, or?
MALDI is the species level.
Yeah. Because some of them are not routinely in the clinical
yes,
form.
That's
no,
why
that
we
is
could,
why
yeah, that's why we could, yeah. Yes.
All right. Thank you.
So whether you used MALDI for drug resistance
no, that was for the
identifications.
Yes. Drug sensitivity was by Vitek.
How drug resistance genes are transferred to other cultures?
That is by gene transfer mechanism. To other cultures, in the sense, in the hospital, They can actually, when they are close by different methods of gene transfer, like transformation, conjugation, all that.
Okay. So why coagulase is very important?
Coagulase is an important enzyme in gram-positive, that is Staphylococcus aureus. It actually has a fortress action and a shielding action, which prevents the antibiotic.
So what is the chemistry in your quantitation of biofilm microtiter method?
Chemistry in the sense it forms the biofilm. You
But
allow
how
it
you determine quantity?
spectrophotometer.
So what is the chemistry for that?
OD, absorbance.
No, OD of what?
OD of you allow this it to form, and you take the OD of the solution. What is there? Microtiter.
Thank you.
So in this, you had this ICAD A and D. You said
Yes,
that, right,
yes,
for the biofilm.
Yes.
So how many of them you found? Both of them, because both are required for the biofilm formation.
Yes.
So
we found in 100. We found in 53.8%.
both of them.
Yes.
Otherwise, either one of them is present. Or
One
something.
was seen only in I think.
But it won't form. It won't form biofilm, right?
No, no.
It won't.
No.
Okay. So and this you have checked the. You said that you found the methicillin resistant genes.
Yeah.
In some of
Not
those.
the genes. It was resistant.
Oh, it was resistant.
Yes.
Sorry, methicillin resistance you found in
Yes.
some of those isolates.
Yeah.
In majority of the isolates or in some?
No. Again, it was out of the 63. We found in 53.8%.
And has anybody tried to look at these genes which are known to be methicillin resistant ones in some other, commensals which are there in the same samples? Has anybody tried to look at that?
No.
Because it will be interesting to see, because these genes do get transferred by horizontal gene transfer.
Yeah.
So it will be interesting to see which are those commensals or any other pathogens which are getting these resistant genes.
Yeah, exactly. Yes.
Because
I
that's
think-
really would
yes,
be
yes,
of interest,
correct,
in
correct,
the
yes,
hospital setting.
Yes, ma'am.
Nobody has looked into that.
So far in aureus, they have it has been studied, but not in.
Thank you. Next presentation by Jayalakshmi K. R., Department of Microbiology, Davangere University.
Yes. Good afternoon. My topic is isolation, antibiotic susceptibility and biofilm formation of ESBL producing organism isolated from the clinical specimens. The extended spectrum beta-lactamases are the enzymes that resistance to the most of the third and fourth generation cephalosporins, which includes the important pathogens such as E. coli, Klebsiella pneumonia, Acinetobacter baumannii, Pseudomonas aeruginosa and Proteus mirabilis. Materials and methods. Isolation of ESBL producing organism by different media, MiCON agar, EMB, CLED, blood agar and chocolate agar. Then the phenotypic screening of ESBL producing organism by chrome agar ESBL, BLSC agar, disc diffusion, double disc diffusion, combination test and each strip test. Then the biofilm formation is by qualitative method, by plate method and tube method. Isolation of different pathogenic bacteria through MiCON agar, EMB, CLED and sheep blood agar, then chocolate agar and the leads Acinetobacter agar. Then this is the performance of chromogenic and non-chromogenic agar, MiCON with cephaloxime antibiotic. Then the BLSC agar, it is a biplate technique using Drygalski agar and MiCON agar with cephalosporin antibiotics. Then that is the chrome ESBL agar. By using chrome ESBL agar, we have identified the organism according to the color formation, chromogenic mixture and chromogenic agar combination. This is the table, phenotypic detection of ESBL producing organism by chromogenic and non-chromogenic agar. Then detection, phenotypic screening of ESPL producing organism by disk diffusion and double disk diffusion test. Disk diffusion test according to CLSI M100 guidelines, zone of diameter greater than 17 mm or 22 mm for cephalosporin and penicillin is the positive for ESPL production, and it is the double disk diffusion test. Combination disk diffusion test, greater than or equal to 5 mm increase in the zone of diameter for the cephalosporin and clavinic acid combination disc shows the positive result. Then E-test, mix plus is the combination of cephalosporin and clavinic acid is more than 8 and the zone is obtained, and mix and RJ5 is showing the negative result for the E-test when there is no zone of inhibition on the other side of the combination disc. This is the biofilm formation. Congruent plate agar method. The black crystalline colonies shows the biofilm producer cell, and the pinkish red colonies is non-biofilm producers. Then tube method. It is the polystyrene tube method using LB broth with glucose, and after the incubation time, safranin stain was done to check the biofilm adherence of the cells. Conclusion, the study emphasized the significant prevalence of ESPL producing bacteria. The ability of these bacteria from the biofilm further complicates treatment. A biofilm formation enhances the bacterial persistence and resistance to the antimicrobials. The detection and the characterization of both ESPL production and the biofilm formation are crucial steps in targeting infectious control strategies and improving patient outcomes. Continued surveillance and the development of inactive approaches to essential to combat these multidimensional threats to ensure better therapeutic interventions. These are my references. Thank you.
Session is open for discussion. Any questions?
The biofilm production you tried with the congruent method, right?
Yes, ma'am.
So how will you quantify that? Will it come like quantity wise proper for all the tubes, whatever you have shown the diagrams?
No, ma'am. Almost 80% of organism shows the film in the thick of the polystyrene tubes. Normally, by using safranin stain, we have identified that one.
Yeah, but when you wash, no, some may go off. So that
By
time.
buffer, first we stain with safranin, then wash with the PBS buffer, then I identify the film.
So whatever is adhering to the tube, you consider it as a biofilm. But what if some amount has gone flushed out, or maybe whatever you gently rinse in that also? So the amount which has the biofilm, if it has during rinsing, if it has gone, that will not be into the account, isn't it?
Only thickly adherence the biofilm producers are positive for this test.
And you say visually you have decided, like
but only qualitative method, ma'am.
Qualitative method. So have you graded them?
No, ma'am.
Only yes or no?
Yes,
biofilm produced or not?
Phenotypic detection for biofilm producer.
But do you think sometimes that pellicle formation is also there? Will it be misunderstood? I mean, misunderstood as a pellicle formation on the culture? Sometimes that can also be misunderstood that it is a biofilm. In that way, how do you distinguish? Or you didn't try for that?
No, ma'am. I didn't try that one, ma'am.
Okay, okay.
So you just identified based on your biofilm production that the ESBL, producing bacteria in the samples, right?
Yes, ma'am.
Now, how would you identify which are those bacteria? Are you planning to do that? And if you are
planning to do Molditov and Biotech.
So you will identify
Fine.
so you plan to do that?
Yes,
I mean,
ma'am.
that how will you do that? I mean, to identify which bacteria are those?
Which up to the species level, I would identify it by Molditov method.
That's what you're planning to do.
What is biofilm and why it is produced?
It is the community of the microorganism that adherence to the matrix formed by the microorganism to the.
What is the component of biofilm?
I don't know.
How is biofilm formed? Basically, that's what he's asking. How is actually biofilm formed?
A number of more colonies, microbial biome confirm and to form matrix adhesion to the organism.
How is the matrix formed? That's what the question is.
Yeah.
More number of organisms and forms.
Or do they secrete something?
Yes, sir.
What
How
that?
do they attach themselves to form the matrix? How do they form the matrix? They come together, you said, right? But not all of the patient samples, you saw that they're forming biofilms, right? So they're not producing, they're not forming those matrices. How are these matrices? How do they adhere to each other? All these bacteria, whatever, now you're going to find out which bacteria, yes, right? How do those bacteria, particular bacteria, say whatever, Staphylococcus, whatever, how do they come together to form a film, biofilm? Right, that's what the question is.
Now that would have been very interesting thing, right? Which you will be curious. You have done so many days you spent with them. You should have known that, right?
Yes.
Yeah.
Why certain bacteria are forming those biofilm and not others? Right? There are so many bacteria there, right, in the sample. But some of them you have identified. Some of them you are saying that there are so many numbered bacteria which form the biofilms, but not others. Why are others not forming those matrices? How are the matrices formed? How do they come together?
Okay, why do you study biofilm formation?
To check whether the organisms are resistant to the antibiotics. So,
How it is related? Okay, thank you.
Thank you.
Try to find out the answer.
Thank you. So next presentation is by Minu Eldu, Hassan Institute of Medical Sciences, Hassan.
I have to use this one. It's not working, huh? Okay. This is for. This is my. Yeah. I'll just. Good afternoon, everyone. My study is prevalence of AMC beta-lactamases among gram-negative bacterial isolates in a tertiary care hospital and their susceptibility to carbapenems. So AMC beta-lactamases are cephalosporinases encoded on the chromosomes of many Enterobacteriaceae as well as few other organisms. They belong to class C enzymes under Ambler classification scheme. The mechanism of AMC beta-lactamase resistance can be divided into three categories. It can be chromosomal as well as plasmid mediated. In chromosomal, it can be inducible resistance via chromosomally encoded AMC genes, or it can be non-inducible, which is because of mutation of the promoter or attenuator genes. And AMC beta-lactamases are clinically significant because they confer resistance to penicillin, cephalosporin, oxyimmunocephalosporins like ceftazidime, cefamycins like cefoxitin and monobactams, and their activity is not affected by beta-lactamase inhibitor like clavulanic acid. And the need for the study is there is a rapid emergence of antibiotic resistance among hospital pathogens, making it difficult to treat the infectious disease. And beta-lactams are commonly used for empirical therapy. So production of beta-lactamases is one of the strategies adopted by bacteria to develop resistance to beta-lactam class of antibiotics. And pseudo-susceptibility seen in AMC producers can lead to therapeutic failures. And plasmid mediation of AMC genes carries the threat of spread to other organisms within the hospital or the geographic region. So this study was done to estimate the prevalence of AMC beta-lactamases among gram-negative bacterial isolates and to determine their susceptibility to imipenem, meropenem, cefepime and piperacillin tazobactam. So the isolates were identified based on microscopy, colony morphology and biochemical reactions according to standard guidelines, and AST was performed by Kirby-Boyer's distribution method and was interpreted based on CLSI guidelines. So screening test was done using cefoxitin disc diffusion method. We have used 30 microgram cefoxitin disc and a zone less than 18 millimeters was taken as positive. So those isolates were subjected to phenotypic conformation test like disc approximation test. In this we were looking for inducible AMC production. So we have used cefoxitin disc and cefotaxime disc. They were kept at a 20 millimeters distance and any indentation or flattening of the zone was taken as positive for inducible AMC production. AMC disc test was done to identify plasmid mediated AMC production. So for that we have used 0.5 McFarland ATCC E. coli 25922 and a sterile disc containing the test isolate. So indentation of the zone again was taken positive for plasmid mediation AMC production, and cefoxitin cloxacillin double disc synergy test is based on the principle cloxacillin can inhibit the AMC. So difference in zone of 4 millimeters was taken positive. So here we had used cefoxitin and cefoxitin cloxacillin disc. So result, we have taken 270 isolates. In that 154, that is 57% were cefoxitin resistant, that is screening test positive, and 116 were cefoxitin sensitive. So among the resistant isolates, 84 were AMC producers by confirmatory methods, that is about 31.11%. So this is similar to the study findings by Anand Manoharan in 2017, where only 36.5% of cefoxitin resistant isolates were confirmed AMC producers by phenotypic methods. Similar prevalence was also found in other studies, and 70 of the screening test positive isolates were not AMC producers by confirmatory methods. The possible explanation is there can be other mechanism of resistance which could not be detected in the phenotypic methods. And again, cefoxitin resistant in non-AMC producers can be because of other mechanism like lack of permeation of porins. And 82% of the positive samples were from inpatients. This could be because most of the samples were from inpatients, as well as there can be nosocomial transmission of plasmid mediated AMC genes, which indicates the need for strict adherence to infection control practices in hospital. Maximum number of positive isolates were obtained from pus samples, that is 50%, and disc approximation test, cefoxitin cloxacillin double disc synergy test, AMC disc test was positive in 23, 64 and 55 isolates respectively. So again, plasmid mediated transmission was common. So this should be confirmed by molecular studies. AMC production was predominant in Acinetobacter species, that is 46.7%, followed by E. coli, Citrobacter frundi and Proteus. So most of the isolates were sensitive to carbapenems except 6 which were resistant and 3 which showed intermediate susceptibility. 15 isolates were resistant to cefepime and 6 were resistant to piperacillin tazobactam. So they still can be considered as therapeutic agents after routine AST testing. So these are the distribution of AMC percentage sample wise as well as isolate wise. So in conclusion, the level of expression of AMC genes are not uniform among gram-negative bacteria. Variation can occur with respect to geographical location, infection control practices and various other factors. So CLSI does not recommend routine testing for AMC production, but epidemiological data, if available, will be helpful in formulating antibiotic policy as well as for patient management. These are my references. Thank you.
Session is open for discussion. Any questions?
Did you examine the Enterobacter species and Serratia for the study?
All gram-negative isolates like pathogen we have included in the studies. And
Enterobacter and Serratia are intrinsically resistant to cefoxitin, so there is no zone of inhibition around the Enterobacter and Serratia is seen. Then how you interpret the test result as AMC positive?
So they are known for inducible AMC production. So we have checked for plasmid inducible AMC production and cefoxitin cloxacillin double disc synergy test.
But the cefoxitin will be resistant, right? As per the CLSI guidelines and the clinical experience, there is no zone of inhibition around the cefoxitin disc.
Yeah.
So how you interpret it as an AMC producer?
So any zone less than 18 millimeters was taken as positive for AMC production in our study.
But in your study, the Enterobacter species in Serratia is found to be as positive for AMC.
Serratia was screening test positive, but confirmatory it was not positive.
How you, so can you please check your slide, previous slide? After that, organism and percentage of the Enterobacter species, 6 isolates is found to be as positive for AMC, right?
Yes.
And Serratia is non-AMC producer. How you confirm the 6 isolates of Enterobacter species as AMC producer?
Based on the 3 phenotypic methods, confirmatory methods.
Okay, right.
But you have not yet confirmed the species level, right? Only genus level.
Yeah, no, sir.
So how will you go about it? Then whether this Enterobacter, whatever he is telling, 6 isolates, their status will change?
Yeah, it can change.
It can change. Okay. So how some bacteria gets antibiotic resistance, some do not get?
And there are 4 mechanisms of antibiotic resistance. One method is by production of enzymes which can degrade the antibiotic, and other mechanism they can alter the binding sites, and efflux mechanism and porin channels.
Do antibiotics are active against fungi? Do antibiotics are active against fungi?
Yes.
Yes.
They act
fungus.
on fungi.
Yeah. Do they act on fungi?
Yes, sir.
So are you calling the compounds which act only on fungi also as antibiotics? Thank you.
Thank you. So next presentation is by Ifra Masoom from the University of Transdisciplinary Health Science and Technology.
Good afternoon, everybody. My name is Ifra Masoom, currently pursuing my MSc in Biological Sciences at TDU, exploring the microbial diversity of Vilwadi Gulika and its potential health benefits. So Vilwadi Gulika is an Ayurvedic formulation derived from the Ayurveda literature Hastanga Hridaya, Hastanga Samagra and Sahasra Yogam. Bilvasya Moolam Surasya Pushpam, Phalam Karanjasya Nata Survam means eagle marmellus, Ossimum tino florum, Pongamya pinnata and Valerina jata mansi. Phalatrikam Vyosha Nishadvayam, Bhasta Motrenam Sukshama Pistam means Triphala Trikatu and other plant ingredients. There are 13 plant ingredients. All together they are grinded and churned for 21 days in the goat's urine. Bhujanga, Luta, Undrora, Vishchika, Adi, Virushchika, Garva, Jwara, Sacha means the snake bite, spider bite, rodent bite, scorpion bite, all these together. If a person is bitten by these insects or reptiles, this particular formulation works on it when applied externally. Arathana Naran Bhuta Vidirshika Thamacha Swasti Karoti Anjana means it also has been from the literature of Ayurveda told that it is effective in psychological disorders, and it can be taken it internally, externally, and also applied for oil. So it acts as a wonder drug, not only on one particular symptoms. So these are the ingredients of this formulation. So from a literature review of Vilwadi Gulika, it says that each ingredient has a specific importance. For example, Bilva, Malaki and Trikatu can act on the symptom pain. Similarly, all other ingredients play a major role in each symptoms given here. So my main objective is to analyze the microbiome composition of Vilwadi Gulika formulation and understand its role in enhancing the health benefits. So I took two formulation 1 and formulation 2. Formulation 1 is made by a traditional practitioner at our university, and formulation 2 is a commercial tablet. Both the samples were further processed. Sample was prepared by grinding them, and metagenomic DNA extraction was done using QIAGEN kit. Then PCR amplification was done using 16S rRNA gene. Nanopore library was prepared, and further the data was sequenced and gone for the analysis. So this is the result: gel image of Vilwadi Gulika amplified at 1500 base pair for both the formulation 1 and 2. And this is the result. It shows that formulation 1 has 60% of Firmicutes phyla, and formulation 2 also has the Firmicutes, but with 30%. So you can see the differences in percentage. Both were having high number of Firmicutes load, but the percentage was different. So this is a Stardust graph which indicates the inner circle is the phyla. As you move outwards, it indicates the genus and species. So again here Firmicutes and Proteobacteria is showing that in the formulation 1, Firmicutes is more in percentage and Proteobacteria is less. And in the formulation 2, when you see Proteobacteria is more in abundance when compared to Firmicutes. So in-house, the formulation 1, higher Firmicutes, 47.72% of Firmicutes was found, and Proteobacteria was 37.09%. In formulation 2, that's the commercial Vilwadi Gulika tablet, it was 42% of Proteobacteria, and Actinobacteria was 12.98. So I found out beneficial microbes here. So presence of probiotics like Streptococcus thermophilus, Panibacillus and Gluconolysis and Bacillus cereus group. These microbes are linked to gut health benefits and improves digestion and other immune modulation. So from Rios et al. 2017, through a thorough literature review, potential protection against gastrointestinal disorders such as gastric ulcers and diarrhea. Microbial species like Bacillus and Panbacillus helps to balance the gut of the microbiota. So the overall outlook is that Vilwadi Gulika may contribute to enhance gut health and digestive function. So this is my research team at TDU, and I would like to acknowledge TDU for this opportunity to pursue my masters, and I would like to thank RIST for funding my project, and extended heartfelt to my guide Dr. Pavitra. Sincerely thank to Mr. Ravi Kumar, and special thank to Subramanya Kumar for invaluable guidance, and heartfelt appreciation to Dr. Uday for providing this traditionally prepared Vilwadi Gulika, and my lab teammates Ms. Rajeshwari, Sunana and Ms. Valakshmi, and thank you to MiCON Yenepoya to give me this opportunity to present here. Thank you.
Thank you. Session is open for discussion. Any questions?
This formulation study is very good to know. Now you said that in the formulation you found the microbial composition in that percentage, what you have shown.
Yeah.
So do you mean the effectiveness of the drug is because of the chemical components of the whatever Vilwadi Gulika you have made, or the microbial? The credit goes to whom?
Actually, credit goes to both of them equally, 50-50, because since it has all the 13 herbs with the goat's urine, so we can't exactly conclude that it is only because of the ingredients or only because of the microbes. So it goes like 50-50 both together.
No, we can't say so, because all the people who take the medicine, they think that it is the power of the chemistry present in that tablet that is effective, right? But we being a microbiologist, we have tried to know that what microbes are there in that and which may be contributing to the positive effect or healing the effect. So
maybe it could be like if we applied externally, there might be that whatever microbes are present, and when we take internally also through our gut, we have also have microbes at gut. Maybe they have an interaction, and might be it could be, because I haven't gone completely into the interaction and pathway analysis. So
fine, no issues. We being microbiologists, we are happy that microbes are playing a well role there. Thank you.
Thank you.
So just a question. So you have your formulation and the tablet for whatever is there in the tablet, and you just check what. So did you give them to some
no, ma'am.
What is there?
No, clinical aspect we haven't done.
So this is what is there in the tablet, or your formulation is what you are looking at, right?
Yes, the formulation I haven't prepared, like the Ayurveda. Yes.
Got it. That is what you have, right?
Yeah.
And at the end you are saying that it helps the firmicutes, bacteroids, all those importance what you have shown in the graph. First thing is what I was going to ask that. So first is that in these two one formulation I did you on the commercial one. First thing is you have to see what ingredients are there.
Yeah.
What metabolites are there?
I am working on the metabolites. That is one of my objective. That's not yet completed. Ingredients are 13 ingredients, 13 plant ingredients.
Secondly,
Yeah.
so you're talking about the microbiome in the tablet itself. That does not work. What happens is whenever you take a medicine or your formulation, so it goes into all these gut microbes and it is useful, which bacteria is good for gut health and all that comes into play. When you take those composition or formulation, it actually increases the good bacteria or improves the gut health and so on, right?
Yeah, right.
So the way, you should have looked at is that you should have checked out what are the composition of these two. Are they different?
No, the composition are same.
Composition are same.
Yeah.
Checked out using, for example, mass spec and check. They will not. There will be differences. It's not same. It's supposed to be the same ingredients. What is written, right?
Yes,
What
right.
has been given in Charaka.
Right.
You have used those. Both of them, but there are differences.
Yeah.
Looking into the microbiome, I didn't understand why did you look into the microbiome in the composition itself, and how are you linking to the gut health and all. So how do you link that?
Actually, why I first proceeded with this, because from the literature there is nothing being given out of the microbiome, because we are randomly taking it, but there is no as such microbiome study of this particular tablet.
No. So when you take something, nobody checks out the microbiome. So when you take microorganism, right, you are saying this tablet has lots of microorganism.
Yeah.
How is it go to the GI tract?
That I am doing with my objective 1, which is not yet completed, so I didn't present. I'm doing a network pharmacology where I'm analyzing the pathway of these metabolites and the gene and looking how if this objective 2 is connected to objective 1 with that aspect.
So ideally what you should do is that you should look into the metabolites which are there to start with,
Yes,
the original. Then you do it in animal studies or whatever and try to see what is that in the gut of the animal, and then you try to see what metabolites you have provided and which are the whether it is bacteroids, firmicutes, proteobacter, whatever, right? And then you make an inference saying that it is improving. Secondly, you are also saying this combination of these 3 or 4 herbs together, it improves, that reduces pain, all that you said, right?
Yeah.
You have to find out what is the mechanism.
Yeah.
Which are the bacteria which are doing that? Is there something which can be studied in that? So I think you should look into from that.
Yes, ma'am.
Right?
Yeah.
So do you have any similarity between the microbial profile what you got here with the cow urine profile?
We haven't checked with the cow urine's profile.
But you could have compared, right? Because that is the only source or endophytic source you have.
Further, we are waiting for funding, sir, so we can go ahead.
No, this
with
doesn't need funding. You have to just do literature comparison.
Yes, sir.
That is, if you are curious, you would have done. Then why are you getting only these 2 or 3 classes of bacteria? Why you are not getting other things in this mixture?
They are mostly abundant one I have presented. I have got other bacteria too, which are less.
What is the cutoff for you calling something as abundant?
Abundant, out of 100% we have seen it.
Now, how much percentage should qualify to tell abundant?
More than 50, sir.
But they are not more than 50. Some of them are 30%.
But within that formulation, that percentage was the highest one.
So, for what purpose you are using this tablet?
Various purpose it is used. Basically, for the snake bites, rodent bites, and insect bites, it is mainly acting because it has certain herbs and the means it's acting as a toxins like.
So, but you already have in the literature some data on how it acts for the snake bite victim.
Sir, there is no.
You may have already data available how this Bilwadi gutica is effective for snake bite.
There is no thorough literature, sir, but with respect to Ayurveda, it only shows the, as ma'am said, Clinical research is not there. There is only
No,
systemic
clinical
review.
research may not be there, but some data will be there. Somebody might have done something. How much literature you have searched for this data?
Around 21 I have got, sir. 21 papers, which has all are thorough systemic literature reviews. It has no mechanism. I mean, none of the paper has shown the mechanism of this particular drug.
Okay, thank you.
Thank you, sir.
These medicines have to be taken orally, or is it applied on the?
It is all, you can apply externally and orally also. Thank you, ma'am. Thank you, sir.
You studied the effect, combined effect, isn't it?
Sir?
It's a combined effect of 3-4 herbs together.
13 herbs.
13 herbs. Is there any information on this single drug?
Single herbs information are there, sir, but all together there is no information that how effective it is working. Each plant-based literature is there, like how eagle marmelis is working, that literature is there, but not all together.
You given the bacterial name, it should be in italics.
Yes, sir.
Okay.
Thank you, sir.
Thank you. So we have come to end of first oral presentation session. So I thank for Dr. Sharmila Mande. And
Thank you for giving us the opportunity to chair the session.
Sure, madam. And also to Dr. Keshav Prasad for chairing this session and making fruitful discussions. Thank you both. I will hand over to Sonam.
Thank you, sir. Thank you to all the presenters and thank you for attending this session. I request Dr. Rekha Pedi, Chairperson of MiCON 2025, to present a token of gratitude to Dr. Sharmila Mande, the chair for the session. Please. To Dr. T.S. Keshav Prasad for being the co-chair of the session. And to Dr. Rajesh P. Shastri, moderating this oral presentation session. Thank you, ma'am. It's now time for the poster presentations, followed by lunch. And all the presenters are requested to be ready with their respective posters. And we all will gather here post-lunch at sharp 2 o'clock. Dear participants, kindly refrain from carrying food to the poster presentation area during the poster evaluation.
All.
I request all the participants to be seated inside. Participants, please be seated. Welcome back to the sessions. We request Dr. Asif Hamid, Organizing Committee Member, MiCON 2025, to introduce our next speaker.
Thank you, Sanjana. Good afternoon, ladies and gentlemen. It is with great honor that I present Dr. Tanaji G. Kudre to the audience at MiCON 2025. Dr. Tanaji G. Kudre currently holds the position of Principal Scientist at the Department of Meat and Marine Science, CFTRI, Mysore, India. He earned his PhD. He earned his PG degree in biotechnology from Swami Ramananda Tirth Marathawada University in Nanded, India, followed by a PhD in food science and technology, specializing in seafood processing and product development from Prince of Songkhla University in Hatay, Thailand. Subsequently, he completed 1 year postdoctoral fellowship at the same university. With over 11 years of research experience, Dr. Kudre's expertise encompasses seafood chemistry and biochemistry, food quality and safety, the development of value-added product from fish, poultry, and seaweeds, as well as microbiology of meat and meat products, including meat authentication through genomics. He has been recipient of Alltech Young Scientist Award in Thailand and served early career research grant from the Department of Science and Technology, India. Dr. Kudre serves as an expert member for various institutions and government bodies, including the Bureau of Indian Standards, FSSAI, and the Ministry of Environment, Forest and Climate Change in India. He has supervised one CSIR-TWAS postdoctoral fellow from Egypt, 6 PhD candidates, and over 35 graduate and postgraduate students. Throughout his career, he has held 15 research projects focused in food processing and product development, primarily in seafood, meat and poultry processing and safety, funded by both the Indian government and private industry. He has developed and transferred more than 50 technologies to private firms and industries. Dr. Kudre has authored over 50 research articles published in peer-reviewed international journals in the field of food science and technology, Additionally, he has presented over 30 posters and delivered invited talks at national and international conference. He has also served as honorary treasurer for the Association of Food Scientists and Technologists and as convener for the 9th International Food Convention, that's IFCON 2023. With this brief introduction, now I would like to invite Dr. Tanaji to take over the dais. Over to you, sir.
Just check it. No, from here only. Then I'll.
From there, if I walk, it will be work, no?
Yeah, it will work.
Thank you much, sir. You introducing me briefly. Very good afternoon to one and all. I think it is very difficult after once we had food, especially after lunch, to speak as well as to listen also. But I will try to crisp my presentation. First of all, I extend my sincere thanks to organizing committee of MiCON 2025 and management of Yenepoya University who have invited me here and given opportunity to share my some piece of research work. So I am going to present here the harnessing power of probiotic bacteria for the development of meat-based biofunctional food and food product. Actually, I am not the hardcore microbiologist, even the medicine microbiologist. I am just food technologist, but I have my eager was there. So how can that microorganisms will be help in the food system? How can make the food biofunctional for the a different type of like nowadays it's called as personal nutritions. Okay, so with that I will here present. So so in my presentation these are the three parts includes. One is a production of bioactive chicken meat protein hydrolysate using the lab lactic bacteria and its application in complementary baby food formulation. The second part of my presentation would be the production of bioactive food, fish protein hydrolysate using the lab and its application in the cookies formulation. And third part is production of bioactive protein hydrolysate from the meat processing waste proteins using the lab and its application in poultry feed formulation as alternative dietary proteins. So these are the three parts I am going to be cover it. But before that, so what are the drivers or what are the things forced me to work on these three parts? So first when you see the, I think we are very aware, well aware of malnutrition, children malnutrition and present status of India. So we are saying we are increasing our the prosperity, we are good in the all, because in populations also, right, we crossed already China. So in this, when you see the present status of the our malnutrition, children malnutrition specifically, so India is 1/3 malnourished child is present in the India. So still we are not successful in the combating this malnutrition, children malnutritions. So when we see the malnutrition children, the present data as per the Portion Tracker report, the present data is say that the 17% children in India is underweight. Is really pathetic. When you see that we are increasing our properties, okay, richness, all fact, but we are suffering, badly suffering in the malnutrition. So in 34% children are the staten. So this is also is very badly suffering with because of we are not providing the proper nutrition to our children, and 6% children are wasted, and this is surprising that the 56% of children are anemic as till the age of the 5 years. So these are the present data for the children malnutrition of the in India. So that was the because when we thought whatever the baby food products are present in the in our market, Indian market, Can you hear me, sir? So you see that whatever the baby products are present in the market, mostly the mothers, they knows they are based on the plant and cereal, plant-based proteins. Maybe it's pulses protein and cereal proteins. So when you see these all, the Nestle also is selling the Cerelac. I think most of people are familiar with Cerelac. Then lactogens. Nowadays is Abbott also come in this market for the baby food manufacturing or baby food productions, complementary product. So because the main thing in the malnutrition is children is we are barely suffering from the protein energy malnutrition, because whatever the products available as a complementary forms, that is not enough in good quality proteins. Is because mostly, as you know, that the plant proteins are the lacking some nutrients as well as some essential amino acids. But for growth development, okay, growth of the children at their developments, we require the quality proteins. Then and then it can be we can combat the malnutrition, children malnutrition. So we thought that because whatever the products are in the market, that is mostly the plant-based protein, but it is not called as a quality protein because it is doesn't have the all essential amino acids and some minerals also. So then I thought that what is the good source for the to alternate or to replace that plant proteins. So as you know that the meat is a quality proteins, okay, it has the all essential amino acids and rich in the some vitamins and minerals also. And this meat have the high digestibility when compared to the plant protein, because people, they used to say they have outer tag is that, so meat is not good, not in the digestibility, because while you chewing it, it is tough, but once you go into the intestinal tract, it will be more absorbable than the plant proteins. So that is the beauty of this non-veg proteins. So with that, I thought that why not will be replaced, because even consumers also, many parents also, they are accepting that. So cannot, whatever the products are in the market, so we can be complete or maybe partial replacement with the meat protein. So with that, I took these objectives for my research. And in that, it is meat protein is good, okay, it is quality protein, high digestibility, high bioavailability. But I use some application more than that. Why not we can make a biofunctional to this meat, and that will be good help. It will be help in the digestion as well and because this, as all people know that the probiotics here, how it can be help in the making the biofunctional protein or protein hydrolysate. And the second part, that is I used that same way, same manner, same things. So I used that the hydrolysate proteins for the cookies formulations, because you know in the present situation, all children are fond of for the all fabricated food products. They like more these products. Okay, so, but that products is completely lacking the quality proteins. So I thought that I cannot replace. We can replace that type of proteins, okay, by the quality proteins. Then I made some cookies which I used meat fermented proteins, that is hydrolysate. And these cookies can be not only the children, but it can be used for even the elderly people also, because the elderly people, they don't have that good property for the digesting the proteins. Okay, so this can be work as for elderly people or all aged group product. Then third part is, while when processing of the meat, mostly fish or livestock, so there is huge amount of waste is generated. I think you people are aware, when you go any domestic fish market or any slaughterhouse, when crossing it, so a lot of smell is come, right? So even though when you see there is smell, a lot of dumps, okay, so that is nothing but the waste byproduct, we call as byproduct, but the beautiful part of that byproduct, they are rich in the quality proteins, apart from, in addition to the lipids also, and minerals and vitamins. So I thought that because when you use this, nowadays the practice is this, when you take this waste, byproduct waste or slaughter waste or fishery waste, and they simply dumping it to the water, right? Water or somewhere it's landfilling, and that also create the environmental pollution. So this, to make this the use of utilization of this byproduct for the feed formulations. So nowadays the case, you know, we are eating the good in chicken also, right? So chicken, the lot of antibiotics, some cases, many consumers, they said, sir, it is lot of antibiotics used. It is correct, because even government policy is not framed the how much antibiotics has to be used in the for poultry farms or any livestock. Okay, so because there is no frame, so farmers they need their profit only, so they put the more antibiotics, right, and through that cycle from chicken to we are getting in the our body. So morning, madam said that antibiotics, sir, Rao sir using that words, so resistance to antibiotic, right? So that is because of this only. So we thought that why not we use this alternative to that antibiotic, this hydrolysate work as a dietary protein as well as the work as a maybe antibiotics, okay? So anti-natural antibiotics to be the feed system of the poultry. So with this, I think in morning, since morning we are listening this one probiotics and all, I will skip these slides, okay? So I think we are very good in this probiotics have the good benefits, health benefits. Now these are the probiotic enlisted, mostly the undercome as a Lactobacillus bifidobacteria, and some others are the Bacillus phthalis, Enterococcus, Streptococcus, or these are the beneficial bacteria. And I think so you know what is the defined as a probiotics. So my interest is here, why? Because as very well known that the probiotic bacteria is a beneficial bacteria, is good for the health. So apart, even that, when you see the my interest in it is already known, but why you can use that this further because this is one factor is enzymatic. Okay, probiotic bacteria when you go for the any fermentation product, they secrete some particular enzymes, okay, and mostly their enzymes why they secrete, because there are the exotrophic for the amino acid, they can't synthesis the amino acids in there, so they have to be depend upon the environment, they have to secrete some enzymes, and environment protein, that enzymes it will get under degradation and they will absorb this amino acid. So because when you see when you compare the enzymatic hydrolysis, okay, whatever the commercial enzymatic hydrolysis is there, but that is not good enough to get the particular bioactive functions. And even see it is a cost intensive. When the enzymes you see that the cost of the enzyme is more to make the hydrolysate preparation. So apart from that, so these probiotic enzymes, that proteinase, a different type of proteinase they secrete at one place, and that proteinase can be make a functional protein, okay, that is protein hydrolysate. And another is bacteriocins. When you go for the fermentation product, it is very well known that this is a probiotic substances they secrete by the lactobacillus or any probiotics, which are good for the even to enhance the for in case of the food. So I can say it is we can use enhance the stability of the food product by using such natural bacteriocins, or when you see the peptidocins also. So these are my concepts. So we are working on this bacteriocins, because when you go for the fermentation, that two concept will become. So it will be developed. It will be secrete some peptides which act as antimicrobial properties, and some enzymes that can be helped to degrade the intact protein into molecular or small molecular peptides, which will have or endow the bioactive properties. Now this is the first part. I given here is a brief flow chart for my work, how I carried out. So first of all, I use the lactobacillus, whatever the probiotics present available with my friends, colleagues. I am not the microbiologist, but I took from him to want to be check the different type of lactobacillus or probiotics will how they will potential for the production of the antibacterial or antioxidant properties peptides. Okay. So I took the first some 5 3 from the lactobacillus and 3 from the peptidococcus strains, and then I work on the preliminary. So from that I choose the peptidococcus. This peptidococcus pentosis is a good one, and then I use that one. I use for the optimization by using the RSM, the CCRD. That is good tools for the optimize the condition for the any enzyme production or hydrolysis. And then I've gone for the fermenter high scale production. Then after that I go for the whatever the liquids produce or hydrolyzer produce for the spray drying. And then spray drying I used first because I want to be used in the complementary baby foods, because I have to check the first safety of that protein hydrolyzer developed by the fermentation by peptidococcus pentosis. And then I using the rat bioassay. Then after that I formulate the baby foods whatever the present in the market. So I use some formulation and I replace whatever the protein content through the plant with the hydrolysate with the same quantity. And then I developed this formulated the complementary baby foods. And because this is already in the process to be transferred to this technology, maybe in the near future it will be such type of products will come in the market, either this one. And once I formulated, then I have to check the quality of that product's complementary baby foods on the, by using the biorats. And this is the so on. Once apart from that, I have seen that really that what antioxidant peptides is which peptides is there in that hydrolysate. Then I subjected for the purification. Then I find out some particular peptide sequence there. Now this is my preliminary work, as I said, because I screened some probiotics which are good for the producing the antioxidant peptides as well as the peptidoglycans or bacteriocins. So in among these all, it is shown the Peptidococcus pentosis is good for the producing the antioxidant as well as the bacteriocins or peptidoglycans. So then I have used that RSM model to be optimized because I use a different variable to optimize the conditions for the hydrolysis as well as for the getting the antioxidant peptides. So I use this RSM methodology under the CCRD designs, and I got the optimum condition in here. So meat concentration, because I used this is a chicken meat, so chicken meat concentration is 45%. And then dextrose is 2%, and inoculum 10%, fermentation time is 48 hours. This is the optimum conditions to produce the maximum antioxidant peptides as well as the antibacterial substances. So these are the ANOVA analysis that show that this whatever the model I used, it is suitable to optimize this condition. So when it is produced, the optimum condition that it has to be shown a different type of antioxidant activities, DPPS, then ABTS radical scavenging activity, then FRAP activity, iron chelating activities. Because so why I choose the antioxidant activities, you know that the oxygen stress is too much for even children also. So to combat that one or to maybe alleviate that one, so that is I choose the antioxidant peptides that can help to the reduce the oxidative stress in the children. Then when you see the scale-up studies, slightly higher activities are observed. When you go, because the once it is this peptide is synthesized or produced, so it is helpful in the GI tract or not. So that's for GI tract also gastrointestinal digestion I have checked. So and that digestion, so almost all activities is written in the that digestion. Then same things I used to check that optimized condition hydrolysate to be having the antibacterial properties against the several pathogenic strains, right? So I used as a reference as a ampicillin as well as the nuisins. So it has shown that almost is against to the Bacillus cereus as well as the E. coli, then monocytogenes and Enterica, Salmonella, Staphylococcus aureus. Okay, so these are the shown by the zone of inhibition, so that whatever the hydrolysate produce, it having the antibacterial peptides, it's derived from the meat protein, maybe or it is from the probiotics. It's maybe in the form of some this bacteriocins. So then once it is hydrolysate, to be confirmed the hydrolysate is converted intact protein into the small molecular weight is not there. So we have to run the protein pattern, and in this you check, see the this is the intact protein. So intact protein have the different high molecular weight in, but once it is become the hydrolysate is having the small molecular weight peptides. It indicates that the protein, whatever the fermented protein is, intact protein is converted into the small molecular weight peptides. That is it's showing the antioxidant properties. When you see as I said, it is meat is a quality protein, but I should see the balance of the essential and non-essential amino acids. In this, you see the ratio is good, 0.9 is their ratio. So it having the good amount of essential amino acids, and that can be helped for the growth, normal growth of the children. Then it is a FTR analysis that show the presence of some functional group of the proteins. But when you go for the fermentations, many people have said it is maybe the really is a beneficial fermentation or it is a putrefactions. So putrefactions means it will be more this bioamines productions that will be not good for the health, even the children's also. So that content of the biomatter, this is there as per the standards below 5% is good for the health. So that all histamine, tyrosine, cadaverine, and putrescine, it is below 5 mg per 100 grams. So it shows that the whatever the produce the hydrolyzed protein is good for the to use for the in food content. Now, so what I have produced that hydrolyzed having the good proteins, that is around 74.94% is good protein is there. And the most important things, it is a good because it is come from the protein, but apart from that, you know that the even micronutrients deficiency, because malnutrition also it is there. So, and this malnutrition associated with the type of proteins. So, when you see the in that hydrolyzed, even though the total iron content also is good enough. So, because the even children suffering from the anemia. Okay, not only that, but the women also having this type of problems with anemia. So that can say this the cocktail of the even minerals as well as the good quality proteins. That's what the hydrolyzed is there. So that can be provided this minerals as iron content. And because I want to have the use that hydrolyzed for the food formulation. So basically that food hydrolyzed really work in the formulation because I have to check some physiochemical properties or their functional properties, and I have checked their solubility, foaming properties, emulsive properties. This is for the food applications or food formulation. So all this data show that it can be compatible for the food formulations. Now, this is the purification. I said that really that antioxidant, what type of that sequence of the peptides which is for the potential for the antioxidant activity. Then I use that hydrolyzed, then I gone for the purification different steps. I subjected to the ultrafiltration using the 3D CADA, below 3 CADA. So that result show that below 3 CADA molecular weight of that peptides are having the more antioxidant properties. Then from that I subjected that 3 CADA fraction to the gel filtrations for the purification by using the Cephalodex 15, and the peaks I obtained the several peaks, and the 5 peaks is shown the potent antioxidant fractions, and then from that antioxidant fraction and then subjected to RP-HPLC for further purification. Then I got the some peaks, then PEEP 8 is showing the higher antioxidant activity, and just to be curious that these are the purified peptides. Now I want to have the what type of sequence is there in that peptides. Then I subjected to that for the identification by using the Mascot, this Mascot software to under the SysPort database, and it's shown the one peptide which is here, which is that called as having this molecular weight of 857 Dalton. So these are the peptides is from the hydrolyzed is showing the potent activity for the antioxidant properties. Now this is now I come with the hydrolyzed already with having the antioxidant potential and as well as the antibacterial. Then I want to use to be check the safety for the because I want to use for the complementary baby food formulation. Then I use here to be using the different rat models, winning rat model, and that different their feed I replace with this hydrolyzed. In this it is a control diet, whatever the standard diet YAN G93 diet that I used, mostly the casein is a protein source. Then that casein I replaced with the 50% this hydrolyzed, another group, and then I replaced that casein protein as a 75% with this hydrolyzed. And one last group is fifth group, it is a completely I replaced the casein with this hydrolyzed chicken meat protein hydrolyzed. So then I have run these trials, and the most quality indicators for this growth for this hydrolyzed, when you go for to check that this quality is good or not. So mostly these are the called as a PER, FCR and PER and FER. This is a feed efficiency ratio, then feed conversion ratio as well as the protein efficiency ratio. That will give the indication this protein is quality or not. So here in this data you can check this wherever the protein this feed conversion ratio, the lesser the feed conversion ratio, it shows that that type of protein, that whatever the protein is there, it is a quality protein. It means have its bioavailability, good one, and digestibility. So, and PR is increasing order, the which higher the PR, that show that protein efficiency is good, and that protein is a quality protein or protein hydrolysate. Even the FER also, it is the same pattern. So in this, it is show that when you see the group, so replacement of 100% casein with the protein, this chicken protein hydrolysate, to show the higher quality. So it means that doses whatever are given, that is complete replacement can be is work here, and it can be absorbed by the rat model, and we can say it is a quality protein can be observed at the whole total percent. And to check this is just to be their growth indicators, it is quality or not, but and check the hematology, even urine analysis, any toxic effect is there on there, then it is safe when compared to controls, the all references values under the control. Then hematological parameters also, but when you see the here slightly increase in the hemoglobins because presence of the iron in that hydrolysates. So even RBC, this one, so slightly increases there. So it shows that it is good for the even iron content also work in this study. So biochemical parameters, mostly when you see the, because you are giving this new protein for the this rat, and this rat, because that is some markers which show that liver functions is not there, that is called as a HUPT and HUOT and alkaline phosphatase. These markers I tested from the serum, and then this will show that it is a slightly increased this because slightly some damaging the liver, but it is under the control. It as it is when you see the reference values for these markers, it is in the reference values, and but I said it is antioxidant peptides, but in our in vivo systems it's work or not, it will show the antioxidant potential or not. Then I check the some catalase is a marker for antioxidant and SUD, that is mutate. So these also see, when you see the higher concentration of the replacement of the chicken protein, it will increase, it is increasing. These values are 11 to, that is standard this one feed, but when you increase the 50, 70, 50, 100%, the antioxidant potential in form of the SUD and this catalase is increased. It shows that it's indicate that, so that whatever the hydrolysate, that antioxidant potential hydrolysate I given to the rat model, so it in vivo also it is showing the antioxidant potential. So this is a histopathological images, so there is no defect in this, it is with the reference and is standard. So it say that there is no damage in the histopathologic structure. So now, so once I run this, I check the safety of this, or how that quality protein or that chicken hydrolysate work in the quality as a quality protein in the rat model. Then I said that is from that previous study is confirmed that up to 100% you can replace, but there is no even pathological signs. Okay, so it show the good quality. Then I formulated as per the complementary baby foods, there are the standards, FSCI standards. Okay, so I have to use the 15% is maximum proteins, and then I have made the 15% and used this is a general formulation for the complementary baby food preparation. And then I replaced chicken protein. I use the chicken protein, it is common for 15% as per the standard. And then different flavors I added. So from these different flavors I give beetroots, because nowadays complementary baby foods are coming with the different flavors, because children cannot go directly with this one. So we have to give some flavors so they can be eat properly. So using these different flavors, beetroot, carrot, it is a natural origin. Just I added the beetroots powder, then carrot powder, date powder and pumpkin powder. So then this is the ready to use formulations of these complementary baby foods. And when you go for the real serving to children's, it has to be made in the porous form, and it is working in porous form also, it's very nicely. So this is from the porous form. But among these 4 formulation, because when I am saying that it is good, all flavors are good, but children has to be our even human being system, that because we are fond of our tongue, our market of this product, it will be different on the tongues. Okay, tongue, if it is say it is good, then that market will become further this product. So then I have gone with the sensory panels and sensory evaluation I have done. But among these 4 formulation, the pumpkin formulation showed rated the higher score. So then I took this pumpkin formulation and I checked again their composition, chemical composition, which having you check the protein concentration is still, it is good when you check the commercial. This is whatever the commercial complementary baby foods are there, mostly cerelac as well as the lactogens. And you see the protein content is also slightly increased in this. And as per the FSSI, that is given that used as a reference. And when we can check that because it is quality proteins having the biofunctional properties like antioxidant activity, so it shows that that can be good. So I use that pumpkin complementary baby foods, which is prepared by the panelist. And I used once I formulate, because other ingredient also using in that formulation, so it will be work properly in the in vivo conditions. That again I have gone with the rat model studies using the weaning rats. And I provided here very interesting result. It's come out of this. I provided as a standard as a casein proteins. Then group commercially whatever the available proteins in the market, complementary baby food, cerelac as well. I used that one. How it is work in the rat model compared to my formulations. And I used the completely formulations without any changing in the protein as like complete chicken protein hydrolysate what I formulated. And another group, 5th group, I used because partial replacement of the chicken protein with the soya protein as well as the milk protein. So because when you use all this protein, complete protein, some different flavors was there. So I thought that we will replace little bit with the plant protein as well. And then I used that one. When you see this group, the protein quality indices, indicators, you see the group which having the 70% chicken protein hydrolysate and 15% the milk protein and the 15% soya protein has shown a good PR, FCR as well as the PR. It shows that instead of this group, the containing those the food formulation having the soya protein and milk protein is given the good one. Yeah. So this is a you see this is a FCR with the 5th group having the lower FCR. It indicates that it is this protein is very palatable for the rats, as well as is given the efficiency, the conversion efficiency to mass. Okay, that is FCR. So it's shown that this group is good, but when compared to the this complete the formulation by the chicken meat protein hydrolysate. So it is this has, I don't know what is the problem with this, because high maybe it is a toxic, but in its previous study it is said it is not toxic, but in the formulation this food formulation, complementary baby food formulation is showing the little toxic and is not giving the growth for this for rat. So this is their body weight gains throughout the study. It is given. So same way it is observed that the group which 5th group is containing the 70% chicken protein and 15% milk as well as the soya protein, that having the good weight gain. And when you see the hematological analysis, urine analysis, it is in the reference. And when you see the hematological property, again it is showing that the 5th group having the HbC is more. Hemoglobin is enhanced in this 5th group as well as the 4th group. So this because mostly the presence of the iron content in the hydrolysate. So, but when I check the adipose tissue, I sacrifice these rats and check the adipose tissue. When this commercial formulation, whatever the present in the market, like cerelac or the lactogen or Abbott's companies, that commercial present existed products which developed a lot of fats tissue, fatty tissues, this adipose tissue. And when you see this complete 100% chicken meat protein, there is no fats, adipose tissue. But when this 5th group having the little fat, but not compared to this one, Okay, so that I don't know. This is the why it is adipose tissue as enhanced in this commercial what available baby food product. But this our product is showing that there is no adipose tissue. It's completely converting that absorption into the muscle mass. So from this work, it can be conclude or summary is that the Pediococcus pentosis showed the maximum antioxidant and antibacterial against strain for 48 hours of fermentation. Then optimum hydrolysis condition, that is a 45% meat, 2% dextrose, 10% inoculum, and 40 hours of the fermentation. So this is the optimum condition to be produce the antioxidant and antibacterial protein hydrolyzed from the chicken meat. And this is one, as I said, one peptides that identified from that hydrolyzed. And this can be, so conclude is that through this all study, we can replace the existing protein, plant protein with the chicken meat hydrolyzed up to the some level, So that can be make the complementary by incorporating this chicken meat hydrolyzed. Now another part, it is as I said, I made the same way use the that Pediococcus pentosis for the meat for fish, because many people fond of coastal communities, they like the fish. So why not go for the fish? So I selected some freshwater fish here. It is called as a ready bell poco fish, and I used again because here I have not gone for the any screening up, because I thought that you can use as a this Pediococcus pentosis for the further optimization for the using the fish meat. Then same way I optimized by using the RSM. Then I scaled up to the 10 liters. Then made a powder, and that some different concentration of this hydrolyzed, which having the antioxidant potential as well as the antibacterial potential. So that different concentration I incorporate in the cookies formulation by replacing the beet powder. So same way again I gone for some purification, which type of peptides present in the fish protein hydrolysis to show the antioxidant potential. So this is the same models I used, and the optimum condition for this fish protein hydrolysis is 30% meat, but in case of chicken it was the 45%, and the sucrose is 2%, inoculum size is 10%, and fermentation time is 48. So this is the optimum condition to be produce the antioxidant potential fish protein hydrolysis. So same way, this is again I have checked the gastrointestinal digestion. So it has the almost showing is slightly increased, but there is no the activities mostly is retained here. So same way again antibacterial properties I have checked. It is showing the many against the many pathogenic bacteria. Same likewise again it has to be show that it is intact protein is converted into the small molecular protein. It's not there. So I checked with hydrolyzed by the protein pattern using the SDS page, and it's showing that the intact protein is converted into the small molecular weight peptides. The amino acids, different essential, non-essential amino acid composition of that hydrolyzed also I have checked. And the FTR analysis, again because I said that bioamines is difficult to be make it in this. So, but because it should not be a more putrefied product, because putrefied means it is if it is higher range of the bioamines present, it means nothing but the putrefied product. It is not good for the health. So that is also the content of the bioamines also it is in the range below the range where it can say that is that hydrolyzed is safe for the consumption. So this is the same things in this almost 71% protein obtained in that hydrolyzed, and iron content is quite enough also, and all the functional properties show that this can be used as a in the food as a ingredient in the food formulations. So same way the purification peptides also have made. I gone with the different purification steps. Through the ultrafiltration, then gel filtration, and the RPS PLC, and finally I gone with the to check the LCMS the sequence of the these peptides. So in this I observed obtained the two peptides where it having the antipotential bioactivity, antioxidant bioactivity. So this is the molecular weight, and these peptides are the novel. It is not anywhere is published, or that's why it is called as a novel peptide. It is a new peptides which showing the antioxidant potential. So now that hydrolyzed fish protein hydrolyzed, then I used for the cookies formulation. And I replace when you see this wheat flour and fish protein hydrolysate, I replace the different concentration, 4%, 8% and 12%, and I formulate these cookies. And this formulation, you can see this different proximate composition of this. As they increase the fish protein hydrolysate, the protein quantity also is increased in the cookies. It shows that whatever the cookies is there available in the market, that is lacking the protein. So, but these cookies, it will come containing the high quality protein. So that can be good for your children growth as well as the all aged people. And for this, which concentration is good for eating. So that's we have to do the sensory evaluations. Then sensory evaluation says that the panel said that up to 8% can be incorporate this fish protein hydrolysate into the cookies through this sensory evaluation. So it's this give the summary that so we can produce the protein hydrolysate with having the antioxidant activity, antibacterial property from the fish protein using the Pediococcus pentosis. And this is optimum condition, required the 30% meat, 2% sucrose, 10% inoculum and 48 hours of fermentation times. And these are the two novel peptides which present in that protein, fish protein hydrolysate. And finally it said that when you go for the formulation, food formulation as a cookies form, the 8% hydrolysate can be used for the formulation of the protein rich cookies. Now this is the food formulation. Now come to the third part, this is olerization of the fisheries waste as well as the slaughterhouse waste for the animal feed formulation. So when you go for because as I said, almost 50-60%, when one animal you are slaughtering, that 50% of that animal weight is used as like a waste byproducts. So when you go for the 100 kg of suppose one animal livestock, then 50 kg of you will getting the waste in the form as a byproducts. Okay, so and that has to be make some utilization, olerize, because it has the resource of the good proteins as well as the oil. So then I gone with took this different slaughterhouse waste as well as the fishery waste. Okay, then I extract the protein by the weight reduction method, and this is come as a solid waste. And then I extract the protein, and then when you go for because while processing this animal, even fishery fish, there is a huge amount of wastewater is generated in the form of, but this water is rich in the protein, good quality protein. But when you throw in the even oceans or some water bodies, it creates the pollution. Then I extracted that by the alkali and isolated pH precipitation, and then I mixed this both as considered as a waste protein from the animal slaughters as well as the fishery, and combine. I used that weight protein for again further the optimization by using the probiotic bacteria. That is I used the probiotic bacteria Lactoplantarum, then subspecies Plantarum and Pediococcus loli. So these strains, among these three strains, the Pediococcus loli have shown the good hydrolysis condition for the antioxidant as well as the antibacterial properties. So that's using that one again I gone for the optimization condition for like how much the meat is required, how much time fermentation time is required, how much the size of the inoculum. So this one is I optimize that condition, and then once it is optimized, I have to see the safety of that hydrolysate which are carried from the waste of animal as well as the fishery waste. So I gone for the first rat model to see that quality of protein as well as the safety. Then after that, once say it is a safety for the rat model, then I use that formulate the feed for the poultry, and I taken the trials for the feed formulation for the poultry. So these are the proteins, different type of protein present in the wastewater from the bloods as well as the muscle proteins are there. And surprisingly, you see that this amino acid composition of this is that the essential amino acid is more than the non-essential amino acid. It is very good protein from the waste biomass. So this is the optimization condition where we got it the optimum consider. This is meat protein has to be 18.4%. Then dextrose concentration 1.42, and fermentation time is 56.18. So with this optimum condition, whatever the antioxidant activity is coming in that, so I check the antibacterial properties against the monocytogen listeria, then Staphylococcus aureus, then E. coli, and Salmonella enteritidis. This is a prevalence bacteria present in the poultry. So because their health, so this can be work against this bacteria. I wanted to be checked, and this is showing the good zone of inhibition. It says that it is antibacterial against this pathogenic bacteria. Again, same the amino acid composition of hydrolysate is because during the hydrolysis, there should not be changes in the amino acid composition. To check it is retained the same amino acid composition, and this is a hydrolysis which converted intact protein into the small molecular weight peptides. And then once I have this hydrolysate, then I plan for the design for the rat to check the safety of this hydrolysate, because it's derived from the waste. It should not have the toxic, because heavy metals and all this, when you take the this waste, it will be more heavy metals. So I wanted to see this, and then again same designs I used their casein protein replacement with this hydrolysate. So first is a 50% replacement this casein with the hydrolysate, then 75 and full replacement. So these are the different group I used, and it is showing that when you go the study, this is body weight gain by this rats, and the 50% replacement by this waste protein hydrolysate, giving the good growth of this rat, which showing by the PR. This is a C2.5. It is giving the good quality, good growth performance. It says that it is a quality protein, and there is no mortality, showing that it is a safe. And then you send this, does that hydrolysate is affect on the some vital organs or not? That all organs you see it is almost it is in their growth is good, normal growth. And then histopathology of this liver, you see that there is a good with compared to the standard. So then this is from the rat model study. It is ascertained that this protein can be used for the poultry. It is safe up to the 50%. But again, I started the different concentration can it's work for the poultry. So I used a different replacement, because this is a chicks. I use broiler chicks. This is a broiler chick, and different concentration of this protein from 25, 50, then 75 and 100% complete replacement of the whatever the standard protein using in the poultry feed. So mostly poultry feed, you know that they are using the soya protein is a protein source, dietary protein source. So but this soya protein is very difficult to get all times for the farmers, because it has the dynamic price. So this month it is come lower price, but immediately the second month it will be high. But for farmer it is very difficult to replace this protein, and it is very expensive also. So I thought that can be replacement as a dietary protein. So I use this hydrolysate for them with having the antioxidant properties. This is the just general formulation for the feed, for broiler feeds. When you come to the result, that I said it's the body weight gain that indicate that protein is quality or not. So when you check this body, when still in this poultry also 50% replacement shown the high quality, high body weight gain, and their growth performance. So in this, just you see the chicks also here 50% having the high weight after 48 days. So it shows that the 50% can be good for the replacement as a soya protein replacement by this protein hydrolysate. And then once because it is growth is good, but this is once you have provided a lot of chicken is growth good coming, but the market will say once it is consumed it is good or not. Then I checked their addressed composition of the chicken. So we see the control as well as the 50% is showing the almost same, better than this 50% is slightly better than the standard diet. So then I gone with because meat analysis, that sensory analysis of that does, because I am giving the waste protein, some smell should not come from the meat. Okay, so that the properties of that chicken has to be because I am saying that this is very good, but once it is go that customer that chicken, when the customer is rejecting their chicken, so there is useless. So I have to check the meat quality, And the sensory I have checked after the cooking of this meat, mostly the 50% showing giving the more red because the replacement of 50% showing the good flavor of the meat. But when you see the 100% of that meat, it is some fishy odor was coming. So I replaced that one. That 50% replacement can be go with the soya protein. That is a good, yeah. So now my curious was that I'm giving this hydrolysate. Does it is impacting on the microflora, gut microflora of chicken or not? Then I outsourced this KK. I collected the KK. Then I sourced this for the metagenomic analysis. And you see the one is a standard diet which is available in the market for the pig using the soya protein, and one is a 50% replacement of that meat protein, waste meat protein hydrolysate. When you see this is a Sankey chart. When you see the Corona chart, where you can see it is giving when you see the standard diet, the total lactobacillus is a 17%. But when you see this, it is a 33%. It says that the good bacteria is increase in their gut. So that can be used as a instead of like antibiotics, that can be it is good. So this some percentage we can increase the good micro in their gut. So this is the that result show that this can be helpful for their gut microflora. So now this is how to come as a conclusion that 50% because you know that this is what replacement I done with 50% soya protein is showing the all properties good one, and I can conclude that this can be replaced, 50% can be replaced, which can see once you replace this protein, definitely it will be the price of that feed it will be reduced because it is coming from the waste, and definitely once you are using the waste, ultimately it will be help in the cleaner the environment also. So this is my conclusion. So we can use this 50% the protein hydrolysis which come from the wastewater protein or slaughterhouse waste, that can be replaced, and that can help in the both as well as the economic as well as the health of the poultry. So I think this is the last slides for me. So thanks to my director because permitted to be present my some work in this, and again I thanks to my HODs, my students who are contributed for this all data. So thank you for all this, for your kind attention.
Thank you, sir, for sharing your expertise. Now the session is open for discussion.
Please, yeah. So please go ahead. Otherwise, my presentation very clear for you, I think, if you are not asking.
I think one suggestion is that don't call it waste. When you say
it is a
nobody
discard.
will buy.
Yes,
I know it's going to be cheaper when you say waste chicken, waste thing. I think it's a leftover.
Discard. It is called as a discard.
Don't say discard. You will say that leftover, unused chicken, unused fish or something. I think sellability will be more.
Yes.
The second one was you started with the malnutrition, and I guess that there is some study which is going on the malnutrition. Is this what is some trial which is going on? Are you using any of these for?
No, madam. This is not with that one.
So you are not focusing on the malnutrition right now for this product?
Just I am giving the alternative because many consumers, even Kerala, because traditionally they are giving the meat to children's because they are making the porridge from that out of, and they have to cook, and it is taking the time consuming. So they asked me, sir, give the packaged with this meat proteins, packaged foods.
No, it has improved. Yeah, it has definitely improved the, whatever you need in this. But you haven't focused on them because you started with the malnutrition.
Yeah, so that is given some.
So I was hoping to see something in those lines. Basically. Thank you.
I could see you have discovered some new novel peptide.
Yes,
novel peptide.
Peptide.
Your study, yes.
Yeah.
Did you patent it? If not, why not?
Patented. I published the paper.
You have published the paper. Okay, okay. And because these whatever hydrolyzed, if you feed into the biological system, it will be further degraded into some other forms by the gastric enzymes and all. Have you ever tried to focus on that aspect, like what happened, like once you treat?
Yeah, I tried. I done the GI, just in vitro GI. That's showing the good this one.
So are they
in vitro
remaining the same status? They are not undergoing further.
No, further I have not gone. Okay. My part is to be see the application just.
Okay, okay, all right. Yeah.
Thank you.
Any other questions?
How do you confirm that peptide is from the hydrolyzed, not from the bacterial?
Yeah, that is, I cleared first because whatever the antibacterial peptides are coming, it's not from the, maybe from the meat proteins, or it may be coming from the secretion by the probiotics. Yeah, that's coming, both are coming.
Thank you, sir, for answering the queries. I now request Dr. Rekha PD,
so mostly bacterial, that antagonistic peptides coming, maybe bactericin only. Yeah, because peptides not show the good antibacterial properties mostly.
I request Dr. Rekha PD, Chairperson MiCON 2025, to present a memento to Dr. Tanaji Kudure as a token of appreciation. Thank you, sir. Let's now resume the oral presentations. The chair for oral presentation is Dr. Tanaji Kudure, and Dr. Rukeya Sultana will be co-chair, and Dr. Asif Hamid will moderate the session.
Good afternoon, everyone. I think we can start. I would like to welcome Dr. Tanaji and Dr. Rukeya Sultana to this session. So the first presenter will be Dr. Bharati Prakash. She's from University College, Mangalore University, Mangalore. She'll be presenting on the topic Artificial Intelligence for Manuscript Writing and Research. Over to you, madam. Yeah. It's 5 minutes for presentation. Five minutes for presentation and 2 minutes for discussion.
Respected chair of the conference, dear organizing committee members, all the dear students and participants, delegates, a very good afternoon to everyone. From morning we have been witnessing quite a good research papers, good research work. So one of the theme of the conference was this one, artificial intelligence. So I thought some of the things which I have learned recently, I thought I will share it with you all. And we all know that change is the only constant thing, and if we don't change according to the change, the change will change us. So in this context, artificial intelligence is a new thing coming. I don't know, it's there in a computer science, machine learning, generative AI, lot many things we have been listening, but when it comes to life science, it may take some time. But nowadays there are many AI tools available which are very user friendly, and both for teachers, professionals, researchers, everyone. So about this, I'm just going to give you the insight. Actually, there are more than 100 such AI tools are available, which are very, very good, and it will make your work very, very easy, and it will save your time. But some of those which I have used have been listed over here, but there are still many more. So this whole process of research, writing the thesis or a project or a PhD work, for everything we need to go for the literature review first, and which is the most time-consuming task. You don't know where to start and how much to take it and how much to leave it. So we always say in just 10 years data that you have to collect for all these research review papers. So all these in the AI tool, some of them they are explained, which is there in bold. Maybe here we can't see that. Yeah. So whenever for these AI tools, when we give any command, it is called as prompt. So this prompt, like just it is just like when you go to Google and give search some word that you want or some thing which you are interested in the research or to find out the literature review. So that is nothing but the prompt, but it has to be very, very precise. So more the precise, your result will be more focused and more specific. So in this case, I'll read out some of them which are very useful. Lit maps is the one which gives you all the papers which are there in your area. So it makes your work easy. And these research AI applications or the AI tools that are used, that depends on which one you exactly want to use and which context, like maybe your research area. Based on that also there are many research like AI apps are there. Yeah, now Research Rabbit, Sourcely, Consensus, Copilot, Semantic and Scholar, these are the very user friendly, like even students can just learn and they are freely available. Now Elicit is the one which even I have used. So it gives the paper, the summary of the paper. Usually in abstract we find it, but altogether if you are going for many papers if you have given, and suppose there are some 100 papers are there, and which one to choose, and the overall summary of all these 100, it will be given by these research apps. So actually even in the Google if you give it, it will give you more than maybe 50, 100 pages. But you always think whatever information comes in the first page, that is the only information available. So we don't have patience to go to the second, third, fourth or maybe 10th page, we never go also. But there are many informations which are useful and needed. But this is what is our pattern of finding the literature in the Google. But these papers that will give you the first top. So you have to give the criteria. I need within 2010, I need the top papers, top 20 or 50 in this particular area. So it will selectively give you in South India, in North India, only in Asia or only in Europe, what the work has been done. So that is a very specific information you need. That is based on which area you are living and you are finding out the information which you need the most. And of course, once we get all the literature review done, there are some paid versions like Research Kick that is too good. So it will tell you in every aspect how you have to start from the beginning. So how to write the paper and how it should be. As we all know, in methodology, there are so many, small changes are there. Suppose there is one high-level biofilm research is there based on if you have a very good infrastructure, definitely you can do using all those. But very small labs, they don't have any such facilities. Suppose morning we were discussing that biofilm work was going on. In that, those who have minimal basic bench facilities, they will just do using congruent method or ELISA reader. So even if you go for microtiter plate method, ELISA reader has to be there or spectrophotometer has to be there. So, but if you do it using just a test tube like congruent method, naturally you can do it visually grading yourself what is a biofilm. Suppose such techniques we have included in our method, in our research work, and if we publish this in a very high quality journal, maybe Access Microbiology or many more, they are like high quality journals, definitely they will give a comment, no, it is not possible. So this is the, AI will tell you based on your methodology which are the best papers, journals that you can search and submit. Otherwise half the time goes into searching the journals and which to choose. Again some are paid, you have to pay for all those. Now SciSpace is another wonderful app where video abstracts will be given. So you don't have to do anything, you have to just give the paper, submit the paper, and it will give the abstract, because nowadays for many journals they ask for the picture abstract for that, so it will be readily available. So there are some, for this presentation, if you have to prepare slides, it will automatically give you all the flashcards and just copy and paste. See, in all this there is a role for ethics. You can't just copy and paste somebody's work and just give to AI and do this. Finally, when we come to the plagiarism, there is one software commonly used called as Rayan that will definitely detect, maybe there are some more other, where it is, whether it is copied, whether it is original work or just copy pasted. And there are some library of images, plenty of them available which are necessary for you. And another wonderful one is Paper Prime. As the name indicates,
please conclude. Ma'am, it's time over. Please conclude.
So students, if they practice well, your work will be really wonderful. And Jenny is the one which will write the paper itself for you. So you have to just do the work and submit it to AI. So it's user friendly, so researchers must use this one. Thank you.
Yeah, the topic is open for discussion.
Madam, do you think use of this area in manuscript writing and research is ethical? Is it ethical?
It is a aid to this. Like it's one of the software is EduAid. So these are the aids one, because finally we all who all are writing the papers, we are written many times, we have gained that experience, and out of that we are writing. But when you give most of your time to the methodology writing, doing work in the lab, and when it comes to the writing part, you get stuck. You have no guidance. So here it is just doing a job of guiding you, not that completely making you dependent on that. Maybe after 1 or 2 papers if you write, you may not even ask. You will know what to add and what to remove from that. So such, it is just an aid. It cannot be said that complete replacement of your original mental things, or intellectual parts of your research methodology.
No, is it ethical? That's what. Can we use that? Yes or no? That's what.
To some extent, yes. It is ethical. We can't completely replace the human brain, your originality, but we can take help and go ahead to save the time, because it is rat race and everybody wants paper and get their work published and everything. So
no, I don't agree that, because AI, we are doing the research because there is a real mind should be involved. If you use AI, it just replaces available literature only.
No, if that is the case, of course, when first time when you hear, we always think that yes, human brain, our originality should not go. Here also we are not disturbing your original research that is done, but so many tools are coming up just to help the students, just to save the time, and where more authenticated information you can get it in saving the time.
No, I agree with him, what he just said. I was just going to say the comment on that. See, scientifically, we are all researchers and scientists. These are all budding scientists. The way I used to do research in those old days is to go to the library, read the papers, read back references, because your curiosity, you want to know more, so you go back to the back reference, go back to that, and that we had to go and do. This is pre-internet, before any of these young kids were born,
even
right?
we all have done like that.
Right? So now with the luxury of having all these available, your abstracts are available, some of the papers, online papers are available. If really the young minds, they have to do the scientific, they have to do that. It's not just writing paper is not the end, right? They have done the experiment, they want to see what has been done by others, how they have done it. If you just ask, the AI-based tools or ChatGPT, it will give. Is that what you want? You want the curiosity to be there, right? So I think the young, all the research, I'm talking to all the researchers and scientists, I think they have to go and see what is there in that particular field, what people have done. They have to read the papers. From there they do it, that the scientific mindset, AI cannot replace. You cannot say that, okay, AI will write the papers.
No, that is too expensive
save
for
their time.
students. No, but what you said about the research paper, to read the paper, that reading is compulsory. Without reading,
with this they will not read. They'll say, okay,
no.
This is what I'm asking. Antimicrobial resistance, and then you'll say biofilm
No.
formation.
It will give the papers. You have to read, and a researcher has to decide which method I have to adapt based on what infrastructure I have. They have to read. Even when I made a project on this, it was so easy. Like in every paper, that material and methods was focused. So it was very easy. What I need exactly. So that is saving time. So reading is not at all replaced.
No, I know. But
that we have to do.
It's important for any researcher and scientist to actually go into because you can get this, abstract or the title of the paper which is relevant for your thing will be there, right? I mean, so that you will get it. I mean, if you search, you don't have to go use any of those AI tools. Then you anyway read that abstract and know whether this abstract is relevant or not, and so on, right? I mean, that's what.
No, in Google, if not AI, in Google also we are doing the same thing. We just put the command, we get some 10 papers. Unless we read, we can't take it. I mean, we can't include it in our. AI has.
You said for the paper
No.
writing and all. I think
That's an old style, because sometimes I also ask my students to print the copy, highlight, show me underlined version.
Exactly. The same thing is done by
And
AI.
don't come with just telling because we insist, because my students are nodding that how many original article you printed, highlighted the methodology. Show me that.
Yeah.
Because
In
that helps them to think, hand-eye brain coordination. Yeah.
And in Paper Digest, it will give you.
We had a wonderful discussion about this topic, and let's move on to the next presenter.
Okay, thank you,
madam.
Thank you so much.
So I call upon Nidia Muhammad Ashraf. She is from Yenepoya Medical College, Yenepoya deemed to be University, Mangalore, India. Number is MOP 7.
Good afternoon. I'm Dr. Nidhi and I'll be telling you a story today. So for that, I'll be taking you to our Optal OPD one day morning when a mother came holding the hands of a 14-year-old son from a village in North Karnataka. So here, this is. Good afternoon, I'm Dr. Nidhya, and today I'll be telling you a story. For the story, I'll be taking you to my Optal OPD, where one day morning a mother walked into our OPD holding the hands of a 14-year-old son. They both hailed from a village in North Karnataka, and she seemed extremely scared. Something was bothering her, so when she started speaking, we understood what her concern was. Her son, this 14-year-old boy, has been having pain in the left eye for the past 1 month and redness for the past 1 week. To make things worse, it had started developing in his right eye also for the past 3 days. He was been having severe pain, severe headache and fever, so bad that he had stopped going to school altogether. Thankfully, the vision was not affected in both his eyes. In spite of consulting 3 different hospitals in the past 2 months, from where he was prescribed topical antibiotics and artificial tear eye drops, there was no relief. Even a CT scan, which was done from a nearby hospital, showed nothing abnormal. So we had one big question in front of us. What is exactly wrong with this kid? So we started examining, and then we found out that both his eyes had redness around the cornea. This is what we call a circumcorneal congestion, which is caused by the inflammation of the ciliary body, which is a part of the uveal tissue. And then slit lamp examination, we saw that there were small deposits of cells and proteins on the cornea, inflammation in the anterior chamber, and because of this inflammation, this 14-year-old boy had started developing cataract in both the eyes. So we were all alarmed. We further investigated the case. We looked into the fundus, that is the inside part of the eye, and initially everything was normal. Optic nerve was normal, retina was normal, so we were happy. But then on further investigation, the small blood vessels inside the retina had started developing sheathing. This was because of vasculitis. And there were few exudates in the periphery of the retina. So now we understood this is not just an eye problem, there was something big in the way. We immediately admitted the child and we started our systemic examination to get a bigger picture of what was happening to the kid. The skin had dark spots, he had joint pain and inflammation, and he had fever and lethargy. So immediate pediatrics and rheumatic expert consultation was taken up, and then we ran the child on every possible test that was possible to us. It ranged from all the autoimmune diseases that was possible for us and infectious diseases. The time was ticking, we had to do something for the kid. We started treating him on hometropin and prednisolone and systemic steroids. But even after 12 days of admission, there was no change. We had two options, either treat him symptomatically or go further, dig deeper. But thankfully, we chose the second option. And on the third day, we had an answer to all our questions. His OX2 antibody titers in the blood was very high, about 1:160. And then we got a culprit in the story. That is rickettsial infection in a 14-year-old boy, caused by a tick bite and had wreaked havoc in his body. So we immediately started the child on systemic therapy of doxycycline and clarithromycin. And then we saw that the child had started becoming better. His inflammations was coming down, he was better. We had a happy child, healthy child, relieved mother and a happy team. We discharged the child on these medications, and on the first post-op visit, his inflammation was minimal. Even the cataractous lens had become crystal clear. So why am I telling the story here? Now, rickettsial inflammation in India, it is considered to be very rare, but it is not so. It's more common than we actually think. Second thing, it's better to go into rare diagnosis even when it doesn't fall into any of the common ones. We usually say the devil is in the details, but now we can say the tick in the forest can even start ticking. I would like to thank Dr. Ashwini from Rheumatology and Dr. Sahana, HOD of Pediatrics. Thank you.
Yeah, topic is open for discussion.
There were earlier cases
in India.
Yeah, okay.
Ma'am, in pediatric cases where kids present with uveitis, most of the time, I would say 80% of the time, it's an autoimmune cause. And then in a country like India, other cause, after you exclude all the autoimmune causes, we consider TB. So none of these, all the other investigations were all negative for this child. So that is why we had to take another extra rheumatology and pediatric opinion, and it took around 12 days for us to get an exact diagnosis. In India, scrub typhus is more common, but then spotted fevers are not that very common. We really don't have an exact number of how many cases have been reported, but now for us, we are all alarmed. Now we are investigating uveitis cases more, and we have new 2 suspected cases in the hospital.
Identifying tick bite as the thing. How did you treat that?
No. But after we getting the report, we started retrospectively asking the history. So the one complaint what the mother said was that this kid was always in the forest. They have a nearby forest. He is from a village area. We have 2 suspected cases, ma'am, not yet diagnosed. We are still investigating.
Is there any easy way of diagnosis? Directly you can?
No, ma'am, because skin lesions. Yes, ma'am. Scrub typhus has, they present with HR on the skin, but for this kid, there was no HR till then.
Any such cases reported in adults?
In adults, yes, ma'am. Adults, scrub typhus has been diagnosed in a lot of cases. The two cases which I've told, one is an adult man, one is a pregnant woman who we are suspecting of having rickettsial infection.
Yeah, ma'am, thank you. Because your presentation in the form of story, I like it really. So, does you people do like this story forms for other cases also?
Not really, but this was one particular case. We wanted to tell it as a story so that
very nice.
There was it. Thank you, sir.
Thank you, Lydia, for a nice presentation of a case report. Let's move on to the next presenter, that is Dr. Rama Bhatt, Department of Postgraduate Studies and Research in Biotechnology, Alvas College, Modabidri. The title of the topic is Isolation and Characterization of Calcium Carbonate Bioprecipitating Bacterial Isolates from Soils. Over to you, sir. Judges, please note this is MOP 8.
Respected judges, other dignitaries, my dear colleagues and dear students. So I am presenting a research work on isolation and characterization of calcium carbonate bioprecipitating bacterial isolates from selected soils. So there are a number of reports we have obtained using the different microbes, bacteria from soils, and they can be used for bioprecipitation methods. So the different adaptive factors and different environmental conditions, it may be acidic soils or even basic or other soils, it is possible to isolate this organism and utilize them for calcium carbonate bioprecipitation. So using ureolytic, these organisms are called ureolytic bacteria. We can use calcium carbonate in the media and they will produce urease, and thereby urease will act upon urea and it produce carbon dioxide and ammonia, and this carbon dioxide can able to produce the calcium carbonate crystals. So we can see the reaction in bacterial cell, what are the changes takes place, and ultimately the carbon dioxide will produce the calcium carbonate crystals by the presence of urease enzyme. So main objective, isolate the calcium carbonate precipitating microorganism, then characterization by morphological and biochemical methods, then estimation of urease activity by the selected isolates, then undertake the bioprecipitation of calcium carbonate in vitro, and then characterization and other properties. So we collected samples from Vamadapadavu, Jodraste and Venur, 3 places. Then we used calcium carbonate precipitating media, pH will be 7.5. Then identification based upon gram staining, then biochemical tests, then determination urease activity on bacterial stains using Nestler's reagent tests, then bioprecipitation of calcium carbonate by bacterial species using calcium carbonate bioprecipitating broth, 50 ml each we have used in triplicates, 7 days experiments using rotary shaker methods. Then characterization of calcium carbonate, whatever produced using scanning electron microscopy, then FTIR. Then other properties of this samples, soil samples were studied, determination of specific gravity using pycnometer, then liquid limit of soil by Casa Grande test unit, and shrinkage limit by shrinkage limit test apparatus. Then the results, so we got more than 10 types of different colonies. We used 10 to the power 3 to 10 to the power 4 dilution methods, among which we isolated 2, 3, 4 types of isolates from these 3 soil samples. Gram reactions, we got 3 gram positive and 1 gram negative, and shapes also 2 rods, 1 short rod and 1 round. So we can identify the colonies based upon shining appearance in the colonies. Then overall biochemical reactions, starch hydrolysis test, indole test and methyl test, which were used for the identifications. Then urease enzyme activity also we got in the isolate 4 higher enzyme activity. Then calcium carbonate precipitation of different isolates in broth. So we got higher in isolate 1.72 gram. Then specific gravity also among the soil samples, which are also in soil 1, 2, 3, we isolated for different soils, different organisms. So specific gravity among the samples also in isolate 5 we got higher, and liquid limit of soil samples in soil sample isolate 4 we got. Then shrinkage limit, which are also been higher in isolate 3. Then we also performed some analysis for control soil, and other 3 types of organism whichever used for the calcium carbonate crystal formations. So these are the different types we got. Then we also studied FTIR for functional group identifications using control 2, 3, 4, 5 isolates, and we have got different results and different functional groups, fluorocompound, alkanes, alkenes, alcohol groups, and different functional groups at different absorption waves. So we compared our the results with earlier results, which are also showed different quantity of calcium carbonate can be produced at different types of isolated organisms, which will be comparable with earlier experiments. And we are going through the identification of the organism. We only studied up to the gram staining and characterization, and earlier studies they concluded even Bacillus subtilis, Bacillus megatherium and Bacillus thuringiensis, which have produced higher calcium carbonate crystal formations. So by conclusion, so biological process and a natural process, the organism will produce the calcium carbonate by producing urease in their body itself. So different microorganisms can utilize urease activity, and they can able to produce this calcium carbonate in natural condition also. So we can produce calcium carbonate by using these organisms, which can help by studying their characterization properties, which can use for strengthening even basic foundation of even construction purposes. Thank you.
The topic is open for discussion.
Yes, sir. Sir, is there any other applications for using this urease producing your isolates? Any commercial, except this calcium carbonate precipitation, but apart from that, any applications are there for urease?
But it can be produced because urease have got higher turnover numbers, even enzymes.
Yeah, that's
so it can industrially it can be utilized.
Utilized. Okay.
Yes.
Sir, good presentation. I had one queries.
Yes, madam.
You have done FTIR, sir.
Yes, yes, madam.
Right, but you are just finding out the calcium carbonate precipitating, right?
Yes, madam.
So
Yes.
which way the other functional group you are comparing?
So we are using one control, madam.
Okay.
Comparing with the control.
Standard compound?
Yeah, standard yeah, calcium carbonate.
Okay.
Yes. Yes, yes. Calcium carbonate.
Which are having other functional groups?
Yes, madam. Yes. yes.
Okay, thank you, sir.
Sir, what was the here? So, what was the source of the precipitate you have provided in the media, and how did you solubilize it in media?
So media it will be produced for 7 days. We are keeping for incubation. So after 7 days we are filtering it using Wattman filter paper.
Okay.
Then we are drying it and going for
So
calibration.
you have removed all the insoluble calcium
Yes, yes,
carbonate
yes,
from
yes,
the media.
yes,
Then
yes.
you provide only the solubilized.
Yes, soluble.
form.
Yes.
Okay. Then you look for.
You are weighing and then going for analysis.
All right. Yeah. Okay. Yeah. I think we don't have any more questions. Thank you, Dr. Ramabhat.
I
we'll
thank
move
the
on to-
organizers for giving me an opportunity, especially Dr. Rekha PD. Thank you.
Yeah, let's move on to our next presenter. That is G.S. Shweta. She's from Department of Studies in Microbiology, Davangere University. The topic of her presentation is characterization of bioactive compound conferring antimicrobial activity isolated from lactic acid bacteria using Fourier transform infrared spectroscopy and gas chromatography, mass spectrometry. This is MOP9. Just please note this is MOP9.
Good afternoon, everyone. I'm going to present on characterization of bioactive component conferring antimicrobial activity isolated from lactic acid bacteria using FTIR and gas chromatography mass spectroscopy. Coming to the introduction, here lactic acid bacteria, commonly abbreviated as LAB, it represents a group of microorganisms, and these are the gram positive, catalyzed negative, and these may be the non-sporing and non-pathogenic. And these are classified under the phylum Firmicutes, that are class Bacilli, and the order Lactobacillus, and produce lactic acid as specific product of carbohydrates fermentation. And these lab are used in the food industries, result for their ability to improve the nutritional properties of foods and produce a large number of compounds with biopreservative potential such as enzyme or hydrogen peroxide, organic acids, and bacteriocins, etc. Coming to my objectives of my presentation, here isolation of lactic acid bacteria from food industrial effluents and identification of isolated lactic acid bacteria, screening of antimicrobial activity which shows the most zone of lactic acid bacteria, and characterization of selected lab from FTIR and for GCMS. For the isolation process, here we are collected the effluents from the different food industrial effluents, food industries. Here the effluents are collected and those are serially diluted and inoculated in the specific selective media, MRS agar media, and the selected colonies are selected, and those organisms again inoculated into the MRS agar, which is supplemented with the calcium carbonate. Here which shows the zone formation. Those isolates we are selected for the further process. And after the screening with the calcium carbonate, we are next go for the identification of lactic acid bacteria by the morphology and some biochemical test and by the other test. Next, we'll for the selected isolates, we did antimicrobial activity. Here the particular pathogen organisms are selected, and those were swabbed under the MHA agar medium, and the well were made, and the cell-free supernatant were subjected to these wells and incubated, and which shows the maximum zone of inhibition. Those isolates were selected for the further process, and those isolates were screening and sequencing are done. And then those isolates were further done for the FTIR and GCMS. Here out of 160 isolates, we have selected the 5 isolates for further process. And in the selective broth, we have inoculated the isolates and centrifuged and then subjected to the FTR and GCMS. Here in the FTR, some peaks were observed in the particular some isolates. Some peak was under the 3033 centimeter. Here some carboxylic amine groups are observed. And
hello? Audience, please maintain the silence.
next, we are subjected to the gas chromatography and mass spectrophotometry. Here, some volatile compounds or some other compounds are detected. In this 0 to 24 minute, we got the peak at 16 minute. Here, some of the compounds which are identified as pyrrole, todecane, heptanol, hexanol, 3-methyl, etc. Coming to the conclusion, here FTR analyses are provided insight into the functional group present in the bioactive extract, indicate the presence of hydroxyl, carbonyl and alkyl groups commonly associated with antimicrobial agents, and these lab as a source of antimicrobial agents provide a molecular basis of their activity. The findings a significant implication for the development of lab derived compounds of alternative to synthetic antimicrobial in food preservation and in other methods. These are my references. And thank you for the organized by the Division of Microbiology and Biotechnology, the MiCON 2025.
The topic is open for discussion. Yeah, first question is from my side. What was the purpose of using this FTIR and GC mass in your study?
To know which component we have, which compound is there for the further analysis or further procedure. We are producing for bacteriocins. So
what was the major compound identified based on your GC and FTIR analysis? What was the major compound? What was the potential compound?
Potential compound, just we identified as that hexanol and volatile compounds we are identified. But we required for the which related to the bacteriocins, so we didn't still get it.
Have you found any evidence for bacteriocin in your GC mass or FTIR?
No, sir, still we didn't get
Right,
it further.
So it has antibacterial properties.
Yes.
Have you tested it?
Yes, ma'am.
Against what pathogens?
Against
you had listed some of the pathogens, but you did not show the activity report, right?
No, ma'am.
Yeah, that's what, you showed the pathogens to which against which it was tested. Was it active against all the pathogens you have tested, or for only selected a few?
No, ma'am, only for selected.
Like
for
which one?
protease, Klebsiella and E. coli and Salmonella, I got the...
It was. What was the, you did a zone of inhibition or MIC?
In zone of inhibition.
Yeah, what you got it more than 16 mm. Okay, good. So what is your next plans?
Further, we are going to production of bacteriocins. Then we will combine with the nanocomposite. The further work is on that.
Your interest is in the volatile compound to show the antibacterial properties.
Yes.
Does it will work?
Sir?
In food condition? You said it will work in the food.
No. We are searching for the which bacteria will produce for the pediocin or niacin in that way. So we are we send for GCMS. Those components we may get in those isolates.
But you are saying that volatile compounds should be antibacterial or antimicrobial.
No.
Nice presentation, ma'am. So here again my doubt also same this thing. So are you looking for volatile components only?
No, ma'am.
Then why only GCMS? When we could go for LCMS, because GCMS will show only the volatile one.
Yeah.
So LCMS, and I would suggest you to do some normal chemical test.
Okay.
That could have been give you the ideas what are the components might be present.
Okay, ma'am.
Yeah, thank you.
Yeah, thank you, judges.
Thank
you, Shweta. Let's move on to our next presenter, that is Mrs. Akshata Rai. She is from a division of Microbiology and Biotechnology, Yenepoya Research Center, Yenepoya deemed to be university. Title of the topic is Antimicrobial Effect of Thymol against biofilm forming opportunistic pathogens implicated with chronic wound infection. Just please note this is MOP 11.
Good evening all. I am Akshita. My today's presentation topic is antimicrobial effect of thymol against biofilm forming opportunistic pathogens implicated with the chronic wound infection. These are the contents. I am going to present about the introduction, methodology, results and highlights of the study. Coming to introduction, microbial biofilm formation is considered as one of the worldwide challenge, as we all know, due to its antibiotic resistant pattern. There are, coming to infection, there are many type of infection we can see. Among that, more prevalent one are surgical, followed by lacerations, burns, pressure ulcer and other infections. When infection cause, the first week of infection, the reported bacteria, the more predominant one was Staphylococcus aureus, followed by Pseudomonas aeruginosa, Escherichia coli and other pathogens. At the initial phase of infection, in the first week of infection, we can see S. aureus, followed by in the second week Pseudomonas aeruginosa, and the other pathogens will cause sepsis in the coming stage. This is the methodology. I am just presenting a part of my work here. Today I am going to present about thymol. Thymol I used on 2 gram-negative and gram-positive bacteria here, that is S. aureus, E. coli, Pseudomonas and E. faecalis. We wanted to observe the growth inhibition, that is minimum inhibitory concentration of this thymol, followed by biofilm inhibition capacity and also its antioxidant and anti-inflammatory properties. Coming to the results, we have examined MIC, MBC of thymol against 2 gram-negative strains. Those are P. aeruginosa and E. coli. We observed minimum inhibitory concentration. We observed where there was no visible growth after treatment in 24 hours. And it is confirmed by taking reading at 600 nanometer. Where MIC of thymol against P. aeruginosa was observed as 1 microliter per ml. And in E. coli, it is observed 0.25 microliter per ml. MBC, minimum bactericidal eradication concentration, where there was 99.9% killing of bacteria was considered as MBC endpoint. Next, we have also tested effect of thymol against 2 gram-positive strains. Those are E. faecalis and S. aureus. The MIC of thymol against E. faecalis was 0.5 microliter, and it was against S. aureus it was very less, 0.125 microliter per ml. Obviously, MBC was at 2 MIC concentration, which was double than the MIC concentration. We have also tested biofilm inhibitory capacity of this thymol against 4 strains, and biofilm inhibitory capacity of thymol, we observed more than 95% inhibition at its 2 MIC concentration. Compared to growth, biofilm need 2 MIC concentration treatment, as it's very difficult to remove. We have also tested its antioxidant and anti-inflammatory activity. Thymol showed 74% activity, anti-inflammatory activity, and 69% antioxidant activity. These are the highlights of my study, where we have showed the MIC, MBC and biofilm inhibitory concentrations of thymol against 4 bacterial strains. These are the reference. Thank you.
Yeah, topic is open for discussion.
Thank you for the
Thank you.
Nice presentation. My doubt is thymol you have procured or you have extracted?
Procured.
Okay. What was the purity?
97%.
I'm not sure, but earlier it is published thymol as an
Oh yeah, mostly on antifungal.
antifungal.
And we are using it on broad spectrum of bacteria. Today I presented only 4, but many more, and even in combinations.
So do you know how it's become antibacterial? Any mechanism?
How it became antibacterial? It's reported already. I'm not sure about how. It has an antibacterial, antifungal, and antiseptic, and it's been used in food industry as a flavoring agent.
What are the natural sources of thymol? You know any natural
Yeah,
source?
aromatic plants.
Like?
Thyme, lavender, rosemary, like aromatic plants.
Mint leaves?
Yes, yes, mint
It
leaves
has?
too. Yes.
Any more questions from audience side? Okay, thank you, Akshata. Let's move on to our next presenter. That is Mr. Yuvarajan Subramanian, the Division of Microbiology and Biotechnology, Yenepoya Research Center. Topic of his presentation is urological metabolic conditions, alters regulation of quorum sensing system and virulence gene expression in uropathogenic Pseudomonas aeruginosa.
Good evening to all. My study title is Urological Metabolic Condition Alters the Regulation of the Quorum Sensing System and the Virulence Gene Expression in the Uropathogenic Pseudomonas aeruginosa. Coming to the background of the study, like Pseudomonas aeruginosa is one of the major pathogen which is involving in the complicated urinary tract infection in different urological disease condition patient, including the CKD, different types of urological cancer, emphysematous pyelonephritis, among others. So this pathogenic virulence mechanism, it will be differentially regulated by the altered compounds which is present in the
Audience are requested to maintain the silence, please.
In different urological condition, the metabolic profile it will be differed. It's like it depends on the elevation of the glucose and the creatinine, albumin, among and including the red blood cells. So this elevated metabolites, it will be differentially regulated the virulence expression by modulating the quorum sensing system. But the influence of the creatinine, albumin, glucose and heme in the Pseudomonas aeruginosa, it's still not comprehensively investigated. So hence we have investigated the system under the different simulated condition. For that we have isolated the different Pseudomonas aeruginosa strains from the urine of the patient who have different urological diseases with the metabolic complications, and further it was preliminary identified the Vitek system, and further it was sequenced and it was confirmed. Further, to check the phenotypic and the genotypic character and its expression under the simulated condition, the synthetic urine was simulated by adding excess supplementation of those metabolites, and further growth, biofilm, urease activity, elastase production, rhamnolipid production, including the QS regulation genes also investigated by using the QRT-PCR. So here we have noted that like among the pathogens, the urease positive pathogen, it showed the higher growth than the urease negative, and also all the tested conditions showed significantly higher growth when compared to the control, and particularly we have noted that under the albuminuria condition the growth was significantly lower than the control. Here C is indicating the control, GU is the glycosuria, CU is the creatinuria, and HU is the indicating the hematuria, by AU is indicating the albuminuria. So here we can see significant reduction in the growth under the albuminuria condition. And also further we have quantified the biofilm, and further it was processed the fluorescence staining method. So here also we can see clearly the strong biofilm formers also turned into the weak biofilm formers under the albuminuria condition. And also here specifically we have noted that strains isolated from the glycosuria showed the strong biofilm formation under glycosuria, and the strain the YPD Y76P and 205, which is isolated from the hematuria, showed significantly higher biofilm formation under hematuria. So its working depends on the strain and the metabolic specific manner. Further, the swarming motility also tested. So this is also aligning with the growth and our biofilm results. So here also we can see significant reduction in the under albuminaria condition, and glycosuria, hematuria, we have observed like same disease and metabolite specific manner. And here we have noted that interestingly, the siderophore production was significantly lower under the hematuria because of here the pathogens acquiring iron from like from the RBC. So here the siderophore production was like significantly lower than the control. And similarly, the rhamnolipid production and RHL IR expression also higher in the glycosuria and hematuria, and in albuminaria it was significantly lower. And further we have here noted the two different interesting results. The pill A expression was like depends on the strain and metabolite specific manner, but alginate production was differ from that. The ALGR expression was significantly higher in all the tested pathogens under the glycosuria condition, and PQS system is differed from the RHL and LAS IR system. So here also we have noted the pyocyanin, the YPD Y70P, it's not produced a significant pyocyanin production. However, the 76 and 82 it's produced the under disease condition, and this is the corresponding RHL IR expression. This was corroborating with the rhamnolipid production. And further elastase activity also, it's we have similar findings we have noted. And further, like urease is one of the major virulence factors to successful adaptation in the urinary environment. So hence we have investigated urease expression and the corresponding URC expression also. Here also the YPD 76, it showed the significantly higher URC expression under hematuria, and the 82 we noted under the glycosuria condition. So finally, the study results showed that the dominance of P. aeruginosa strains in patients with urological disease highlights challenge in clinical management, and it is indicating niche adaptation, and also albumin significantly inhibiting the virulence factors. Hence, it may further take into the to develop this treatment strategy. Thank you.
Yeah, thank you, Rajan. The topic is open for discussion. The first question is from my side. Yuraj, how did you replicate this pathophysiological condition in vitro?
Yes, actually like in normal urine, there are like traces of glucose will be there, and also there are so many reports, and depends on the like clinical reports we have added to mimic the glycosuria by supplementing the like 20 millimolar of glucose, that is indicating the like moderate to severe glycosuria condition. And also in if you are seeing the albumin condition, there are in urine up to 20 millimolar, 20 mg per deciliter of the albumin will be there, but when it's going about 20 to 200 mg per deciliter, it's indicating the microalbuminuria, and we have created the like microalbuminuria by supplementing the 200 mg of the albumin in deciliter. And also in if you are seeing the hematuria, like when we are observing in the higher magnification per field more than 5 RBC, if it is there, that is indicating the hematuria. So when we have tested at different concentration by adding the 0.2% of the heme, it's given the per field more than 5 RBC. So hence like we have used.
What was the basal media?
Basal media is like a normal synthetic urine without any elevation, this elevation.
And the pH?
pH is we have set 6.8, that is like normal pH of the urine.
Okay, after the addition of these component, are you did you set the pH, or pH still remain the same?
No. After addition of this component, we have set the pH.
Okay.
Yes.
Right. Any more questions from audience?
You collected the clinical isolates?
Yes, ma'am.
Okay. So I hope you must write the ethical number.
Yes. Yeah. Okay. Yeah, sure.
Yeah, thank you.
Study was conducted with the approval of ethics committee and medical SRB committee. Yes. Thank you.
Yeah. By this we have concluded our oral presentation for today's session. Yeah. Thank you very much for all the participants.
Thank you, presenters. Now I request Dr. Rekha Pedi, Chairperson MiCON 2025, to present a token of gratitude to Dr. Tanaji Kudre and Dr. Rukeya Sultana for chairing the session. And to Dr. Asif Hamid for moderating the session. And I also request ma'am to present a token of gratitude for the judges who judged for poster presentation. Dr. Yashodhar P. Bandari. Okay. Okay, okay. After tea break. Okay. Now let's have a quick tea break and come back to the most awaited part of the conference, that is award distribution ceremony. And I request all the participants to give the feedback which is pasted outside the hall on the wall. So please give the feedback.
Deleted my slide.
who has a hidden talent, who is going to sing a song.
Hari. Yeah. So I know most of you are very familiar with Bhava Gita. So Bhava Gita is one thing that normally we sing or we say through our heart, right? So it's a kind of feelings and emotion. So one Bhava Gita which was written by Dr. N.S. Lakshmi Narayana Bhatt, that is a very famous song, Ni Sigade Badondu Bade Krishna. I hope you are familiar, and I just try to sing two charanas of it, Charana and Pallavi. ಕಮಲವಿಲ್ಲದ ಕೆರೆ ನನ್ನ ಬಾಳು, ಚಂದ್ರನಿಲ್ಲದ ರಾತ್ರಿ ಬೀಳು. ಕಮಲವಿಲ್ಲದ ಕೆರೆ ನನ್ನ ಬಾಳು, ಚಂದ್ರನಿಲ್ಲದ ರಾತ್ರಿ ಬೀಳು. ನೀ ಸಿಗದೆ ಉರಿ ಉರಿ ಕಳೆದೆ ಇರುಳ, ಮಾತೆಲ್ಲ ಬಿಗಿದಿದೆ ದುಃಖ ಕೊರಳ. ನೀ ಸಿಗದೆ ಬಾಳೊಂದು ಬಾಳೆ ಕೃಷ್ಣ, ನೀ ಸಿಗದೆ ಬಾಳೊಂದು ಬಾಳೆ ಕೃಷ್ಣ. ಅನ್ನ ಸೇರದು ನಿದ್ದೆ ಬಂದು ಹುದುದೆಂದು ಕೂತಿಹೆ ಒಂದೇ ಸಮ ಕೃಷ್ಣ ಎಂದು. ಅನ್ನ ಸೇರದು ನಿದ್ದೆ ಬಂದು ಹುದುದೆಂದು ಕೂತಿಹೆ ಒಂದೇ ಸಮ ಕೃಷ್ಣ ಎಂದು. ಯಾರು ಅರಿವರು ಹೇಳು ನನ್ನ ನೋವಾ, ಯಾರು ಅರಿವರು ಹೇಳು ನನ್ನ ನೋವಾ. ತಳ್ಳನಿಸಿ ಕೂಗುತಿಹೆ ದಾಸಿ ಜೀವ, ನೀ ಸಿಗದೆ ಬಾಳೊಂದು ಬಾಳೆ ಕೃಷ್ಣ. ಒಳಗಿರುವ ಗಿರಿಧರನೆ ಹೊರಗೆ ಬಾರೋ, ಕಣ್ಣೆದುರು ನಿಂತು ಆರೂಪ ತೋರೋ. ಒಳಗಿರುವ ಗಿರಿಧರನೆ ಹೊರಗೆ ಬಾರೋ, ಕಣ್ಣೆದುರು ನಿಂತು ಆರೂಪ ತೋರೋ. ಜನುಮ ಜನುಮದ ರಾಗ ನನ್ನ ಪ್ರೀತಿ, ಜನುಮ ಜನುಮದ ರಾಗ ನನ್ನ ಪ್ರೀತಿ. ನಿನ್ನೊಳಗೆ ಹರಿವುದೇ ಅದರ ರೀತಿ, ನಿನ್ನೊಳಗೆ ಹರಿವುದೇ ಅದರ ರೀತಿ. ನೀ ಸಿಗದೆ ಬಾಳೊಂದು ಬಾಳೆ ಕೃಷ್ಣ. ನಾ ತಾಳಲಾರೆ ಈ ವಿರಹ ಕೃಷ್ಣ, ನಾ ತಾಳಲಾರೆ ಈ ವಿರಹ ಕೃಷ್ಣ. ನೀ ಸಿಗದೆ ಬಾಳೊಂದು ಬಾಳೆ ಕೃಷ್ಣ, ಬಾಳೊಂದು ಬಾಳೆ ಕೃಷ್ಣ, ಬಾಳೊಂದು ಬಾಳೆ ಕೃಷ್ಣ. ಥ್ಯಾಂಕ್ ಯು.
Before we move on to the award distribution, I request Dr. Rekha Pedi, Chairperson MiCON 2025, to give a token of gratitude to Dr. Ashodhar P. Bandari, Deputy Director, Yenepoya Research Center, and Dr. Zenica D'Souza, Professor, Ayurveda Medical College and Hospital, Yenepoya, for judging for poster presentation. If anyone is interested to give a feedback for the conference, please come forward.
Respected dignitaries, dear delegates and my dear students. So we had a wonderful session from today morning to till now with the different topic lead lecture presentations along with the posters and oral presentations. So this gives a immense opportunity for the younger generation how to go for even higher studies and research and what are the recent development in the biological sciences. So this is the 4th year the MiCON is going on, and even last year also I had been here along with our MSc students, which is an opportunity even for our students. They can showcase their project work as well as updated informations. Another important for even students and even younger generation, they can interact with the eminent professors as well as students from other institutions about the research as well as what are the available opportunities. So that can be utilized. So today we had a good hospitality and also good topic presentations and a wonderful session. I thank everyone for giving this opportunity to express my view. Thank you.
Good evening all. Thank you for being here for MiCON 2025 National Conference, and we also thank you other people, those are for the workshop. They are attending since yesterday, and they may be feeling tired for this MiCON, and it is coming to the conclusion. So please bear for a few moments more. And we are actually announcing awards for orals and poster presentations. Before that, actually we just summarize what happened in a day. So actually this day actually started with the furious talks of Dr. Sharmila Mandi, followed by oral presentations, that followed by Dr. Tanaji Kudre's talk, and later followed by the other oral presentations, and simultaneously our judges were evaluating the poster presentations also. We have two secret judges. They actually marked anonymously, they don't know you, and we are not given your names also to them. So they have judged you based on your presentations and what the quality of the work that is actually presented in MiCON 2025. So I request our team to bring the scores for the award presentations. Are you ready? Dhanyashri and Mayuri? Yeah. Just a few moments. Yes, yes. Yeah. They are actually doing your work. Just bear them for few moments. No, no, no. Yeah. Which one is done for poster? You can bring the results. Mega. Poster is done, you can bring that. Yeah. It is done? Yeah. This is for poster presentation, right?
Now I call upon Dr. Yashodhar P. Bandari, Deputy Director at Yenepoya Research Center, and Dr. Tanaji Kudre for awarding the winners.
Okay, we have results of our poster presentations. These are based on, results are actually compiled based on your poster presentations in front of the judges and marked by the secret judges. From that we have announced the 3 presenters, actually oral presentations, as of, poster presentations, first, second and third category. First prize goes to Miss Megha KS. She is from Yenepoya Research Center. Thank you, sir. Second prize goes to Megha D. Sawant. Institution name? Dharwad? Alwaz, Autonomous College. Thank you, sir. Congratulations to the award winners. Third prize goes to Prabhu Gauri. What is your institution name? Kasturba Medical College. Congratulations. Okay. Thank you, sir. No, we have oral presentations also. You can join, madam. Oral presentations. First prize goes to Urajan S. Yenepoya deemed to university. Congratulations, Urajan. Thank you, sir. Second prize goes to Akshata Rai. Congratulations, Akshita. Third prize goes to Efra Masum, the University of Transdisciplinary Health Sciences and Technology. Congratulations, Efra. Thank you, sir. Yeah. Yeah. Budding scientists were there. Yes, yes. Calculate that. Yeah, yeah. Yeah, we have a other award, that is Budding Scientist Award this year. So that Budding Scientist Award we actually are giving to the student who is just started their journey or research, or those are fresh MScs are actually working for their dissertations, or those are registered for the PhDs just in first year or about to register, or they have some piece of research, nice research work that can be presented at oral and posters. So this category comes under the Budding Scientist Award. Prajana VS. Prajana, correct. From Alvas College. Congratulations, Prajana. Okay. Judges and questions.
Thank you all for a very wonderful cooperation in this MiCON 2025. And whatever the judges' decisions have been taken, I understand that there was no other influence or anything in the decision making based on the scores given by the judges who were here as well as the secret judges who have evaluated it. And congratulations all the winners, and we look forward greater research aspirations of yours and greater success in coming years. And I'm sure you are going to join us in MiCON 2026 Yenepoya University with more research, more groundbreaking innovations for showcase in this audience. And we once again thank you for your kind participation and supporting MiCON 2025. And those who have supported in the past, we really are grateful to you, and we look forward for your continuous support in MiCON series of conference. And if you can give your honest feedback, and I think last year there was a feedback that we have to extend this conference for 2 days, but we did not execute it. Perhaps in 2026 we look that we will have 2 days conference with more number of abstracts, more number of research papers, and more invited lectures. And I'm sure you all enjoyed with us this conference, and wish you all safe journey back home, and wish you all the best for your future research works. Good luck, and see you again in Yenepoya.
With that, we conclude the National Conference MiCON 2025, Divergence and Surveillance of Microorganisms. Let us carry forward the knowledge and inspiration that we have gained today. Thank you all. All are requested to come forward towards the stage for a group photograph.